Okay. There's some inspection is going on, recovery
Okay.
is going on.
Okay.
USB가 이렇게 뜨는데 이것만 되면 되겠죠? 안 익혀가지고 이게 떠서 하고 있는데 여기 연결했거든요. 이게 오류가 왜 떴지? 오류가 떠가지고
The recovery of
Yeah.
USB.
Yes, right.
이거 어떻게 할 수가 없는데?
이렇게 다 되면 읽히겠죠?
And you can check the date.
Yeah. This one,
Yeah, right.
this one. I will replace your slide with this one.
So when I speak, I just use this.
Yes. Or
And how?
like this.
Yeah. And is any cursor? I use the cursor here.
Yes.
Can I use the laser? There's a laser.
Laser.
Yeah.
하나 둘 하나 둘. 하나 둘 하나 둘 하나 둘.
Can you help? Somehow the background. Can you just ignore the background?
Background?
You can. There's a function to ignore the background. Ignore the background somewhere. Can I read? Maybe the
okay.
last one.
Like this? Okay.
can I see? Is it the timing?
Uh,
20 minutes?
We're going to notice you the every 5 minutes and 3
5
minutes
minutes.
left.
5 minutes and 3
3
minutes.
and 1 minute. And
Okay.
right.
Okay, thank you.
Thank you.
되게 한데요?
계속 이렇게 되는데
무슨 소리 하는 거야?
이메일로 보내야 된다고 해야 될 것 같아요. 잠시만요. 왜 마우스도 안 되지? 안 돼요.
한번 큐.
안 되는 거죠, 이렇게?
네, 형식이 달라서.
그럼 메일로
네, 안 돼요. 이건 안 돼요. 네, 이건 안 돼요.
There's we can't.
Okay, okay. Mark we Indonesian what day? How do I do? Well, I can email you the file. It can be heavy, but I wanna check the format to be honest. I mean, whenever we open the file, they made in the Mac in Windows.
Okay.
Characters will change.
Okay.
So, how do I do? Let me check.
Ladies and gentlemen, we are now ready to begin the 17th National Cancer Center International Symposium. We kindly ask all the attendants who are still in the lobby to enter the auditorium and take their seats. Good morning, distinguished guests, speakers, and participants. Welcome to the 17th National Cancer Center Korea International Symposium. It is our great honor to host this event under the title, Innovating Beyond Cancer with Cutting-Edge Technology. My name is Yoonjung Choi from National Cancer Center Graduate School of Cancer Science and Policy. Before we begin, I'd like to make a brief announcement regarding CME attendance tracking. For those of you who want to receive CME credits, your attendance is recorded according to the lunch break session. For online participants, at the end of the morning session, please click the exit button and then click the enter again when joining the afternoon session. For on-site participants, please scan the QR code on your name badge at the registration desk in the lobby during the designated time. For more details, please refer to the guidance email sent prior to the event. Now, without further delay, let us begin with the opening remarks. It is my great honor to welcome Dr. Han Kwang-yang, President of National Cancer Center Korea.
Good morning, distinguished guests, colleagues and participants, especially Dr. Chen from Taiwan, Dr. Okazaki from Japan, and also Dr. Masaoka from Japan too. It is my great honor to welcome you all to the 17th National Cancer Center International Conference. This year holds a special significance because National Cancer Center celebrates its 25th anniversary. Over the past quarter century, we have made remarkable strides in cancer control in Korea. Thanks to our comprehensive cancer registry, nationwide prevention efforts such as anti-smoking campaign, and the successful implementation of national cancer screening programs, these achievements have elevated the Korean cancer control system to one of the best in the world. This progress would not have been possible without continuing commitment of our government, the dedication of so many healthcare professionals in Korea, and the strength of Korean medical system, particularly its accessibility and the responsiveness. Today we gather here to explore new horizons in fight against cancer. Promising frontiers, including today, advances in proteomics for personalized oncology, breakthroughs in immunotherapy, including innovative cell-based therapies, updates on clinical trials, and the strategies to enhance cancer survivorship, ensuring quality of life beyond the treatment. I would like to extend my sincere gratitude to all distinguished guests and the participants joining us today. I hope today's conference will not only inspire new ideas and collaboration, but also reaffirm our shared commitment and the sense of camaraderie in our ongoing mission to conquer cancer together. Thank you very much.
Thank you very much for your inspiring words. Please give him a warm round of applause again. We will now begin the first part of today's symposium, the keynote lectures. The session will be chaired by Dr. Gun-guk Lee from National Cancer Center Korea. Please welcome Dr. Lee with a warm round of applause.
Good morning, everyone. Welcome all of the who are attended this meeting. I'm so glad to pleased to able to contribute as a moderator for this first plenary session, and I'm also grateful to have Dr. Henry Rodriguez as the first keynote speaker of this symposium this morning. Actually, Dr. Rodriguez is not a very famous person, so I don't have to introduce him in detail. But I just want to mention he's a very active member of the NCI, the founding director of the Office of Cancer Clinical Proteomics Research, and director of the Antibody Characterization Program, senior scientific officer, and senior leadership board member of NCI. And he is a very important person working at the White House for the precision medicine in the Biden, in the period of the President Biden. And I just want to mention his career, and he studied as a director and PI at the National Institute of Standards and Technology from 1998 to 2006. Although he's not unable to be here, he will present his lecture through prepared video clip, and then participate in an online Q&A session after the lecture. And he will give us the lecture titled as Collaborations and Breakthroughs in Cancer Research, Bridging Observational Proteogenomic Studies and Clinical Trials. Please start the lecture video, please.
Good morning. My name is Henry Rodriguez, and I'm the founding director of the National Cancer Institute's Office of Cancer Clinical Proteomics Research. I also serve on the National Cancer Institute Senior Management Board, and most recently held the role of Assistant Director for Strategic Health and Cancer Science at the White House. And it is an honor to be participating and delivering today's keynote address at this year's Korea National Cancer Center International Symposium. And for today, what I'm going to do, I'm going to speak to a topic that is very dear to me, and that is the topic of precision medicine in the space of oncology. But more specifically, what I'm going to do, I'm going to describe what I believe are ways of accelerating the field of precision medicine. And that is by taking the rich history of the genomics landscape, but now beginning to infuse within it simultaneously the high throughput based measurements of proteins. And at the same time, what I'm going to do, I'm going to weave into the fabric of my story today the importance of team science in a collaborative manner. Because I believe, again, that's one of the best ways to accelerate discovery-based science, translated into a preclinical environment, and now actually moved into clinical trials. Again, I will give examples to each one of these. So with that as a backdrop, let's begin. If I were to ask the average individual, define for me what you think is precision medicine, I would not be surprised that the majority come back and they simply will easily say, it is the field of our genes. It is genomics. And I'm not surprised because a lot of the marketing that has now been taking place for the past 25 years, but at the same time, it did have a jump start to other omic-based fields. But specifically in the field of oncology and cancer, today we recognize that cancer, yes, it is ultimately a disease of the genome. And major strides have been made with a genomic-based approach. Now, one of the metrics that we could utilize for this, that you could actually truly now begin to quantify the impact of precision medicine, you could actually now look at, here in the United States, our cancer mortality rates. So what exactly do I mean by that? If you look at the left side of this figure, so prior to 1991, the overall cancer death rate was always steadily rising. But after 1991, the overall rate has been falling. And it's been falling on average of about 1.5% per year, which I should actually note that from 1991 to today, that drop, that actually translates into an estimated nearly 3 million lives now that have been saved. However, the really big news is that it turns out that rate actually fell 2.3% from the period of 2016 to 2017. To be clear, that represents the largest single one-year drop since 1930. And while this progress is going to be mainly driven by declines in death rates from the 4 leading cancers in the United States, and that's the figures on the right-hand side, lung, colorectal, breast, and prostate cancer, turns out declines have dramatically accelerated for lung cancer, which in the United States happens to account for almost 25% of all cancer deaths. So the question is, why? What is going on with lung cancer? So for that, we need to do a little deeper dive into the data. And fortunately, this was actually published. It's 2 articles that I'll be referencing. The first one came out in the New England Journal of Medicine in 2020, and the other one came out in Cancer Discovery in 2023. So while we know that smoking cessation has played an important role to decline in lung cancer deaths since 1991, the real exciting news, it turns out, when we looked at the data, is that it turns out advances in targeted therapies is actually what's contributing to the sudden rate drop, specifically in non-small cell lung cancer, where targeted therapies actually became available in 2013. And we started utilizing them where we started targeting EGFR in first-line therapy. These are compounds such as Tarceva and Iressa. And those are the 2 red arrows that are shown on the left-hand side. Now, I should also note that these data points that I'm showing on this slide actually don't take into account immune checkpoint inhibitors that target proteins such as PD-1 and PD-L1. So even more exciting, one could even now begin to expect further declines in death rates from non-small cell lung cancer in the future years. But with all this sort of exciting news, the reality is what we know is that although some niche applications have been produced by precision medicine, the reality is that the full potential, that promise, the promise of precision medicine on public health, have relatively been small. And you can see this now sort of being played out within the public. Here's a couple of articles from STAT and another one from the New York Times. And they're basically arguing, well, why is precision medicine taking so long? And what they mean by so long is that on the right-hand side is the way that it was presented to the public. So from a history perspective, the first draft of the Human Genome Project got released in 2020. But if you read all the articles that came out in the way that this was rolled out to the public, they basically argue, the scientists, is that it was predicted that this first draft would transform medical care by 2010 in a span of 10 years. This is now 25 years later because now we're in 2025. And the question is, has it truly transformed medical care? And that's the concern that some people are asking. Why is it taking so long? And I think part of the reasons is the way that it was sort of tried to make it understandable to the public, but also the way that they've used biology as a whole. Again, I always go back to the way that I learned biology and the way that I learned my biochemistry. Mother Nature, biology, is very complex, and there's a lot of areas of redundancy, and that redundancy enables it to be very elusive. But the way that the genomics was presented is that the view of biology was greatly oversimplified. One of the arguments that it made is that the majority of genes, they happen to have a preset function that could easily be determined by simply looking at your DNA sequence. Today, we know that is not the case. So if you look at the area of what we would now call new biology, I think it's time that a lot of us, we begin to understand, again, biology is complex. We need to admit genes are not the sole blueprint for life. There's functional products, such as proteins, metabolites. Disease is complex. And in my view, the more that we can measure at the molecular level, the better off we're going to be to understand the underlying biology and take that underlying biology and translate it towards clinical care. So the National Cancer Institute has been making strides in this area. In this slide, I'm actually showing sort of a chronological timeline, greatly oversimplified, but I'm trying to basically help people understand the challenges that were faced and the opportunities that arose from that. Again, going back in the year 2000, the first draft of the Human Genome Project got released to the public, and that was a catalyst for change, a great cause. Now, that also gave birth to our ability to conduct high-throughput protein-based measurements using specific types of technologies or platforms. And 2 years later, investigators then released a study strictly now focused on protein-based measurements, really not even referencing it back to the genomics-based information from trying to understand the biology. But the argument was this was an early detection marker study, and it focused on ovarian cancer. And this got released in Lancet. And I believe the claim was that these investigators were able now to detect early stage ovarian cancer with greater than 95% sensitivity and specificity. Now, this raised great interest within the cancer community, such that 3 years later, the National Cancer Advisory Board at the National Cancer Institute actually put out a working group that focused on biomedical technologies. And they said, in this era of our ability now to molecularly characterize cancer at such depth, DNA, RNA, and at the protein level, what should we do in the field of oncology? And the recommendation that came back to the National Cancer Institute from this advisory board was, you need to move forward on a very large scale, taking all cancer types and molecularly characterize them at the DNA, RNA, and at the protein level. And to empower the cancer research community, make all that information available. Very powerful recommendation. But what happened was that same calendar year then, investigators at the Believe in Texas, they actually then looked at that study that came out in 2002 on the early detection biomarker study on ovarian cancer. And they basically said, we believe that the study lacks rigor and reproducibility. They identified what they believe were errors within that study, that it lacked standards, that it lacked proper experimental and statistical design. So one of the things that they requested was to make all the data at that time available to the public. Once they did, they recognized that the way the study was put together, the way the power calculations were made, the way that these samples were collected, the way the samples were analytically processed, the algorithmic tools that were used to interpret the information, there were errors within that. And unfortunately, a lot of those claims did not hold out. Now, the National Cancer Institute was involved in this whole area and they took notice. But to their credit, they still believed that the future would be molecular characterization of cancer. So 1 year later, what the National Cancer Institute created were 2 initiatives to tackle that approach. In the genomics landscape, the community had a lot of trust when it came to the metrology of high throughput DNA and RNA-based measurement platforms. So they launched the Cancer Genome Atlas to begin to catalog all the perturbations within the cancers, the various different types that are there, and at the same time to make that information available to the public. But when it came to the protein-based measurements, because they lacked that confidence of the analytical and of the computational, they lacked the rigor and reproducibility, they decided to take a different approach. Specifically, first begin to standardize these very promising technologies, and then once you get able to standardize it and demonstrate the rigor and reproducibility, move forward with biology. And that program on the top right of that slide is referred to as the Clinical Proteomic Tumor Analysis Consortium, or CPTAC. So this is sort of a structure of the CPTAC the way that it exists today. But our core mission has not changed for the past 17 years now. And that is to support the standardization, the application of proteomics, and now an era that we've been pioneering, the fusion of every patient sample that we obtain to be conducting comprehensive DNA, RNA, and protein sequencing simultaneously, that we define it as proteogenomics, to taking this science and moving it in cancer research. And we operate in 3 areas: discovery, translational, and now working collaboratively with clinical trial studies. But at the same time, our core mission, we want to accelerate science all around the world to empower the cancer research community. So one of our core missions is still and will be to develop public resources, again, to way to accelerate hypothesis-driven science by others. And we have different types of resources that we offer to the public. In addition to our data sets, we also have target-based assays, we have physical reagents, and a variety of computational tools. The bottom of the slide kind of shows the structure of the program. It's an academic-based program. It involves numerous academic institutions here within the United States. It's team science at its finest, and it's very interdisciplinary. We have individuals that are basic biologists, we have clinicians, we have translational scientists, and so forth. But the right-hand side is something that I'd really love to kind of discuss. CPTAC, the program, operates on 5-year cycles, and I'm very proud that the day that the program was created, we take every lesson that we learn in a 5-year period, and then we apply it and we evolve, we move forward. And this is sort of a cartoonish way of kind of viewing it. The very first 5 years of CPTAC conducted no biology. Strictly it was to demonstrate whether or not high throughput protein-based measurement platforms could actually be reproducible amongst multiple laboratories, and not just within the United States, but even on an international level. And we demonstrated it could be. The next 5 years was dedicated to sort of an experiment. If you were to take cancers, tumors that were already genomically characterized, and you layer protein biology above it, do you identify new biology that was missed or simply could not be obtained through genomic-based sequencing technologies? We demonstrated yes. You identify new biology that is not easily discernible through either DNA or RNA-based sequencing platforms. Once we did that, then we began to expand. The next iteration for the next 5 years involved a tumor characterization program, very analogous to TCGA. The distinction now, we would take tumors along with normal adjacent tissues, comprehensively characterize it, give all the information, the molecular data back to the public. But we also started to piloting translational studies, where investigators then would conduct preclinical research that in the context of a clinical trial question. And if the data would be meritorious, then they could potentially gain access to samples from clinical trials. That proved quite successful, where today we've expanded the tumor characterization program, and we've also expanded the translational clinical trial program. At our laboratories, they operate both in GLP, and now we do have a CLIA certified laboratory. So, focusing on the tumor characterization arm of the program. This has really now been in operation for approximately 8 years. And in 8 years, the question is, how many cancer types have now CPTAC, the tumor characterization arm, been able to comprehensively characterize and give all that molecular characterization back to the public? This slide sort of gives a snapshot. To date, we've now have put out what we define as a flagship study. It's the very first time that we characterize a tumor, and then after we interpret it and we publish on it, all the data is made available to the public. And to date, we now have 16 flagship studies that spans 12 distinct cancer types. The other thing that's quite nice about this now is that we could begin to emulate what we call pan-cancer studies that were very common within the genomics community. And it begins to understand the similarities and the differences amongst different cancer types. We started this approximately in 2023. Actually, we started in 2022. The first publications were released in 2023. You can see that on the bottom left of the slide, and there were 6 papers that came out. In 2024, we had an additional 5. And now in 2025, already 2 have been fully published, we still anticipate about another 3 to wrap up this calendar year. But this represents, to our understanding, the largest multi-omics cancer study to date on a pan-cancer level. Again, all of these publications, we looked at over 1,000 patients. We spanned 10 distinct cancer types, and we looked at DNA, RNA, protein, and images, H&E stains from the pathology lab, and DICOMs from the radiological side. Now, one of the things that people might ask is, well, you conduct a lot of these tumor characterization, and these are observational studies. Are there instances where these observational studies have been able then to influence and launch a new clinical trial? Yes. Here's just one example that I could speak to. So this one focuses on pediatric cancer, and more specifically on brain cancer. From a big perspective, it turns out there are just a little bit over 100 different types of childhood brain tumors, many of which there are few or literally no treatment options based on their genomic understanding. So to tackle this problem, there was an outside research group that's referred to the Children's Brain Tumor Network, and they approached the CPTech program. This group traditionally conducts genomic characterization. They heard about our program and they asked, would we be willing to collaborate with them and conduct protein characterization? So the study that we ultimately decided to partner on, it ended up characterizing a little bit over 200 tumors, and it spans 7 histological types of brain cancers. Now, for background perspective, for context, it turns out pediatric, if we focus on craniopharyngioma patients, those individuals, they happen to harbor a beta-catenin gene mutations. But adults that happen to have that cancer types, they don't have beta-catenin gene mutations. They happen to have BRAF mutations. The result of this at a clinical level is that BRAF inhibitors are simply not a treatment option now for these children. However, what this study was able to find was that it turned out that we were able to identify a subset of pediatric craniopharyngioma tumors that it happened to have shared similar protein signatures now and potential therapeutic targets, very similar to pediatric low-grade glioma patients that happen to harbor a BRAF mutation, even though those individuals, those pediatric craniopharyngioma individuals were wild type now for PRAF. So the clinical implication of this, which is actually quite interesting, is that now based on protein expression, is that the signaling networks in pediatric craniopharyngioma and in tumors with BRAF mutations, those actually now appear to be related and potentially now could be treated with targeted therapies such as MEK inhibitors, for example. And let me just say that RNA-based clustering, which is information that has previously known, we also reconfirmed it, RNA-based clustering did not capture these novel subtypes. Now, on the bottom of that slide is the very nice part. This information ultimately got presented to clinical trialists at the hospital in Philadelphia, and it ultimately now provided brand new clinical support that now led to development of a brand new phase 2 study investigating now a combo treatment strategy for pediatric craniopharyngioma children. Now, in terms of the broader picture of that translational research space of the CPTech program, this sort of now begins to capture the various activities that we've been conducting. Again, this initiative has now been around. We've just wrapped up the 8th year of the program. And in the first iteration, which is shown on the bottom of that slide, it was sort of this learning process. We conducted a lot of successful preclinical projects, but one of those studies was able to tap samples from a clinical trial. The top represents now the studies that we've conducted in the past 3 years. And as you can see there, out of the 5 publications that we can now go public with, 4 of those studies have now successfully been able to tap samples from clinical trials to further their studies, to further add value to those clinical trial studies that were all strictly historically genomically driven. But even from a broader perspective, CPTech is now starting to even partner with a lot more clinical trials. And this is shown within this slide. But the part that I simply want to emphasize is you could see CPTech from the preclinical moving into samples from clinical trials. We're now partnering either within our own initiative or with outside groups that would like their samples from the clinical trials that they've run, historically simply looking at DNA and RNA-based measurements, and now they would like protein measurements labeled on top of that. You could see ours, they straddle the full spectrum, all the way from the phase 1, moving up to a phase 2, and even now, just more recently, just published 1 week ago on breast cancer, samples from a phase 3 clinical trial. The other part now that is for me very exciting on how people see this idea of taking DNA, RNA, and now infusing it with high throughput protein-based measurements, whether it be in our GLP laboratories or in our CLIA laboratories, is outside groups that are starting to approach us to further conduct research in a collaborative manner. On the left-hand side, this demonstrates studies that we officially got launched in 2024, and a lot of these are now within the pediatric oncology space. We have a brand new study with a foundation, OligoNation. We have another one with the Linden Light Collective Foundation, and these are both focused on brain cancer. One looks at ODG, the other one looks at LGG, and we have a collaboration with the Children's Hospital of Philadelphia, which is an N of 1 project, which we will be working very closely with the Tumor Molecular Board. And brand new collaborative studies that we're conducting. One focuses on breast cancer. These now directly involve samples from clinical trials. That's a collaboration with Washington University in St. Louis. And another foundation that now we're working very closely with is the Beat Childhood Cancer Foundation at Penn State. And this one focuses on CNS tumors. And again, this one is also quite nice because here the information that we're going to generate, we plan to go back to the Tumor Molecular Board. These trials, they're already closed, but we're going to go back to the board with our protein information integrated with the genomics information and try to determine in a mock setting, would we or would the clinicians have made a different call for the treatment that were administered to the children, now having the protein information interconnected, very well intertwined with the genomics knowledge. So now let me shift gears a little bit. Let me focus on the other aspect that I think is very important on moving this field forward, and that is collaboration in a team-based structure. All the work that I presented up to this point involves laboratories situated within the United States. But I think one of the great things, and one of the reasons that I'm very proud to be giving this keynote address, is the value and the importance of even international collaboration. And one of those activities that we now have launched within the United States is a program that's referred to as the International Cancer Proteogenome Consortium. And I'm listing this slide because this is the very first public event that the program got kicked off. And this was in 2016, and at the time, it involved about 14 research institutions that span 8 countries. And a big thank you has to go to Dr. Yudu Chen, her colleagues at Academia Sinica, which has also helped to spearhead not just the launching of this initiative, but in the belief of being able to infuse and integrate genomics with proteomics. A big thank you has to go to her, that I'm fully aware that she's also speaking and she's participating in this conference. But as you can see on the left-hand side of that slide, we also had colleagues from the Republic of Korea, which have always played a tremendous role in the expansion and in the success of the International Cancer Proteogenome Consortium. And how is that expansion and how is that success has played with colleagues in the Republic of Korea? Well, that's actually shown within this slide. In this slide, it shows that all the way from 2016, the first institute that signed on was the Korea Institute of Science and Technology. And you can see subsequently other institutes have joined, such as Korea University. NCC joined, I'm very proud, in 2019. And as you can see, we've had DGIST and also with Kyunghee University. But what was quite nice, which is shown on the right-hand side of that slide, in 2023, the very successful and fruitful collaboration that started initially with the CPTAC program and also NCCK in 2019, further expanded in 2023. And there's a signing ceremony that we held in the United States. In 2023, we had the Deputy Minister Lim, at the time President So from NCC Korea, Dr. Kim, who was at the time was the director of the Research Institute, also at NCC Korea. And of course, I have to give a huge thank you to Dr. John Bay Park, where in 2019, he played a key role in actually bringing NCCK first into the fold of this International Cancer Proteogenome Consortium. At the time, played a very significant role at NCCK. And a huge thank you also has to go to NCCK, because every year, the International Cancer Proteogenome Consortium, we bring all the investigators from all the various collaborating partners together to discuss science, to discuss the progress that we've made, to also understand how we could further collaborate in new research activities. And in 2023, we were very fortunate that NCCK was willing to participate as the host of that event. And that took place in September of 2023. And there's a beautiful picture that you could see, not just the people interacting together, but the spirit of and the belief that by fusing genomics with proteomics-based information, we're able to move the science even further out. So how much science has now successfully been produced by this program? Well, here's a great example. The International Cancer Proteogenome Consortium got created, as I indicated, in 2016. To date, 16 flagship studies have now been released by this consortium that now fully spans 13 distinct cancer types. And there's over 25 research institutions involved that spans now 14 countries. Other examples of importance here in the United States that I like to talk about, our office, we have other partnerships with federal agencies. One involves the National Cancer Institute, the Food and Drug Administration, and also HRSA. And the idea there is to facilitate the development of point-of-care diagnostic testing devices. We also have another one that involves the government of the United States and the European Union, and that specifically focuses on cancer health cooperation. We have one on lung cancer, and another one focuses on childhood cancers. And we also have numerous public-private partnerships that's shown with the individuals on the bottom right-hand side of that slide. In terms of the resources now that we've been able to deliver to the public at large, I'm not going to give you all the statistics that's shown on this slide, but the point is that CPTAC, along with our international collaborators, we have molecular and clinical data that's available to the public at the DNA, RNA, protein, and at the image level. We also give away targeted-based assays. These are all of our standard operating procedures from a high-throughput protein-based platforms that can easily be downloaded into one's platform and run those experiments. And some of our assays do require a higher level of sensitivity. So for that, we have affinity reagents. All that is made available to the public. And one of the other things that we're now starting to do is we're starting to ask the question on a grander scale. What are the areas of barriers that still exist to move molecular-based diagnostics forward? And this is a nice summit that myself and colleagues, we held in Philadelphia last calendar year. And the whole purpose was begin to identify what those roadblocks are, and they begin to identify what potential ways of overcoming them could be. And what we plan to do in the next year is hopefully to write a white paper on this and release it to the public. And lastly, what I would like to emphasize is if people would like to understand this vision of fusing together proteomics with genomic-based information dedicated to the field of oncology, I encourage people to read this perspective. It was written by myself and my colleagues, and it got released in 2021 in the journal Cell. And I like to leave people with the following. I've always believed in this proverb, and this happens to be an African proverb. If you want to go fast, you could easily do it and go alone. However, if you really want to go far and you want to have that impact, you need to work as a team, go together. And if we do that, I ultimately believe that precision medicine will reach that promise, and hopefully within a short time. So thank you again, everyone. And at this point, I will be more than welcome to take questions from the audience. Thank you again.
Thank you very much, Dr. Rodriguez, for your very comprehensive review and the emphasis on the international cooperation in the CPTEC and the IPCP activities. Are there any questions? Please, Dr. Park.
Thank you, Henry. Thank you very much for your wonderful talk, and also I really appreciate your leadership on our Cancer Proteogenomics Society. I have a very simple question about, so as you mentioned, we are actually approaching precision medicine based on proteogenomic measurement. Is there any idea how we can leverage the current data sets to discover new molecular targets in rare cancer studies? Can we use current data sets to increase our understanding for rare or understudied cancer types? Thank you.
Any other questions? Let's go ahead. Dr. President Young.
Dr. Henry Rodriguez, I wonder if you can hear me? Yes, this is Han Wang-yang. I'm a new president of National Cancer Center Korea. I want to first congratulate on your great success in this long endeavor, and to echo to your mentioning about National Cancer Center Korea, although Dr. Park, he just gave a comment or question, he left, but he's inside Korea at Kyunghee University, and our National Cancer Center Korea has still strong commitment working together with NCI, and I expect your continuing international leadership in this long our endeavor. I would like to thank you for your kindness. Thank you.
Any other questions? We also have a long history of collaboration with Dr. Rodriguez and NCI from 1916. we also very successful confirmed the successful projects with them. And yet, unfortunately, we don't have enough time to discuss thoroughly. So I'd like to close this session. Thank you very much. Thank you very much.
We greatly appreciate Dr. Kwang-Luk Lee of the National Cancer Center for chairing the keynote lecture. Please give him a round of applause, please. We will now move on to session 1, From Code to Action, Proteomics in Translation. Please welcome the co-chairs of this session, Professor Kwang-Pyo Kim from Kyunghee University and Dr. Ho-Shin Kwak from National Cancer Center, Korea. Please give them a warm round of applause.
Audiences, this is the first session of our symposium titled From Code to Action. I think that this title is very appropriate. In other words, from genomics to proteomics. Proteomics, cutting it the state of art, it has a many high resolution and do some translational works, brilliant. So I'm Dr. Kwak from NCC and Kwang-Pyo Kim from Kyunghee University. And let's begin. And first two presentations will be moderated by Dr. Kim. Yeah.
Thank you. This is Kwang-Pyo Kim from Kyunghee University. I'm so honored to be a chairperson in such a prestigious events, and also it's a big honor to introduce my old friend Yuju Chen. Okay, Yuju Chen has a very long history of resume as leading science societies, including World WHO President. And also, she is serving as various important journals as editorial board or chief editorial function. And also, she is one of the world leader in the proteogenomics research, especially in the lung cancer. And her leadership is expanding more understanding into the very complex disease, lung cancer. Today, she is presenting her very recent researches. The title is Evolution of Micro to Nano Proteomics Toward Precision Oncology. Please welcome Yuju Chen.
Thank you very much for the invitation from the NCC Korea. It's actually I don't even remember how many times I have been in Korea, but this is the very first time I have been in NCC. And since my visit about 10 years ago to Korea, I really see very promising development and evolution of proteomic technology to the impact of the society here. So big congratulations to the proteomics team in Korea. And I'm actually a chemist. So whenever I need to speak in the clinical conference, I'm always very nervous. And my research focuses on technology development for proteomics. But since few years ago, like Henry just introduced, I have the opportunity to launch a Taiwan Cancer Musha program in Taiwan. And that really changed my whole career. I really realized that proteomics could be a useful technology if we work on the right direction, if we work as a team. So today I'm going to share with you about my journey. How does the proteomics work with the clinical community, especially on the cancer precision oncology? We probably know that the cancer, for example, is a very complex disease, and there's a lot of factors to contribute the initiation progression of the cancer. For example, if we look at the different population, we do have the different genetic background, and that may impact the initiation and the phenotype of the disease. On the other side, we also have very different lifestyle. For example, in Taiwan, it's basically a non-smoking country. We don't have many people for smoking, but this is an important etiology factors in the Western population. And we also have very different food style. Like last night, I tried a very spicy, not so spicy food in Korea, but that's actually my favorite food. But the food style may also change the phenotype of the disease. And finally, in Asia, we do have the factors for environmental carcinogen. And this is unfortunately a very important issue to the region. So if we want to really understand whether we are able to have a new technology or new solution to the precision oncology, I think the most important thing is to understand what does it happen to individual patient. And also keep in mind that we live in the same sky, we live in the same countries, but not every patient develop cancer. So that means there are some endogenous factors to cause the susceptibility or the prevention of the cancer. So under these many factors, proteomics is actually a very powerful tool because it provides the system view on the network of the protein, which is the molecule that executes the cellular function and also important for initiation and the progression of the cancer. If I use the lung cancer as one example, EGFR is the most widely used target therapy. And under the EGFR molecules, actually the protein phosphorylation is the one to really indicate whether the patient has activation or inhibition with the therapy. And outside the extracellular domain, the protein glycosylation may also likely change the phenotype. In the case of the PD-L1, the glycosylation is really important. And so this is a very nice tool to tell you when the patient has any change, for example, the mutation on the EGFR or the mutation on the KRAS. This is a very nice tool to tell you what is happening. And this is a real data from several patients who develop third-generation resistance to the third-generation tyrosine kinase inhibitor. And you could see that a lot of change associated with the glycosylation and further translate into the intracellular protein phosphorylation. So this would be the tool to tell you what is happening for individual patient. And in 2016, like Henry just introduced, I have the great opportunity to be the partnership with the ICPC to push the proteogenomic technology into the clinical community. And I spent about 1 year talking to the different government, except the Ministry of the Military Affairs. And about 1 year later, we are lucky to get the funding. So on the left-hand side, you can see that these are all the clinical leadership in Taiwan. And on the right-hand side, we have the team members from the different kind of technology, genomics, proteomics, and the data informatics. And I want to take this opportunity to emphasize the importance of the teamwork. And I have a lot of team project, but this is a very, very unique project. For each cancer type, we only work on one direction, single direction, no different sub-project. So the idea is the clinical leader raise the questions. They tell us what are the unmet needs. And we are the worker to find the technology to solve the answer, to answer the questions from the clinical team. And once we get the data, once we have the interesting finding, we come back to talk to the clinicians. They are the ones to make the decision. What are the important questions that we should continue to work together? So this is a very, very inspiring process for me. When we started, we actually do not know how to do together. I know proteomics, but I never heard about somatic mutation. I still remember I tried to Google somatic mutation. I don't understand, even I understand every Chinese word. But luckily, our very first study focusing on non-smoking lung cancer has a very fruitful result. This is the first cancer type we try to focus. The most important question we want to understand is what are the etiology, what are the molecular mechanism for the non-smoking patients to develop lung cancer. And I don't have time to go through the detail today, but I can just tell you that by comparing the genomics profile, proteomic profile, I can tell you that we are very, very different compared to the TCGA cohort, who are mostly smoking male. And we also discovered the endogenous environmental carcinogen associated with the female, the young female, and the older female. So the environmental carcinogen is extremely important. I will elaborate a little bit later. And the topic I'm going to share today is we actually found that proteomics is a very, very nice tool to closely reveal the phenotype of the patient and the clinical outcome. So following this, we have some very exciting development I want to share with you. Before I go to the detail, I want to share with you a very long-standing question about lung cancer. Currently, the genome typing on EGFR is still the gold standard for the treatment. However, if you look at the gene typing alone, does EGFR typing tell you about a clinical outcome? The answer is likely not. For example, if we look at the two major mutation types for lung cancer, the L858R and deletion 19 are the two major types. And Dr. Yang, the leaders of the Taiwan oncology, so Dr. Yang, President Yang told me that the L858R patient has a higher tendency to develop brain metastasis, and the patient also have the earlier response, earlier development to the recurrence and the resistance to tyrosine kinase inhibitor. So in summary, the patient of the AFIR has a poor survival compared to the patient from deletion 19. But they do not know why it's happening. So clinically, they still have the similar treatment. And this is a big question for lung cancer community. So we are wondering if protein is the molecule for target therapy, if protein or the phosphoprotein are the one to regulate the initiation, progression of the cancer, whether we are able to utilize the technology to identify a better approach to see what are the current status of the patient. So we just use the proteomic profile and unsupervised clustering to see the similarity of the proteomic profile for the patient. And based on this result, we have the three groups. And this is a prospective study for early stage. But you can see almost all the patients after stage 2 all clustered in the green subtype. And all the aggressive phenotype, including the pleural invasion and the lymph node metastasis, are also in the green subtype. But on the other side, the red and the blue subtype are clinically defined stage 1A and 1B. So this seems to suggest that the green subtype may be an aggressive molecular subtyping. So we come up with the idea to combine the clinical staging and the molecular staging into 5 stages. So for example, if the patient in the stage 1B are in the green subtype, that means they have the similar profile to the late stage patient. So we call this kind of subtype, we call late-like. That means they have the protein profile very, very similar to the late stage. So when we do this new classification, I immediately discovered that almost all the EGFR L858R patients are in the stage 1B late-like. But if you look at the stage 1A, they don't have the trend. So we start to wonder whether this is likely the point that the two types of the patients start to be different. And we consult another independent cohort, and you could see that L858R does have a poor survival compared to the deletion 19. And this difference was not observed in the stage 1A. So this seems to suggest that the EGFR mutation type may be different on the stage 1B. But after the paper was published, I was actually very nervous because this is a prospective study. We don't really know whether the late-line subtype really developed early recurrence. And 4 years later, we got the follow-up data. And you could see that the purple color means recurrence. It's so surprising to see that the 1B late-line subtype has very, very high recurrence, 43%, almost similar to the patient in the late stage. And the stage 1A late-line subtype is even more amazing. It's more than 5-fold higher recurrence rate compared to the molecularly defined good stage 1A. So this really confirmed that proteomics may be a good predictor for the early recurrence for the stage 1 patient. And for your information, that the stage 1B is a troublesome stage. There's no standardization about the treatment and also the drug for the stage 1B. So we think that this is an important point that we have to define to identify this high-risk patient for AT treatment. So we actually spent several years of the effort trying to verify the biomarker from tissue all the way to the plasma, which is more accessible for the clinical practice. And we are very happy to see that we have 4 protein panels which can predict the late-line signature, the recurrence of patient for the stage 1. And now we hope that this can be pushed as an IVD kit, but it's not ready yet. The antibody is so troublesome. And so we are also now thinking whether mass spec could be another approach to identify more biomarkers for this kind of similar purpose. I think the most exciting part is following the finding of our study. Taiwan actually has a consensus meeting from the resectable early stage non-smoking lung cancer. And based on our finding, they determined that we should have the need for biomarker for the stage 1A and 1B. So I think this is an interesting example that the new scientific finding is likely change the clinical mindset and the setting. And we now have an independent study with the 250 patients. And we also registered for the early clinical trial. So we are really hoping that this assay could be the very first one to identify the late-line patients for AT treatment in the futures. And the next question is whether the late-line subtype is Taiwan-specific or it could be present in other cohort. And so we have a collaboration with the CPTEC program from the Broad Institute about 2-3 years ago. This is an extremely painful process. We have weekly meeting for nearly 2 years, weekly meeting every week. And so we hope to push to see whether we are able to see the difference and the common features about lung cancer globally. So this is the largest cohort as far as I know. And I just want to show you some of the exciting results. So we also identified the late-line subtype in the global cohort. And you could see that even in the stage 1, the late-line subtype really have the poor survival compared to other subtype. And you could see the blue color here. These are all the stage 1 patients. So this means that the late-line subtype is likely a general subtype globally. And when we look into the detail about this late-line subtype, you could actually see that the environmental carcinogen plays an important role. So for example, you could see the different colors. We find out the smoking males from the Europe and the US has more susceptibility to the nitro pH and the pH, which are related to air pollutant. And for the Asia male, they have the more susceptibility to the nitro pH, which is actually more toxic compared to the pH. And finally, it's kind of interesting to see that we have a lot of Asian females, mostly from Taiwan, and they have more susceptibility to the green colors and carcinogen nitrosamine, which is commonly used in the food industry. So I think this emphasizes that environmental factors are extremely important in Asia and also to the Western population. And by using proteomics, we are able to dissect the molecular mechanism of this late-line subtype. Initially, it's kind of interesting to see that these two groups of the patients are totally different, male and the female. And EGFR mutation, KRAS mutation, they are totally different. But you could see that these are the common pathway related to more aggressive lung cancer. But on the other side, you can also see the difference between the male for immunoproteasome activation, but for male, it's only the standard proteasome activation. So this is a very fresh result we just published, and we are hoping by this molecular profile, we are able to suggest the best treatment for this late-line patient in the near future. So based on this result, we are so excited that proteomics should be very useful. But if you look at the sensitivity and the current technology, it's probably not sufficient. For many, many patients, especially the late-stage patient, there's no surgery. So we can only have the biopsy sample, FAP tissue, and also if you want to look at the tumor microenvironment, the sensitivity extremely important. So this further inspired my team to develop technology to the very small scale. I call microscale, nanoscale, that means a microgram, nanogram. So this is the first paper we published for single-cell technology, but the story behind this invention is not about single cell. Initially, I want to develop fully automated protein chip for patient analysis because I believe the robustness is important in the clinical setting. So we try to implement the proteomics station in the micro device that can be fully automatic. And at the end, we find out that at a single cell level, this is an excellent technology for the very small scale sample. And even at a single cell resolution, I'm so surprised to see that many, many molecules can be observed for the EGFR pathway. So that means this important protein could be observed at a very small scale. And for immune cell, you also see a lot of important molecules, for example, BCR or the kinase at a single cell level. So I think the technology is sufficient for research, but whether it's useful for clinical setting still depends on the application later. But for me, I believe that protein modification is probably more useful because most of the target therapy are targeting kinase. So protein phosphorylation should be more important. But if you look at the protein phosphorylation, it's only 1% of the protein. And it also takes more technology and more steps to enrich the phosphopeptide or the modified peptide. So if you look at the literature about the technology, the blue color are the single cell development. There's a lot of exciting publication. But if you look at the phosphoproteomics, the pink color, the most sensitive is only about 100 cells. So from 100 cell to the single cell level, there's a huge space to further overcome. And we are working hard to push the microfluidic device into the phosphoproteomics. And from 10 cell to 1,000 cell, again, you could see very, very exciting data for the EGFR, for the non-small cell lung cancer pathway. So for example, for EGFR target therapy, the tyrosine these two sites are the activating site to indicate the activation or inhibition of the patient. And you can see these two sites are there at the 50 cell. And for EGFR, for ERPB2 is the same. You are able to see the activation site. So we further push to the single cell. And even the coverage is not that good now, but we are still able to see a lot of drug target. And finally, we want to see whether this is likely useful. So we try to apply the technology to the patient who develop resistance after third generation of the OC treatment. We want to see whether we are able to use this kind of technology to suggest alternative drug. And if we are able to do that, what is the best way to guide the selection of the treatment? So we apply the technology at the 10 cell level to analyze 3 patients from the late stage patient. And it's interesting to see that even the 3 patients are clinically similar type for the lung adenocarcinoma. But we find out that one of the patients actually developed the transition to the SCCC subtype. And we further use the IHC to confirm that this patient has a very minor component of the SCCC. So at a very small scale, you can actually see the heterogeneity or the transition of the subtype change for individual patient. And even the 3 patients are similar, but you could see that they are so different, especially for this specific this specific patient, she developed the YAP1 resistance. And finally, the most important job is to see whether we are able to identify the drug. So from that specific patient, we actually see the activation of the EGFR and the ERBB2. So this is the primary cell from the impura invasion. So my colleague further helped us to verify whether the obtainable targeting the EGFR and ERBB is likely useful for the patient. So you can see that the data looks very nicely that obtainable is able to inhibit the primary cell from the lung cancer patient. But the most exciting part is here. So we further apply this, the change of the drug to the patient using the obtainable. And you could see that the patient has a stable disease for a few months after the change of the treatment. And even though, unfortunately, she still developed resistance, but I think this is one of the examples to tell us that the new findings or the new technology by using phosphoproteomics is likely a good indication to suggest the patient for the next line treatment. And in Taiwan, we have the highest number of such a patient who developed resistance. So we are hoping and we are working hard to push the technology to guide the next line generation for these terminal patients who are going to die very, very quickly. And finally, this is my encouragement for my team and also for everyone sitting here. I was very fortunate to be the keynote speaker in European lung cancer oncology last year. And this is very interesting experience. When I was invited, they already gave me the presentation title, Proteomics, not Proteogenomics. And they also gave me the title, Lung Cancer in Never Smoker. I was hesitating whether I should accept the invitation because it's a big meeting for 4,000 people. But finally, when I went there, I was so happy to know that for the clinical community, they have the high expectation for proteomics. And almost every talk is about biomarker. But the biomarker always show in the last slide the future perspective for biomarker. So I think the biomarker for different purpose of the different stage of the patient is urgently needed. And that could be a good direction for proteomics to go. But again, I think proteomics is useful for mechanism, but we really have to push the proteomics in the real world, in the clinical setting, in the hospital. So I think we have to work hard for harmonization, for regulatory consideration. And I want to thank my colleague. And also, again, I think the teamwork is extremely important. I really enjoy the journey, from the integrated work from the clinical community and also the basic technology providers. And this is really the last slide. I personally do not believe hero, but I think the teamwork is going to make great. And for the CPTech program, I really enjoy I want to thank Henry to give us a long-term support, not only for Taiwan, I think for the global community. And the lung cancer study is a joint work with Mike July's team. And these 2 years really give me a lot of inspiration. So I'm happy to be here today. I'm also hoping that we can keep up the collaboration in Asia. We have similar patients, we have the similar problems. So I think we should work together as a team to really give the benefit for the patient. And thank you very much. And your comment will be welcome. Thank you.
Thank you, Yuzu. Our schedule is a little bit behind, so if you have any questions, just one questions. And also we have discussion sessions after all the presentations, so we are going to use that time too. Any questions? Okay, thank you, Yuzu, again. Our next speaker is Professor Sang-won Lee from Korea University. He is also world-leading scientist in the proteomics area, and especially he was like a leading scientist in the two very important proteogenomics research in Korea, including early onset gastric cancer and pancreatic cancer. Today he is presenting his research on translating molecular subtypes into precision oncology in pancreatic ductal adenocarcinomas, implication for a proteogenomic basket trial. Please, Sangwon. Please welcome Sangwon Mi.
Thank you, Professor Kang-Pyo Kim, for the nice introduction. Today, I'll be discussing about our precision oncology, development of precision oncology for pancreatic ductal adenocarcinoma, with a particular emphasis on its implications for proteogenomic basket trial. So previously we carried out proteogenomic characterization of pancreatic ductal adenocarcinoma. Very briefly, in this study we analyzed 150 PDAC patient tumor tissues and generating mRNA sequencing data along with the global and phosphoproteome data. And based on this mRNA sequencing data, three distinct subtypes were identified. So, for example, this 461 transcript was particularly highly expressed in a patient group of RNA2 compared to other subtype patients. And this RNA2 exhibited the greatest resistance to the standard therapy compared to other RNA1 and RNA2 subtype. For the same patients, the global and phosphoproteome data identified 5 different subtypes each. And then we performed integrative clusterings using this individual clustering result. And we identified 6 distinctive proteogenomic subtypes. So by adding the data, we can should I do this? We could more precisely stratify the patients. So as you can see here, RNA1 subtype remained as a single subtype in the proteogenomic subtypes, and RNA2 divided into 3 different subtypes, and RNA3 divided into 2 different proteogenomic subtypes as we add proteomics data. So, and as you can see here, among these, subtype 2, 3, 4 emerging from the RNA2 exhibited significantly poorer survival compared to other subtype RNA, subtype 1, 5, and 6. And especially this RNA subtype 4 shows the worst clinical outcomes under standard treatment regimens. And these 6 proteogenomic subtypes were associated with distinct cellular pathways and protein core networks. And given that this each subtype was associated with distinct protein networks, it is the development of the subtype-specific therapeutic strategies will likely be essential to address the molecular heterogeneity of PDAC. So, in order to test this hypothesis, we try to establish patient-derived cell lines by using these 150 PDAC tumor tissues. Luckily, we successfully constructed cell lines that represents all the 6 PDAC subtypes. So this actually allowed us to investigate the subtype-specific drug responses in vitro, which we did. So using this subtype-specific cell lines, we conducted high-throughput drug screenings using clinical compound library comprising of 676 compounds. By actually the unsupervised clustering of the drug response profiles lead to identification of subtype-specific compounds. For example, these 11 compounds shows a very specific response to the subtype 2 cell lines compared whereas these two compounds showed very specific responses to subtype 4 cell lines and so on. So, and I also like to emphasize that all these compounds were never previously been utilized in PDAC treatment before, and never been evaluated in clinical trials for this disease. Several of these compounds shows very potent effect with the EC50 value in nanomolar ranges. So indicating that their efficacies, high efficacies to specific subtypes. So we are currently working with pharmaceutical partners to advance the development of the subtype-specific therapeutics strategies for this PDAC, and ultimately we anticipate that these efforts will lead to companion diagnostics for PDAC, enabling selection of specific drugs for the specific, based on specific subtype informations. So even if we get all the subtype-specific therapeutic drugs, we still lack this part, which is the method to core proteogenic subtype informations of the patient enrolled coming to the hospitals. So in order to address this unmet need, we developed what is called subtype identification technologies for PDAC or PDAC SIT. So to develop this method, we searched our proteomics data for subtype-specific proteins and peptides by using PLS analysis and by referencing the already constructed subtype-specific protein networks. This approach led us to identify 153 what is called subtype representative peptides, whose specific expression among the 6 subtypes should have enough statistical power to distinguish subtypes. So we then synthesized heavy isotope labeled peptide counterparts, which we call cell peptides, of these peptides in-house, and followed by the purification and the amino acid analysis to ensure the purity and accurate quantifications. We then formulated a peptide, PDAC cell peptide mixtures, which is optimized to generate robust and reproducible signals from the MRM assays. So we did MRM experiments on the leftover samples from our proteogenomic studies, and with this mixture spiked in. So to make the long story short, Professor Dave Huang at Seoul National University did unsupervised clustering using the MRM data, and during the course he was able to refine the peptides panel into 82 subtype representative peptides. And at the same time, he was able to develop subtype identification algorithms that uses MRM data as an input to call the subtype. So also, in order to translate our PDS set into a clinically applicable platform, we also developed dual online micro-LC system, which is capable of completing one MRM assay in 15 minutes. And we integrate this LC system with a medical grade triple quadruple mass spectrometer. And using this platform, we performed MRM experiment again. But and then Professor Huang processed the data using his algorithm to predict the subtype information for each sample without prior analysis. So during the course, he was also again was able to further refine the final peptide panel into 77 peptides, and as you can see here, our final PDS set was able to predict PDC subtypes with over 90% accuracies. So now we can use this technique to analyze patient samples to call the patient subtype information, and then we can treat the patient with the correct specific therapeutics. As I have demonstrated today, each cancer patient should be treated based on the underlying molecular mechanism driving their diseases. So, and at the same time, over and over again, we also learned that different cancer types, such as PDAC and gastric cancer and others, also shares common key oncogenic proteins and signaling pathways, which actually provides potential opportunities for therapeutic strategies that transcend traditional tumor type boundaries. NCI match is a basket trial approach that tries to seek a match seek a match for a patient with a specific treatment based on the specific genetic changes, no matter the cancer types. So based on, and then according to their publications, about 6,000 biopsy samples will be genome sequenced to guide the treatment selections. So based on this concept, build upon on this concept, we initiated our proteogenomic basket trials here in National Cancer Center in Korea. In this trial, we are aiming to seek a treatment selection that is guided not only by genomic sequencing but also quantitative proteomic profiling. So in this trial, we are going to collect biopsy samples prospectively from the patient who are enrolled in our clinical trials, basket trials. So in the first phase, we actually prepared the sample preparation SOPs that uses this cryopulverization. This SOP, we found out that this SOP provides a high quality DNA along with enough peptides for proteomics from a very tiny biopsy samples as small as 3 milligrams. We also developed advanced analytical platform that combines our mutually independent dual align nano-AC system with the orbital astral mass spectrometer. This platform, as you can see here, allows identification of over 14,000 protein groups within 3 hours using as little as 2 microgram of tissue peptides. Into this 2 microgram of tissue peptide, we also spiked cell surrogate peptides that is representative of the known target proteins of the investigational drugs that in our basket trials. And as you can see here, along with the very extensive proteome informations, we can measure the target amounts directly from the tumor tissues, as you can by comparing the area under the curve of the potential profile between cell and endopeptides. So we are going to, in this trial, we are going to combine, integrate this quantitative proteomics informations with genomic data derived from the whole genome sequencing experiment. And this combined proteogenomic information will guide the selection of personalized therapeutics regimens tailored to each patient molecular profile. And I'd like to finish my talk by acknowledging my collaborators whose contributions were essential for this work. And here are some funding informations that made this work possible. Thank you for your attention.
Thank you, Simon. We are going to have a question and discussion session after all the presentation. Okay. Thank you.
The last speaker is Dr. Woo Sang-myung. Dr. Woo is a clinician like me and he graduated Seoul National University Medical College and trained in Seoul National University Hospital. He's working as a biliary tract specialist from 2008 in this Institute of National Cancer Center. He's doing visiting scholar in UC San Diego. And in his outlook, he's an ordinary man and he's a man with many humor, but his excellency is in his research work. Today he will present us his research work that the subtyping biliary tract cancer and draw the conclusion this subtyping is related to drug sensitivity. Please start, Dr. Wu.
Thank you for kind introduction. Good morning, everyone. It's a great honor to make a presentation in front of distinguished audience. My presentation consists of 3 parts. First, I'll discuss the complex and heterogeneous nature of biliary tract cancer and limitations of current treatment approaches. And then I'll review our research result regarding proteogenomic approach for stratifying biliary tract cancers into clinically relevant molecular subtype. And finally, I'll highlight some innovative precision medicine strategies we are exploring utilizing patient-derived organoid and high-throughput drug screening platforms. Biliary tract cancer are a heterogeneous group of cancers that arise in either intra or extrahepatic bile duct or gallbladder. Intrahepatic cholangiocarcinoma develop within the liver parenchyma, whereas extrahepatic cholangiocarcinoma involve the biliary tree outside the liver. The global mortality data shows the steady increase in biliary tract cancer deaths over the past 3 decades. According to the Global Burden of Disease 2019, biliary tract cancer was ranked 16th its number of cancer deaths, approaching 170,000. The incidence of biliary tract cancer varies among population of different geographic origins because of differences in risk factor. With the pink color are represented area, the with lower incidence, less than 6 per 100,000 cases, whereas in green color indicated the biliary cancer, in which the incidence of biliary cancer is not rare cancer. Especially in Thailand, China, and Korea, the incidence of cholangiocarcinoma is very high. The primary sclerosing cholangitis is a well-known risk factor in Western countries. In contrast, hepatolysiasis, stone disease, liver fluke, calorie disease, and HBV infection is the more frequently observed in Eastern countries. Biliary cancer is the ninth most common cancer in Korea, with about 8,000 new cases in 2022. 5-year survival rate was about 30%, with only 4% in distant stage. This cancer shows the striking regional disparities, up to 2.3 times higher incidence in rural southeastern provinces compared to metropolitan Incheon. The chronic cystitis is the most common human liver flu in East Asia, including Korea, and the chronic chiasis and cholangiocarcinoma have a positive cross-relation. The liver basin with high infection rate of the cholangiocarcinoma patient involved the Yeongsan, Hyeongsan, and Nakdong river. And these regions shows also high instance of cholangiocarcinoma. There are distinct mutational patterns in liver fluke-related and non-infection-related cholangiocarcinomas. This whole exome sequence data from Singapore group shows the BOB1 were more frequently mutated in non-ovarian cholangiocarcinoma, whereas the TP53 shows the reciprocal pattern. These findings indicated that the different causative etiology may induce distinct somatic alteration even within the same tumor type. Although the molecular genetic characteristics differ in the biliary subtype. Intrahepatic cholangiocarcinoma, the rate of potentially targetable genetic alteration differed in biliary cancer subtype. Whereas the intrahepatic cholangiocarcinoma harbored recurrent mutation in FGFR and IDH mutation, and whereas extrahepatic cholangiocarcinoma and gallbladder cancer have the ERBB23 amplification. The anatomic classification can be ambiguous due to the anatomic complexity. The anatomic margin for distinguishing intra and extrahepatic bile duct are the second order bile duct. However, it is very difficult to distinguishing intrahepatic cholangiocarcinoma with secondary involvement of hilar bile duct from extrahepatic cholangiocarcinoma with liver parenchyma invasion. The classification based on gross morphology can be bettered. The best point type forms a deposited mass in the liver parenchyma, whereas the periductal infiltrating type is defined tumor that extending longitudinally along the bile duct, and the intraductal growth type forms the papillary growth inside the lumen of the bile duct. Mass forming type is the most common subtype. However, the combined growth pattern, the mass forming type and periductal infiltrating type is commonly encountered in real practice. Currently, the surgical resection remains the only potentially curative treatment modality, but many patients develop recurrence. In addition, the limited number of patients can be candidated for local therapy. Therefore, the systemic cytotoxic chemotherapy is an inevitable choice for most biliary cancer patients. However, there are many controversies in non-surgical management regarding the role of adjuvant therapy, radiation therapy, or standard chemotherapy option in the management of biliary cancer. Since 2010, during 15 years, the gemcitabine plus cisplatin combination chemotherapy has been only one cytotoxic chemotherapy option in patients for this heterogeneous disease, regardless of their diverse etiologies. In the landmark trial, the median overall survival was 11.7 months in gemcitabine-cisplatin group compared with 8.1 months in gemcitabine-only group. The 3 months survival extension is modest at best. 3 years ago, the superiority of adding durvalumab, immune checkpoint inhibitor, to gemcitabine-cisplatin, GP versus GP alone was shown in phase 3 2+1 trials. The median of survival was 12.8 months with durvalumab plus gemcitabine-cisplatin versus 11.5 months with GP alone. The clinical benefit over GP are not satisfactory. Stratified medicine can be a bridge to precision medicine. Stratified medicine based on the identifying subgroup of patients with distinct mechanism of disease or particular response to treatment. The stratified medicine located between conventional and precision medicine allows the healthcare providers to make more informed decisions about which treatments are likely to be effective for each subgroup. Our research team proposed a personalized medicine strategy for intrahepatic cholangiocarcinoma, identifying 3 groups through comprehensive analysis of DNA, RNA, protein, and metabolite. This is a workflow of our intrahepatic cholangiocarcinoma proteogenomic study. DNA, RNA analysis included 83 patients and 88 patients respectively. Protein analysis included 101 patients. We identified 2 prognostic subtypes based on the transcriptome profiling. The results of our transcriptomic clusters are consistent with the previous study. RNA subtype with poor prognosis showed KRAS mutation, high tumor marker levels, and enriched with inflammatory pathway. In contrast, RNA subtype with good prognosis revealed IDH1 mutation, high viral hepatitis, and enriched with metabolism pathway. Proteomic approach can make a more precise molecular classification of intrahepatic cholangiocarcinoma. We identified the 3 subtypes, such as stem-like, poor immunogenic, and metabolism, using unsupervised clustering of proteomic data. Overall survival and disease-free survival differed according to 3 molecular subtypes. Clinical features such as tumor markers, ADH family, ADH1A1, and IDH1 level differed according to biliary cancer subtype. Target gene expression of stemness regulators such as MYC, SOX, NANOS was significantly higher in stem-like subtype, indicated by green color, than in other subtype. The stemness-related gene, SOX2 and NANOS, was higher in ADH1A1 positive patient-derived organoid. These ADH1A1 expressions were validated by immunohistochemistry in patient tumor samples. There are relationships between the oncometabolite and the molecular subtype. The level of formaldehyde was significantly higher in metabolism subtype than in stem-like or poor immunogenic subtype. The other 3 aldehydes did not differ in among subtype. We performed the integrative analysis of proteomic and metabolite pattern to calculate the correlation between formaldehyde level and the protein expression. To validate the association between the metabolism subtype and IDH mutation, the plasma level of 2 representative oncometabolites were investigated in cholangiocarcinoma patients and healthy individuals. The plasma level of 2-hydroxychlorotarate and malate was significantly higher in metabolism subtype than in other subtype or in healthy individuals. Because the ADH1A1 is an important molecular subtype marker, we investigated whether ADH1A1 inhibition can enhance the drug sensitivity. The NCT501 is a potent ADH1A1 inhibitor, inhibited the activity of ADH1 enzyme in ADH1A1 positive cancer cells but negative cancer cells. The NCT501 and albumin-bound pancreatic cell shows the synergistic killing effect on the positive cancer cells, but not negative cancer cells. This LDH1A1 dependent synergistic killing effect was also found in patient-derived organoid, and SOX2 and nanos was decreased in LDH1A1 positive organoid with NCT501 treatment. Our proteomic clustering was completely validated by transcriptome meta-analysis of the data compendium assembled by combining 88, 36, 160, and 115 samples from Korea, TCJ, Japanese, and Singapore cohort, respectively. There is a positive correlation between the mRNA protein in most over 90% of patient tumor samples. From this meta-cohort, the three clusters shows the similar pattern of change in overall survival, LDH1A1, and LDH family expression, gene expression, as the corresponding proteomic clusters. We proposed both the subtype-specific marker and their therapeutic strategy according to three molecular subtypes. However, there is only one therapeutic strategy for each subtype. To make our molecular classification more clinically relevant, we need the more therapeutic option for each subtype. To achieve this, we planned the drug response evaluation using patient-derived organoid. We have a stable and standardized biliary cancer patient-derived organoid platform. Organoids are generated using the surgery, biopsy, and oscillated specimen. And in the case of surgical tissue, normal tissue in the neighboring location can be collected and result in a library, including the malignancy and normal tissue. We can maintain up to 5 passages of normal organoid and up to 5 to 10 passages of tumor organoid, and making library using frozen biobanking. Quality check is very important. We analyzed the histology, morphology, genetic alteration, and compare these with the patient samples and clinical outcome. Organoid-based drug response involved 5 steps. The organoid establishment, multi-omics-based subtype classification, anti-cancer hit and read selection, and in vivo validation. The level of apoptosis can be measured by calculating the AUC of ATP assay. Currently, we are using the 3D high-throughput screening platforms developed by the MBD company, Korean Bioventure. This process starts with the optimized organoid culture system, utilizing the cell printing of pillar plate and sandwiched culture plate. This MBD's integrated automation system, including the dispensers and high-throughput image, facilitated large-scale analysis. This platform can measure various parameters such as ATP, morphology, and viability, and to completely assess the drug response in organoid models. Currently, we have analyzed the molecular characteristics of 9 cholangiocarcinoma cell lines to understand the cell viability. Each cell line represents a different molecular subtype. We are analyzed the 14 cholangiocarcinoma patient-derived organoids together with these cell lines. We obtained the drug response data for 15 compounds, and we are analyzed the diverse drug response profiles observed in cancer cell lines and patient-derived organoid. This figure shows the development of deep learning-based molecular classification and the evaluation of this predictive accuracy. There are response differences between the classified subtype and highlighting some drug with differential efficacy. We planned also the pharmacogenomic analysis. The pharmacogenomic analysis have revealed the clear genetic signal associated with the variability of the drug response. We will integrate the single RNA sequence data with the drug response data, and then finally we will determine the therapeutic differences among cell population. We are extending this proteomic approach to gallbladder cancer, the unique subtype of biliary cancer. Our gallbladder cancer cohort consists of 119 patients recruited from 3 institutions in Korea: National Cancer Center, Samsung Medical Center, and Assam Medical Center. We identified the 2 or 3 subgroups based on the RNA protein glycosylation data. One subgroup shows the upregulation of lipid metabolism, and TMM signature and TMM score was significantly higher in this subtype than in other subtypes, indicating the involvement of immune microenvironment. Another subtype shows the significant association with ERBB23 amplification expression in gallbladder cancer. There are a positive correlation between copy number variation and ERBB2 expression at RNA and protein level. The one subtype with poor prognosis showed the context-dependent effect for TP53 mutation. Although the TP53 mutation are commonly mutated across subtype, its functional effect depends on the molecular context for each subtype. In TP53 activity is elevated, accompanied by the increased expression of downstream target ANXA1 and decreased expression of HNF4A. These functional relationships are observed only in this group. However, this is preliminary result. These are key elements or unanswered questions to persist in finding more effective treatment for biliary cancer patients. Stratification of risk factors, biomarker, adequate patient selection, and high-quality study design. This project will help us to answer this question. Ultimately, I hope the biomarker-driven therapeutic clinical trial based on the result of our project. I'd like to express sincere gratitude to everyone involved in this project and BTC patients and their family. Thank you for kind attention.
Thank you for a nice presentation. Now we will have a discussion panel, and we have 2 designated discussants. So would you please, Dr. Kim Jong-bae, start from discussion panel? Yeah, give any comment or direct question to the presenters.
Yeah, thank you for giving me some chance to lead some discussion. So I'm very happy to be here to be witness of some outstanding scientific outcome from the proteogenomics and in NCC International Symposium today here. So I would like to ask to all of some speakers. Dr. Yu Chen and Professor Sang-won Lee is the scientific side, and Dr. Wu is belong to some clinical side. So I think it can be some good combination for some questions. So could you please share your some perspective on strategies how to bridge this kind of some proteogenomics finding from the clinical sample to real-world clinical implementation. So Dr. Yu Chen just tried to make some diagnosis tool, and Professor Sang-won Lee just make some kind of trial for some new drug discovery. And also Dr. Wu just tried to get some organoid, some validation system. So I think there must be some kind of hidden limitation to translate this scientific outcome to the clinical side. So I would like to ask to Dr. Yu Chen at first, and move to Sang-won Lee, and let's move to the Dr. Wu.
This is a very important question, and also that's something that we have to really work hard. I think it takes several aspects. For the clinical community, the clinical doctor has to think about the clinical design of the cohort for the trial. So for example, in Taiwan, we have a long discussion about how to make it work. And my collaborator, the clinician, already have the registration for the clinical trial. And we also talk to our government what are needed to push this further. But I think the more challenging part is the technology side. I think some of us are the expert in the field. Even we have, it's not like genomics. The genomics is a very standardized platform. But for proteomics, we have different kind of instrumentation, different kind of sample preparation. So harmonization at the international level would be very, very important. And we have some experience to work with the US team, also under the CBTech, to push a protein mass spectrometry assay in a CLIA equivalent laboratory in Taiwan. And even it's a simple assay, it took 3 years to really transfer and also perform the parallel comparison on the clinical sample. So, but I think that is a wonderful example to demonstrate it's feasible at the international level. So I think if we can work together, say Taiwan, Korea, or even Japan, US, but I think maybe we can start from the Asia first because we have the similar patients' background. And we have to do the international harmonization and maybe create a standard kit for this kind of assay. And make sure that we have the same goal for each, maybe we can start from each specific disease type, maybe a specific cancer type, and we can use this kind of model to demonstrate the feasibility first and implement to other disease types.
Yeah, thanks.
So two things comes up to my mind at this time. One is it's very difficult to find the right pharmaceutical partners to develop what we found from our proteogenomic studies. So, like I mentioned, like I demonstrated in my talk, we discovered some very potent subtype-specific drug candidates, and when we try to contact with pharmaceutical partners, it's very difficult for them to start developments on these new drugs because of the industry, the medical industry in Korea is still very weak. So in that regard, I think it's maybe like Yuzu mentioned, that we can expand our territories into Asia, like including Taiwan and Japan. So they have a new therapeutic targets developing in their own countries, and we can collaborate with them to further develop our drug candidates. And another thing is that, as I mentioned in my talk in the last section, we are currently doing basket trials, and we need as many as drugs that can be used in the basket trials. So right now we are only looking at the clinical drugs only within the Korea. But I think if we can collaborate on that internationally with Taiwan and Japan, I think it will be a great help. You can share that too.
Yeah, Dr. Jong-Bae Park, would you please comment on us?
I have a question for Dr. Sang-Myung Woo. I really like this very well-organized study and talk. My question is, so in this study, you mentioned very interesting strategies, stratified medicine. So for that purpose, patient-derived organoids was used for validation tools, right? I was wondering, can we implement this multimodality, even from multiomics to patient-derived organoids, can be applicable in the clinic or do you think it's just for the experimental settings?
You mentioned that there is a significant challenge in the validation of this study before the clinical application. However, the patient-derived organoid is, as far as I know, is the good platform for validation before clinical trial. Also, we are performing the in vivo validation is very important for such as PDX model is another good platform for clinical translation. Yeah, with this preclinical model is the help us each other for people to clinical trial.
Yeah. Let's Dr. Kim.
Yeah, I'm very happy to ask this kind of question to the expert. So, yeah, I want to ask to the Professor Sang-won. So in mass spec data, as far as I know, if we get a spectrum data, I think the 30 or 20% of spectrum can be matched, can be annotated with the proteome data. So that means the other part, so 70% or 80% of the spectrum does not match the human proteome, but it's still big data and it's very valuable data. So in these days, many scientists try to get us some valuable information from the kind of unannotated spectrum data for some next level proteome. So is there any your opinion about that kind of some approach?
That's very interesting question, and I think you would agree with that. That's what it called black matters of the proteome data. I'm going to tell my experience with our recent studies on plasma proteomics, where we are going to analyze 20,000 plasma samples using mass spec-based proteomics technique. So the technique is evolved to a level where we can identify very close to 7,000 protein groups from one plasma samples within 24 minutes. So 7,000 protein is very large numbers for plasma proteome analysis. But as Dr. Kim mentioned, that only 20% of the MSMS data that we accumulated from the 24 minutes experiments were annotated to identify those 7,000 proteins. So we still have 80% of MSMS data that has not been annotated or interpreted yet. So we decide not to try to understand those uninterpreted MSMS data by using the conventional bioinformatics tools. So instead, we are going to use AI technique to find the hidden patterns among the uninterpreted MSMS data. So in order to do that, in order to get this AI method to be a viable method, we need to accumulate the data, not only in the depth, but also in the number of data. So hopefully we can get some hidden patterns out of these 20,000 plasma sample data.
Thank you for your answer. And one last comment from Dr. Ra. Yeah.
Yes, Sang-Ra from Korea. It's great to see all the progress. And I have a question to Dr. Chen. So I'm a clinician. So currently, most of the drugs are targeting tumor microenvironment, and one of the reasons why we are doing the RNA sequencing is to understand about the tumor microenvironment. So with the current improved technology of proteomics, how good it is for understanding of TME with the proteomics? And that is a question. And does that mean that we can replace the RNA sequencing to the proteomics? Because, you know, it's good to have all those data, but sometimes it's not, it has some limitation, but also too much information doesn't give us a good clinically applicable data. So sometimes simpler and abstracted is better. So I want to hear your thoughts from the expert point of view.
Thank you very much for this very important question. I'm so happy you raised the questions. I think in front of the genomics community, we have to be very humble. That means by genomics, my team is actually able to identify the different kind of mutational signature associated with the environmental carcinogen. So that is still a very important technology to tell us the impact of the different exogenous factors, including microbiota, including pathogen. But you know, when it comes to the practical application, it's not possible for the individual, the healthy control, to have the extensive genomic profile. That is so expensive and so difficult to do the so-called biomarker detection. So from our side, President Yang, our leaders, he felt that we should use the genomics to document the impact, the category of the carcinogen. But on the other side, use the protein to tell us the biomarker, the detection, and also the druggable target. So that is our critical consideration. And for tumor microenvironment, we are still under the investigation. We see very different correlation between RNA, genomics, and the protein. So I don't have time to go through today. We can discuss later. So we have some idea how to utilize the very small scale proteome to deconvolute the tumor microenvironment, especially the cancer cell, immune cell interplay, and complexity.
Thank you so much.
Yeah, thank you for answer. As a chairman, I have to close this session. We are 15 minutes behind the schedule. And we will shorten the break time, so the next session will be start on time. Thank you.
Thank you for the wonderful session. That concludes session 1. We would like to thank chairs, discussants, and speakers for their valuable contributions. Please give them a round of applause again. We will now proceed with group photo session. I'd like to kindly invite all the speakers, chairs, and discussants to come forward to the stage. We also warmly welcome all distinguished guests to join us for the photo session. Thank you. Next session will begin at 11:20.
일단 먼저 하자. 아, 이거 아니지. 나오네요. 보이시죠? 확인 했죠? 근데 이게 4대3이라로 제작해서 이렇게 양쪽이 잘려있는 거예요.
네.
일단은 미리.
잠시만요.
아니 그거 하셔도 돼요. 저 이거 여기에 연결된 게 아니어서.
강도가 너무 낮다.
네.
We will now begin session 2. The title of this session is Next Generation Immunotherapy in Cancer. We are pleased to have Professor Junho Chung from Seoul National University and Dr. Hyun-seok from National Cancer Center Korea as session chairs. Please welcome them with a big round of applause.
Hello everyone. It's my great pleasure to welcome you to this session 2, Next Generation Immunotherapy in Cancer. My name is Hyun-seok from National Cancer Center. I am hemato-oncologist and I'm co-chairing this session with Professor Junho Chung. He's here. He's an expert in therapeutic antibody and protein engineering. He's a professor in Seoul National University. And I'm privileged to hear from esteemed researchers who will present latest advances in T-cell regulation and adoptive T-cell therapy and CAR T-cell innovation. And after 3 topics, we'll have discussion time for about 20 minutes. And I will introduce today's speaker, Professor Taku Okazaki first. Briefly, I'll introduce him. And he graduated from Kyoto University Faculty of Medicine. And he is a director of the Japanese Biochemical Society. And his main research interest is the pioneering PD-1 research, and also his current research focus on the functional interplay among inhibitory and stimulatory receptors and T-cell activation, autoimmunity, and anti-tumor immunity. And now he's a professor at the University of Kyoto Institute for Quantitative Bioscience. Today's topic is illustrating molecular mechanisms of T-cell suppression by checkpoint molecules. Professor Okazaki, please.
Thank you for the kind introduction. I'm Taku Okazaki from the University of Tokyo. First of all, I'd like to express my sincere gratitude to the organizing committee, particularly Dr. Eun-jun Park, for giving me this opportunity to visit this esteemed Institute for Cancer Research in Korea and share our recent research activities on immune checkpoint molecules. Okay, so as you know, Drs. Allison and Honjo received Nobel Prize in Physiology or Medicine 2018 for their contributions to the discovery of cancer therapy by inhibition of negative immune regulation. Since I was engaged in PD-1 research in Dr. Honjo's lab for 9 years from 1999, I'd like to start my presentation by briefly reviewing the history of PD-1 research. So PD-1 was identified as a gene whose expression is induced during programmed cell death by apoptosis in 1992. However, the overexpression of PD-1 did not induce apoptosis of the cells used for the screening. So PD-1 function remained unknown for many years. The situation changed in 1999 by the discovery that aged PD-1 knockout mice developed autoimmune diseases in B6 background, which led us to recognize PD-1 as a negative immune regulator. Soon after that, we succeeded in identifying PD ligand 1 and ligand 2 and elucidated the inhibitory mechanisms by PD-1. And in 2002, we found that PD-1 inhibits anti-cancer immunity in addition to autoimmunity. Based on these findings, clinical trials to test the therapeutic efficacy of PD-1 blocking antibody in cancer were started from 2006 in US. The interim report in 2010 was very promising and attracted wide attention, leading to its approval for the treatment of melanoma in 2014. As you know, its indications expanding to the tumors in other tissues, and the combinatorial therapy with anti-CTLA-4 blocking antibody, which was developed by Dr. Allison, was also approved for the treatment of several types of cancer. Since there are many inhibitory co-receptors in addition to CTLA-4 and PD-1, they are recognized as promising therapeutic targets next to PD-1 and CTLA-4. But the development of novel therapeutics targeting these co-receptors did not go smoothly, and we had to wait until 2022 when anti-LAG3 blocking antibody was approved for the treatment of melanoma in combination with anti-PD-1 blocking antibody. Today, I'd like to talk about our basic studies on LAG3. So in a physiological condition, PD-1 is especially important for the regulation of autoimmunity, as evidenced by the spontaneous development of autoimmune diseases by PD-1 knockout mice. As shown here, PD-1 knockout mice develop various types of autoimmune diseases depending on the genetic background of the mice, just like the IRAE in human patients. On a biopsy background, PD-1 knockout mice develop dilated cardiomyopathy and gastritis. Interestingly, they produce tissue-specific autoantibodies. We are particularly interested in the class switch and affinity maturation of autoantibody, since the class switch recombination of the immunoglobulin gene is the utmost priority in Professor Honjo's lab. And Dr. Muramatsu had just discovered the key enzyme of the class switch and affinity maturation, that is AID. We initially expected the resolution of the disease by the introduction of AID deficiency, but AID-PD-1 double knockout mice developed very severe myocarditis and died soon after weaning. AID deficiency also exacerbated the type 1 diabetes in NOD background. However, these phenotypes are not due to AID deficiency itself, but to a mutation, loss of function mutation in LAG3 gene, which locates next to AID on mouse chromosome 6. So we serendipitously discovered that LAG3 functions cooperatively with PD-1. So LAG3 is widely recognized as inhibitory co-receptor by now, but even though many of the basic characteristics, such as ligand specificity or inhibitory mechanisms, remain unknown or controversial. LAG3 shares about 25% amino acid homology with CD4, and early studies implicated that LAG3 might associate with MSC class II with higher affinity than CD4. Thereby, LAG3 was postulated to suppress T-cell activation by competitively inhibit CD4. But the experimental evidence was rather insufficient, so we started by analyzing its inhibitory function. We stimulated DO11 T cells with antigen presented on antigen-presenting cells. When we overexpress LAG3 on T cells, so we measured the magnitude of activation of T cells by measuring the amount of IL-2 secreted by T cells upon activation. So when LAG3 is engaged, IL-2 production is substantially decreased, as shown with red bars, and its inhibitory effect is comparable to that of PD-1. When both of these molecules are engaged, IL-2 production was further suppressed, indicating that these two molecules act synergistically to suppress T-cell activation. It may sound odd, but this T-cell line did not express CD4, although this T-cell activation is restricted to class II. So this inhibition cannot be explained by the competitive inhibition of CD4. That casts doubts on the involvement of class II as a ligand. So we tested it by making LAG3 protein and staining various cell lines with or without class II expression. As shown here, some of the cells could be stained with LAG3 protein very strongly, while other cell lines scarcely bound LAG3. When we use these cells to stimulate T cells, all of the cells could support the T cell activation, but some of the cells failed to activate LAG3 signal, which suggests the presence of the non-MSC class ligands for LAG3 or requirement of modification of MSC class II for the interaction. So as I will show later, the latter was actually the case. So peptides are loaded in MSC class II in a very controlled manner. CD74 invariant chain and H2DM are reported to function as chaperone or editor to support the proper and stable formation of the peptide MSC class II complex. In the case of IA, mouse MSC class II, first, fourth, sixth, and ninth amino acids of the peptides occupy the pocket-like holes on the MSC class II. Because the size and electrostatic characteristics of the pockets differ, amino acid residues that are favored by each pocket differ. So, depending on the amino acid residues that occupy these pockets, there's binding affinity between peptide and MSC class II, and thus the structural conformation of the peptide MSC class II differ. So I will show one, here I show one example of the self-antigen in NOD mice. That is 9th to 23rd peptide fragment of the insulin B chain. And this peptide is reported to form stable or unstable complex with the MSC class II of the NOD mouse, that is IAG7. When 13th glutamate or 12th valine occupies the pocket 1. So when 13th glutamate occupy the pocket 1, they form stable complex. So we covalently attach this peptide to the IAG7 so that this peptide is presented in a stable or unstable structural conformation and stain the cells with LAG3 protein. As shown here, LAG3 preferentially recognize the stable complex of the autoantigen and MSC class II. Then we immunized mice with these peptides and tested the expansion of the T cells specific to this peptide MSC complex. As shown in the right slide, the number of the T cells expanded upon against the stable peptide MSC complex was apparently fewer compared to that in the unstable complex, which likely reflects the deletion of the T cells that recognize the stable complex of autoantigen and MSC class II in the thymus by negative selection, because this is a self-antigen. However, when we blocked LAG3 function by antibody, the number of the stable T cells reactive to the stable peptide MSC class II, but not to the unstable complex, substantially increased, indicating that such autoreactive T cells do exist in the periphery and be suppressed by LAG3. Accordingly, we also observed the substantial increase of T cells reactive to the stable autoantigen MSC class II complex in newly diabetic NOD LAG3 knockout mice, suggesting that the activation of these T cells is actually the key to exacerbate type 1 diabetes. So we found that the LAG3 selectively recognizes stable peptide MSC class II to suppress T cells that respond to the stable peptide MSC class II. In addition to MSC class II, there are several potential LAG3 candidates, ligand candidates reported. Among them, we could reproduce the binding of FGL1 to LAG3, but not the others. So we decided to validate the functional contribution of FGL1 binding to LAG3. So we first tried to obtain mutants that associate either with FGL1 alone or class 2 alone. So as shown here, the LAG3 mutants that can associate only FGL1 failed to suppress T-cell activation, while LAG3 mutants that can only associate with class 2 suppress T-cell activation as strongly as wild type, indicating that T-cell activation, inhibition of T-cell activation by LAG3 solely depending on its binding ability to class 2. So we generated knock-in mice of the mutants in the background, and as before, the prevention of fulminant type 1 diabetes is purely dependent on its ability to associate with MS class 2, but not FGL1. We also generated the LAG3 knockout mice on B6 background and inoculated these mice with MC38 colon carcinoma cells and found that, as shown in red circles, LAG3 mutants that cannot associate with MSC class 2, tumor growth was retarded almost similarly to LAG3 knockout mice. So these results clearly indicate the stable peptide MSC class 2 is the functional ligand of LAG3 in the suppression of T-cell activation or prevention of autoimmunity and retardation of the, anti-tumor immune responses. As mentioned in the beginning, many of the basic properties of LAG3 remains unknown or controversial. Particularly, we still don't know well how LAG3 inhibits T-cell activation, but we noticed that LAG3 strongly accumulates at the immunological synapse upon recognition of the stable peptide MSC class 2. So we contemplated that we may be able to trigger LAG3 function by recruiting LAG3 to immunological synapse. So we generated bispecific antibody that simultaneously recognized LAG3 and TCR. As expected, when we added this bispecific antibody, IL-2 production from T cells was suppressed in a LAG3 P118 mutant dependent manner. This mutant lacked the ability to associate with class 2. So we could actually trigger LAG3 function in a condition where LAG3 cannot function. Okay, so we also could therapeutic effects of this bispecific antibody in several models of autoimmune diseases in mouse, such as EAE. So there exist many inhibitory and stimulatory co-receptors, and they are supposed to function cooperatively to regulate the magnitude and outcome of T-cell activation. And thus, they are regarded as potential therapeutic targets. But our mechanistic understandings of these pathways are still limited. And more precise and deeper understandings of these pathways are expected to facilitate the development of safe and effective immunotherapies. Okay, I'd like to acknowledge all the lab members and collaborators, especially most of the work presented today were done by Takumi Maruhashi here. Thank you for the kind attention.
Thank you for the wonderful presentation. Question and comment will be done after topics. All topics are done. I will introduce the next speaker. Second speaker will be Chung-Young Han, National Cancer Center, Korea. I briefly introduce him. He graduated from the University of Tokyo, Japan, biotechnology, and he got PhD at same university, and now he's chief immuno-oncology branch, National Cancer Center Institute. His main research topic is CAR-T therapy, and he's working on the cancer immunotherapy and T-cell engineering. Today's topic is factors influencing the efficacy of adoptive T-cell therapy in solid tumor. Please.
Thank you, Dr. Om, for the introduction, and thank you for inviting me to this amazing session today. What I'm going to talk about today is the factors that impact the efficacy of most T-cell based therapies in solid tumors. So I'm going to start with a brief introduction into the T-cell immunology in cancer. The cancer immune cycle is a very well-defined concept in the immune oncology. T cells infiltrate into tumor tissues and kill the cancer cells. And then the released tumor antigens are captured by antigen-presenting cells, and the cells re-educate other T cells in the lymphoid tissues. One another key characteristics of T cell immunology is TCR diversity. Theoretically, more than 10 billion repertoire can exist, so with that diversity, T cells effectively target heterogeneous tumor antigens. However, if there is immune resistance mechanism, then the T cells fall into the status of exhaustion. Immune checkpoint blockade can reinvigorate these T cells, and adoptive T-cell therapy, which is the main topic of my talk, will add more T cells on this side to attack cancer. Generally, in adoptive T-cell therapy, we collect T cells from the patients, enrich the tumor-specific T cells, and then reinfuse them back to the patient. And historically, the adoptive T-cell therapy has been developed in a way of increasing the tumor reactivity of the infusion product. And because of the recent advances in genetic engineering, we can convert most of the T cells taken from the patients into highly tumor-specific T cells. Generally, a T cell receptor or chimeric antigen receptor that is specific to the tumor antigens are used to generate the gene-engineered T cells. Here I briefly summarize the FDA-approved adoptive T-cell therapy. The 7 CAR-T therapies were approved by US FDA for the indications of leukemia, lymphoma, and multiple myeloma. In the last year, 2 T-cell therapeutics were approved for solid tumors, one for melanoma and the other for synovial carcinoma. CAR T-cell trials for solid tumors are ongoing, but there is still unsatisfactory results compared to the hematologic malignancies. However, in the last year, here I highlighted in red, some reports said some promising results were revealed by those clinical trials. Renal cell carcinoma and diffuse midline glioma indicated long-term complete responses in a small fraction of the patients. Then what are the challenges in CAR-T for solid tumors? One is inefficient trafficking of T-cell into tumors, and the other is another component is tumor microenvironment. Heterogeneous antigen expression is one another issue because most tumor-associated antigens are also expressed in the normal tissues. And today, I'm going to focus on discussing the CAR-T persistence. One of the key findings from the CAR-T trials that were conducted for the last 10 years is that there is a huge difference in the CAR persistence between hematologic cancer and solid tumors. In the patients with leukemia, CAR-T can persist for months, and in some cases, that can persist for more than 10 years. However, in the case of solid tumors, CAR T disappears within a month, and the expansion rate is 10 to 100-fold lower than the hematologic cancer. So I thought this is an important point because when developing CAR T therapies, we use the same mouse model regardless of the persistent difference in the disease model. There are two preclinical models for studying CAR T. One is immune deficient xenograft mouse model, and the other is syngeneic mouse model. In a nutshell, the xenograft mouse model uses human CAR T and human tumor cells. There is no immune cells, and stromal cells are all mouse originated. And in the case of syngeneic mouse model, the stromal cells, immune cells, and tumor cells in CAR T are all originated from the mouse. So this can see the complex interaction between these cells, and because of these factors, the CAR T behave very differently depending on each model. In xenograft mouse model, CAR T persists for more than months, and in the syngeneic mouse model, CAR T disappear within a week. So I thought, okay, now the solid tumor model can be better represented by using syngeneic mouse model. And we try to see some factors that impact the CAR T therapy in solid tumors by using a syngeneic mouse model. One thing we focused on was CAR constructs. All FDA-approved CAR T therapy use either 28Z or BBZ construct. It differs in using the costimulation domain, CD28 or 4-1BB. And because of these molecular characteristics, these two CAR constructs indicate different functional characteristics. 28Z indicates the strong effector function, and BBZ indicate rapid cell division and memory formation. And interestingly, the CAR T trials that used these two CAR constructs has no significant differences in between therapeutic efficacy. Both are effective in most of the hematologic malignancies. And we thought maybe this efficacy could be varied in the solid tumor model. So we wanted to check the differences in the syngeneic mouse model for solid tumors. And before to start, we want to make sure that between these two species, the 20Z CAR and BBZ CAR has the same differences in the function. So we generated the 20Z and BBZ CAR from the T cells from these two species and activated that for 2 hours. From the single cell transcriptome analysis, we found the activated T cells were clustered in two-dimensional spaces. Cluster 2 was associated with BBZ construct, and cluster 3 was related to the 20Z construct. We found that there's a huge homology between human and murine CAR-induced genes. Cluster 2, which was related to the BBZ construct, expressed high level of chromosome segregation and cell division-related genes. Cluster 3 expressed many genes that related to the effector functions such as cytokine production. Okay, now the human CAR T and mouse CAR T has the similar differences in 28Z and BBZ constructs. So we tried to see the therapeutic efficacy of these two constructs. We used B16F10 melanoma mouse model, and we preconditioned the mice and infused the CAR T. We found that tumor growth was the anti-tumor efficacy was higher in 28Z constructs, and the CAR T persistence was higher in 28Z construct. Next thing we've looked into was trafficking of CAR T in different tissues. There is a very small fraction of paper that investigated the trafficking of CAR T in human clinical trials. One study in 2006, they investigated the trafficking of CAR T in human trial, and the CAR T enriched in lung and liver site at the initial stage, and 2 days post-infusion, they infiltrated into lymphoid and tumor tissues. And we want to check if this phenomenon is actually repeated in our syngeneic mouse model. After injection, the CAR T were enriched in lung and liver site, and on day 1 post-transfer, the CAR T moved into the lymphoid tissues. And from day 2, they are infiltrated into tumor tissues and lasted for 10 days. After a couple of weeks later, the CAR T completely disappeared in mice. So we checked the trafficking of these two constructs in the mouse model. Overall persistence was higher in 28Z in lymphoid and tumor tissues. But the interesting finding we saw was that on day 5 post-transfer, BBZ indicated higher signal in the tumor sites. It increased sharply on day 5 post-transfer and rapidly declined within a couple of days. It was interesting because BBZ indicated the gene expression is related to the rapid cell division. So we performed some in vitro experiment to find if this is happening in cellular level. We co-cultured CAR T with tumor cells, and we cultured the CAR Ts with tumor cells, and we found that the BBZ construct rapidly divided. It's faster than the 28Z construct. And the strong activation of CAR constructs can lead to activation-induced cell death of CAR T. So we checked the apoptotic cell ratio in BBZ CAR constructs. And interestingly, these constructs induced more apoptotic cell deaths than BBZ 20Z construct. And because of these contrasting effects, the BBZ frequency was higher compared to 20Z construct at 6-hour post-tumor contact, but it became lower after 24 hours. We also checked the polyfunctionality of these two constructs because 28Z was known for the higher expression level of the cytokine. As expected, the 20Z construct expressed high level of cytokines such as TNF, interferon, and homeostatic cytokine IL-2. In addition, the CAR T 20Z CAR induced high fraction of polyfunctional T cells, which means the cell fraction that can produce multiple cytokines simultaneously. Lastly, we checked the safety profile of these two constructs. We checked pro-inflammatory and cytokine plasma level in these mice. BBZ indicated higher concentration of these chemokine and cytokines compared to the 20Z construct. Despite the high persistence and polyfunctionality, 20Z retained these markers at low level, and this indicates the safety profiles of 20Z construct in CAR T trial for solid tumors. Next couple of slides, I'm going to show the recent updates of our study on developing the CAR T therapy for solid tumors. In this study, we chose to design a glypican-3 targeted CAR because we thought GPC3 is a relatively safe target and expressed in various solid tumors, including liver cancer. The monoclonal antibody against GPC3 was developed by traditional hybridoma screening, and the antibody fragment was fused to second-generation CAR construct. We chose CD28 costimulation domain based on the findings from the syngeneic mouse model. The designed CAR T expressed sufficient level of CAR molecules on the surface, and it bound to the specific antigen. And in response to the GPC3 expressing cell line, the CAR T expressed high level of IL-2 molecules. In the efficacy evaluation, we used xenograft mouse model because we used human-derived CAR T. And when we injected HCC model cell line HepG2 into these mice, we saw that the designed CAR T products can suppress the tumor growth that was comparable to the already FDA-approved CAR T products. One interesting thing we found was that on day 55, in the blood there was higher persistence of 19 BBZ construct. This is a well-known fact because in xenograft mouse model, the BBZ construct shows more proliferative potential and memory potential. But interestingly, our designed GPC3 28Z construct indicated higher persistence rate that is comparable to the BBZ construct. And in the next step, we humanized this GPC3 CAR by in silico design and prediction. And we chose the 4 candidates of the CAR molecule. And we found that the affinity to the GPC3 antigen was different among these 4 candidate molecules. Version 1 and 3 was slightly lower than the original mouse antibody, but it was higher. It has higher affinity than version 6 and 8 molecules. In the efficacy analysis, we found that version 1 candidate induced faster effector kinetics compared to the other constructs. And in the case of version 6 and 8, CAR candidates, which have the lower affinity, they failed to completely eliminate the tumor cells. And in the persistence of CAR T, we found that on day 5 post-transfer, most of the CAR T in the blood was comparable among the group. But on day 12 post-transfer, version 1 CAR candidate induced highest persistence in these mice. In the polyfunctionality analysis, the high affinity candidate version 1 and 3 indicated higher polyfunctional capacity compared to the version 6 and 8 candidates which have lower affinity. Now, the CAR T efficacy evaluation using syngeneic mouse model is ongoing, and after choosing the final candidates, we are planning a preclinical test for IND application. To conclude, there was 4 major challenges in ACT for solid tumors: the trafficking issues and heterogeneous tumor antigens and immune suppressed TME. And I stressed the importance of persistence in CAR T therapy. And I also provided some insights from syngeneic mouse model. In this model, we found that 20Z demonstrated enhanced functional fitness compared to BBZ construct. And in the therapeutic potential of GPC3 targeted CAR T-cell, we found that the HCC model showed GPC3 CAR T induced rapid tumor regression and demonstrated sustained persistence and polyfunctionality. With that, I'll end my talk, and I'd like to thank all the members and collaborators who did so much for this study. Thank you.
Thank you for your nice presentation. I apologize for this inconvenience during the presentation because of some technical problem. Do we need time to fix this problem or start right now? Is it okay to start right now? Okay.
Okay, I'll introduce the third speaker of this session. He is Professor Kyung-ho Choi from Seoul National University at Department of Biochemistry and Molecular Biology. He got MD from Seoul National University, and also he followed the dual degree track and got a PhD from the same university. And then after postdoctoral training at NIH, he became a principal investigator of this institute for a while. And then he moved to Seoul National University, now serving as associate professor. And also he's following a kind of academia and the entrepreneurship at the same time. And then he's a co-founder of the T-Caros company. That's a CAR-T company, one of the which pipelines is now in phase 1/3 trial. And please welcome Kyung-ho Choi.
Thank you for a nice introduction, Dr. Jung. So, may I have a slide, please? So, to start, so I apologize for changing my title. So it's a little different, but I thought it might be more interesting. So, title of my talk will be CTLA-4 CD20 chimera potentiates CAR T cells via CD86 on both T and B cells. To briefly mention that, because you may be very familiar with CTLA-4, CTLA-4 is an inhibitory receptor on T cell surface. So, and is target of first immune checkpoint inhibitor, ipilimumab. So, but if you, so this is inhibitory receptor on the T cell surface, but it should engage the antigen presenting cells rather than tumor cells to get inhibitory signal. So, and then ipilimumab, with anti-CTLA-4 antibody, was the first immune checkpoint antibody that has been used as systemic administration, but it showed systemic autoimmunity as a side effect. So, so we have to be very careful with the dosage. So that made me think that probably if you have a certain method to specifically inhibit CTLA-4 on tumor-specific T cells, and then if you use that as a therapeutic T cells, then we may evade those side effect of a CTLA-4 antibody and may have a better T cells to kill the tumor cells. Okay. So this was the introduction of CTLA-4. So CTLA-4 is expressed on T cells, I mean, after activation. But in order to be activated, T cells should engage peptide T cell receptor signal and CD20 signal at the same time. But after T cell activation, it induces CTLA4, upregulates CTLA4. But CTLA4 shares B7 ligand with CD28. It hijacks B7 and delivers inhibitory signal. So anti-CTLA4 antibody can inhibit can enhance T cell activation. But if you use that systemically, you will activate all the T cells in your body, and that will lead a systemic autoimmune side effect. The selective inhibition of CTLA4 tumor-specific T cells would be an ideal way of enhancing anti-tumor immunity without systemic autoimmunity. As Dr. Han said, that adoptive T-cell therapy may be a solution, because in probably 20 years ago, the NCI group isolate tumor antigen-specific T cells and then expand them and reinfuse back. So at that time, for the melanoma patients, the response rate was 50%, but that efficacy has not been achieved for other cells. So we developed CTLA4. If we develop certain CTLA4 mutant that can inhibit CTLA4, then if you make the antigen-specific T cells, and then if you make them CTLA4 inactive and then reinfuse them, then there will be better T cells. So for that, we designed the CTLA4 dominant negative mutant. Initially, we thought probably CTLA4 has an inhibitory motif at the cytoplasmic domain. If we have extracellular and transmembrane domain, and then overexpress that to the T cells, it will competitively inhibit compete out with endogenous CTLA4 for binding B7, so it will inhibit inhibitory signal. But problem is this one may also engage positive, also compete out the positive CD20 signal, because these two molecules shares the B7 ligand. So we, to overcome this problem, we take out this stimulatory domain of CD28 and then linked it to the decoy receptor to make the CTLA4 chimera. When you overexpress this molecule, they will compete with CD28, but still delivers positive signal. And CTLA4 comes up, then it will compete out the CTLA4 and inhibit inhibitory signal. In the adoptive therapy setting, it worked well. I mean, so in vitro, so decoy receptor prevented proliferation and cytokine production, but this chimera receptor fully restored the proliferation and enhanced the huge production of interferon gamma. So in adoptive transport T-cell therapy model, it actually enhances a dramatic efficacy, anti-tumor efficacy dramatically in both lymphoma and melanoma model. But adoptive T-cell therapy has a limitation. So because in order to get the antigen-specific T cells, the small number of tumor-specific T cells isolated from either tumor or peripheral blood should be expanded greatly, which takes more than 1 month. So that's quite a long time for an advanced cancer patient. But CAR T cells are different. If you transduce CAR molecule to the T cells and then expand them in vitro, they will take around 1 or 2 weeks. So in 1 or 2 weeks, you will have a large number of tumor-specific T cells that will recognize tumor cells with CAR molecule. So that means you can generate a large number of tumor-specific T cells in a short amount of time. But then, because this CAR T cells could be made by retroviral transduction, if you have one retrovirus that can harbor both CAR and CD28, then you can have two benefits. One is you are getting a large number of tumor-specific T cells with enhanced anti-tumor function. So with that, as Dr. Han said, we chose the immunocompetent mouse model. And this is the CD19 positive lymphoma in biopsy background. So we generate biopsy CAR T cells with CD28 molecule. So in vitro, actually, it showed a similar cytotoxicity to the tumor cells, but showed a modest enhancement of cytokine production. But But if you go to the in vivo, it drastically enhances anti-tumor efficacy. So with 5 times 10 to the 6th transfer T cells, CD19 CAR alone, which is kind of Kymriah mimetics, showed a quite good response, but does not show complete remission. But with CTS20 modification, it showed a full remission. And if you reduce the number 1 to 1/5, then the Kymriah-like cells do not show any efficacy, but this CTS-modified cell is full efficacy. So it's more than 5-fold enhanced therapeutic efficacy. And if you trace the cell number, then you realize that for the CD8 CAR T cells, with 5 times 10 to the 6 cells, it showed a little bit better. But if you go down to the 1 to the 1 million cells, then it shows a dramatic enhancement of CD8 CAR T cell in expansion and contraction, and still persists with much more cells, which is similar to the plain CAR T cells with 5 times more cells. For the CD4 CAR T cells, it showed a delayed but a dramatic expansion of CD4 CAR T cells at both doses. So, and this CD4 preferential expansion is pretty well known with our previous study with adoptive T-cell therapy. So for summarize first part, the CTS20 dual CAR modified T cells show a far better anti-tumor effect than conventional CAR T cells. I didn't show you this data, but it has dependent on CD4 help on CD8 T cells, and is dependent on IL-2. But the more important question is, what I couldn't understand. It was good. I mean, it showed a very better effect, a huge expansion. But what I couldn't understand is, then who gives the ligand to these CAR T cells? Because for the conventional adoptive T cell model, there was definitely APC, because APC expressed the B7 on the surface. So conventional T cells will be activated and then move to the tumor site and then kill the tumor cells. But what about CAR T cells? Because CAR molecule itself engages tumor antigen itself. It can bypass APC, theoretically. So that in that case, CTS20 doesn't have B7 to ligate to be enhanced. So then the only possible scenario in that case it would be if you have a blood tumor like lymphoma, it expresses B7 on the surface of tumor cells. So then tumor cells will give CAR signal together with CTS20 together. So this is a more possible scenario. Or the other scenario will be if tumor is B7 negative, then APCs in the tumor microenvironment should have a separate engagement which is trans, and then these T cells will be activated. But this is maybe quite inefficient. So we tried to test that by deleting, because this is A20 cells. It's B-cell lymphoma, so it expresses CD88 and 6. So B7 is an old name. So B7.1 is CD80, and B7.2 is CD86. So I will call that interchangeably. So we tried to knock out this CD88 to deplete the B7 deficient tumor cells. And then we did that CRISPR-Cas9 technology. But surprisingly, we found that without this B7 on tumor cells, it still showed a dramatic anti-tumor effect, either high dose and low dose. And CAR T cell expands greatly, just like B7 positive tumors. So that happened similarly in B6 mice. In this case, we used the EL4 lymphoma, which is derived T cell in this case. So overexpressed CD19, and because they express slightly CD80, not CD86. So we deleted CD80. So to make the B7 deleted the EL4 cells. Here is the same. So with EL4 CD19 or EL4 CD19 B7 negative, it showed a similar dramatic anti-tumor effect and dramatic T cell expansion in both the CD4 and CD8. So then this scenario might not work. Then this one is okay. I mean, then a host APCs actually provide B7 ligand to the CAR T cells. To answer the question, we brought the CD8086 double knockout mice as a host to delete the B7 on APCs. And with that host, actually the enhancing efficacy was a little bit reduced, but still substantially higher rate. So I'll come back to this phenomenon later. But if you look at the CAR T cell expansion, then CAR T cell expansion is dramatically reduced. So them as host APCs are main drivers for CD20 CAR expansion in the blood. Then which APCs are major ligand provider for this CAR T cell expansion? So the APCs will be dendritic cells, macrophages, or B cells. The B cells might be more efficient because it will express CD19 and B7 together, and it will ligate cis-ligate these T cells. But this case, the mouse were lymphodepleted before CAR T cell transfer. So theoretically, there is no B cells in the body. But this one, the dendritic cell macrophages, it should transligate the CAR T cells. The B7 should be provided by APC, and antigen should be provided to tumor cells. It's inefficient, but because APCs are radiation resistant, there might be sufficient APCs there. But the student, my student, So Lee, who did all this experiment, found that although there's lymphodepleted, there are a small number of B cells left in the peripheral blood. So we checked this first. So then we checked this by using a rank knockout mouse. In this case, genetically is T cell and B cell deficient. So using that mouse as a host, basically, actually, we could not see any enhanced CAR T cell expansion in the presence of CD28, although we have some anti-tumor benefit. So, but this is whole B cell knockout mice, this is lymphopenic mice. So we generated B cell specific B7 double knockout mice. So for this, we did the mixed lymphocyte, mixed bone marrow chimera. 80% of bone marrow is derived from B cell deficient mice and 20% of B cell sufficient mice. In this case, all the B cells lose CD886, but all the blood cells only lose 20% of blood cells lose CD826. So it's B cell specific. So in this case, actually we also could see dramatic decrease of peripheral B cell CAR T expansion, although we show some effect remained. So that means host B cells are main driver of blood cell expansion, but how could we explain the anti-tumor effect of CAR T cells in the absence of host and tumor B cell? That means in the absence of B7 double knockout and in the absence of tumor B7, so CAR T cells should still show considerable amount of CAR T cell efficacy. Then the only remaining cell is CAR T cell itself. But traditionally, B7 is not important for T cell expansion. T cell function. But we look at the T cells, CAR T cells, they actually express B7, CD8086 expresses, and then in the presence of CD20, that expression level actually decreases. So that means somehow this CD28 engaged a T cell express CD8086. So we use the CD8086 double knockout CAR T cells. In that case, we lost the complete enhancement. So that means this B7 molecule actually contributes this strengthening effect. So does B7 of the CAR T cells contribute to CAR T cell functionality rather than T cell expansion? CD20 CAR T cells show reduced B7 expression. This is interesting. So not all ligand receptor interaction reduces ligand itself. So then why B7 is downregulated on the T cell surface? There might be transcriptional controlled, internalization, but both we checked, that didn't happen. So then the only thing remaining in our mind was that CTLA4 is suppressed in regulatory T cells, and in regulatory T cells engages APCs that actually take out the B7 molecule on the surface and then suck up. So that's called transendocytosis. So we thought probably then this is transendocytosis. So to try to solve the question, we separated the CTS20 expressor and B7 expressor. So we call that Ripper cells, and then this one is a ripped CAR T cells. So, and then we co-culture them to see whether this surface molecule is transferred to this Ripper cells. If you look at the ripped cells, actually, those CD86 expression was drastically decreased by CTS28 Ripper cells. But is it really transendocytosis? We made another transduction using another ripped cells using CD80 GFP fusion protein, and then co-cultured with the Ripper cells. And we looked at Ripper cells. When we looked at Ripper cells in the presence of CTS28, actually all the GFP signal was transferred to the Ripper T cells. That means it has been transferred to these T cells. That has been confirmed by confocal microscopy. I didn't have time to show that. So then is it important, downregulation? So there has been report that B7 is expressed on T cells a little bit. And then if you remove that B7, actually T cell function goes up. So that means B7 on the T cells may be inhibitory. So then just the ripping function may give the CAR T cells better function. So when you make the ripped T cells and we eliminate Ripper T cells and then incubate it with tumor cells, then we showed better proliferation, better cytokine production, but not that to the level to the CTS20 CAR T cells. To compare that with ripped CTS20 CAR T cells, which we usually generate, then they proliferate more and produce cytokine drastically, actually. So that means CAR T cell ripping function and CTS20 induced signaling may both work to enhance the CTS20 function. So then other people says that is it autonomous ligation? Is single cell ligase their own B7? So to see that, we generated CD20 responsive element GFP CAR T cells, and then we inject them to the CD86 knockout mice with CD80 knockout tumor cells. So the only source of B7 is CAR T cell itself. Then that GFP signal goes up. So that means this ligand happens, and then B7 double knockout mice, actually that signal goes down. So that means this B7 ligase autonomously. So both transendocytosis mediated B7 downregulation is CTS20 CAR T cells, potentially CAR T cells, and collectively B cells for cell expansion and T cells for functional enhancement, which was totally unexpected ligand providers. So in human CAR T cells, actually that B7 expression and downregulation of CTS28 also happened. And then if you go to this the CD19 CAR T cell model, this is human CTS28 CAR T cells, actually that dramatic enhancement also happened, and peripheral blood CAR T cell expansion also happened. So to summarize this part, CTS28 CAR T cells show dramatically enhanced CAR T cell function and number based on unexpected ligands on T cells and B cells, not the apices. So CTS28 CAR T cells provide a novel approach for next generation CAR T cell therapy using a very unusual mechanism. So this, the discovery of CTS28 was done when I worked in NCC before 10 years ago, in collaboration of Hyun-Suk Am and Sung-Jin Lee. And then at the actual discoverer Jae-Woon Shin is now became a professor at Yonsei University, and So Lee and Jian Lee did all this experiment. And based on this data, actually, I founded a company, CAR T company named T-Carus. Okay, thank you very much.
Now we'll have a discussion, and we have two discussants, and I think that the Prof. Dr. Kim of National Cancer Center give us a 7-minute talk, and then afterwards we'll receive the questions from the floors.
Yeah, today I will briefly discuss the update of the clinical trials to prepare for the future economic national burden related to the global cell therapy competition. First, let's briefly discuss the Dr. Han's presentation, the only presentation file I was given. Glypican-3 expressed in 72% of the malignant hepatocytes, but not in benign liver or other benign tissue except placenta. And the first phase 1 trial was disappointing, but with armored approach, IL-15 armored GPC3 CAR T cells showed increased expansion and enhanced clinical response according to the Baylor group clinical trial. As you see, the unarmored GP3 CAR T was not responsive, while the IL-15 armored GPC3 CAR T showed clinical response in 4 of 12 study participants. Please note that the retroviral vector used for these both CAR was manufactured in academic GMP in Baylor and published in Nature. In 2000, last year, TILs and MAGE-4 TCR-Ts were FDA approved, while there have been no CAR-Ts were approved for solid tumors. But notably, the price tag of these two cell products were $500,000 and $700,000. Here you see the current ongoing cell therapy clinical trials for cancer. And as you see, the TCR-T and TILs, TIL clinical trials represent approximately half the number of CAR-T clinical trials globally. And since our institution just launched a program to sponsor academic clinical trial, early phase clinical trials for not only for CAR-T, but also TCR-T and TILs, I briefly touch base also on the TCR, which targets PMHC with lower affinity, but with lower activation threshold than CAR-T. And notably, given that TCR-T can recognize intracellular antigen and that most of the tumor-specific antigens are intracellular, TCR-T can target neoantigens, which are present in the majority of the common solid tumors, unlike MAGE-A4 or other cancer tests antigen. So we may consider a highly individualized approach targeting neoantigen using TCR-T. As published by Dr. Rosenberg group, the neoantigen TCR-T achieved clinical response in 3 out of 7 patients with refractory colorectal cancers. Here, the autologous peripheral T cells are transduced with single-use personalized retroviral vectors for TCR, which was experimentally identified in neoantigen-loaded DC co-cultured with TIL grown from the fresh patient tumor. This process appears very complex, but actually the process have been validated here in National Cancer Center GMP in collaboration with Dr. Rosenberg. So then, what cell therapy modalities is to be considered for which type of solid tumor? Claudin 8.2 CAR-T may become the first FDA-approved CAR-T for solid tumors because they showed significantly higher response rate, 22% versus 4% for treating physician's choice, with longer progression-free survival, with very infrequent, just 5% of the grade 3 cytokine release syndrome. But not all common solid tumors express such relatively ideal surface antigens. So for those tumors, neoantigen-targeted TCR-Ts or TIL, which achieve clinical response in 43% and 24% of the patients, and the response lasted as long as 9 months, with at least as equivalently favorable toxicity profiles to Claudin 8.4 CAR-Ts, and those treatments may be considered as well. And please note that all these cell therapy modalities were not associated with the grave toxicity such as ICANS or hemophagocytic lymphohistiocytosis. So, is GP3 CAR-T worth pursuing? My answer is yes, especially as cytokine armored CAR-T, perhaps with intraarterial injection, which cell therapy is to be considered for each solid tumor type. So we think that the cell therapy is for immunologically cold tumors, and for tumors with strong homogeneous expression of membranous tumor-associated antigen, maybe CAR-T may be the first choice. And otherwise, neoantigen TCR-T or TCR-T or TIL may be considered. And for immunologically hot tumors, rather than CAR-T or TCR-T, autologous TIL or just second-line IO may be considered at the IO refractory state. But importantly, given that our institution just started a program under the leadership of our president, Dr. Yang, will support the investigator-initiated clinical trial, which we believe are very crucial for validating at early stage the clinical proof of concept of the novel cell therapies developed by Korean scientists. And also, those early phase clinical trials may help broaden patients' access to innovative cell therapy at significantly reduced cost instead of paying the full list price of the commercial cell products from global cell therapy companies. The greatest bottleneck of this approach may be the GMP production of lenti or retroviral vectors, but our program, led by Dr. The chair of this session, plans to provide lenti or retroviral vector either in GMP or non-GMP form free of charge to all program grantees. So I think that bottleneck may be reduced by this approach. Also, many solid tumors are heterogeneous and do not have the strong homogeneously expressed surface antigen. Neoantigen targeted approach may be furthered in this nation for broadening the patient access. And for that purpose, we are having extensive conversation with Korean FDA. And I'd like to take this opportunity to thank my God and NCI investigators such as the CAN Act Network Program Directors and Dr. Rosenberg for sharing SOPs and reagents for GMP-grade lenti or retroviral vectors with us. And thank you for your attention.
Thank you, Professor Kim. And actually, while we will start our discussion, maybe we have a discussion from from Jeonnam University, Professor Lee Jae-jung. I think that he can start the discussion with questions.
Due to the time limit, I briefly comment. So I would like to comment the biobench aspect. So we also preparing IND of dual CAR-T for solid tumor. Actually, what the most difficult thing to develop CAR-T is that the barrier to clinical trial entry is very high. It needs to be expanded to R&D for development of CAR-T for solid tumor cancer and domestic industrialization of viral vectors. Recently, the CAR-T project announced by NCC is expected to play a big role in promoting development of CAR-T for solid tumors. We are also thanks for the NCC. In the aspect of academic field, I think that there are 3 points to enhance the CAR-T therapy. The one is the improved efficacy such as T-cell exhaustion, antigen target tumor microenvironment needed to be improved. The second is to reduce toxicity. Actually, a lot of clinical trials performed in the world, so investigators try to reduce this kind of toxicity. The third one is to improve availability. Actually, currently it takes a very long manufacturing time, 6 to 8 weeks, and 10 to 15 of patients can receive can't receive this kind of treatment due to progression and death while waiting the CAR-T treatment. So it's recently more rapid CAR-T generation of autologous CAR-T and is in clinical trial, and also allogeneic CAR-T and in vivo CAR-T generation is also important. The final issue is the cost, very high cost. So actually I have several questions for the speakers, but it's due to time limitation, I will communicate with personal communication. Thank you.
Okay, Professor Kyung Kim, do you have a question to the previous three speakers? No? And then I think it's good time for us to take the questions from the audience. So if you have any questions. Okay, if not, I actually have a question to Professor Okazaki. And you showed, I think this is the last part of your presentation, you briefly introduced the bispecific antibody targeting LAG-3. And so can you explain a little bit more about that interesting entity?
So it's very simple.
Yeah.
We wanted to recruit LAG-3 to the immunological synapse.
Yeah.
Because we found the massive accumulation of the immunological synapse.
Yeah.
But still we don't know how LAG-3 inhibits T-cell activation. We expect LAG-3 to transduce specific signal and found some interesting motifs, but we're not completely sure how these motifs inhibit, transduce the signal. Yeah.
Okay, yeah, thank you.
Thank you.
If there are no more questions, it's time for us to close this session. Thank you very much for all the speakers and audiences.
That concludes session 2. We would like to thank all the speakers. Give a big round of applause. This concludes the morning session of the symposium. We will resume the afternoon session at 1:30 p.m. We kindly ask all the speakers, chairs, and invited guests to move to the seminar room on the 8th floor for lunch and follow the steps to move into the dining area. Lunch coupon provided by registrants may be used at the hospital cafeterias located at basement level 1 at the hospital building. Please present your coupon to have your meal. Please note that the coupon is valid only for today. For online participants, please click the exit button at the bottom of the live screen to record your attendance for the morning sessions. To rejoin the afternoon sessions at 1:30 p.m., click the enter button again. Alternatively, you may log out and log back in. For on-site participants, please tag your QR code at the registrant's desk upon exit to record your attendance. Thank you.
하나 둘 하나 둘 셋 밖으로 나오죠 지금 하나 네, 나와요 지금 하나 둘 하나 둘 셋 하나 둘 나오는데 하나 둘 셋 내 거에서 더 키워야 돼 하나 둘 여기서 제 거는 지금 이게 여기도 있어요 하나 둘 하나 둘 셋
하나, 둘, 셋. 하나, 둘, 셋. 총 양방의 테스트입니다. 근데 줌에서 어떻게 들리는지 봐야 돼. 아, 유튜브에서. 여기는 무한정 올릴 수는 있어. 게이지는 괜찮아, 그럼?
여기서 올려
마이크 테스트? 왜 안 들어가 또? 너무 작은데? 줌 양방향 테스트입니다. 5를 위한 양방향 테스트입니다. 왜냐면, 아니, 이게 지금 봐봐. 마이크도 어디 갔어? 이거. 말해봐. 말해봐. 이 정도 볼륨 레벨이야? 마이크 테스트? 테스트. 짜고. 줌 양방향 테스트입니다. 5 세션을 위한 양방향 테스트입니다. 여기만 키워주면 돼, 그럼? 너무 잡음이 많어. 양방향 테스트입니다. 아니겠지. 5 세션을 위한 양방향 테스트입니다. 이거 아까 오전에 이 정도 하고도 크게 들렸어. 아까 그 줌에서 말씀하실 때. 아까 이 볼륨이었어, 이 볼륨. 그럼 혹시
Good afternoon from Boston, Massachusetts. I greatly appreciate the invitation from Professor Young and Professor Young to give this lecture. I'm extremely sorry that my schedule won't allow for me to travel to Seoul to be able to deliver this talk in person. These are my disclosures, and they're not related to the content of today's talk. Before I begin, I'd like to express my deepest appreciation to Professor Young. I estimate that he has 10,000 students or students of students worldwide, and I feel privileged to be one of them. I'll always
바뀐 게 없는데? 뮤트한 것도 없고. 아니야, 아까 전에 이대로 지금 끊긴 것도 없어.
근데 왜 영상이 소리가
네가 뭘 바꿨겠지?
네? 안 바꿨다니까요? 그래서 마이크로 그게 아니라
맞아. 단상났어, 말 좀.
Good afternoon from Boston, Massachusetts. I greatly appreciate the invitation from Professor Young and Professor Young to give this lecture. I'm extremely sorry that my schedule won't allow for me to travel to Seoul to be able to deliver this talk in person. These are my disclosures, and they're not related to the content of today's talk. Before I begin, I'd like to express my deepest appreciation to Professor Young. I estimate that he has 10,000 students or students of students worldwide, and I feel privileged to be one of them. I'll always be grateful. Professor Young asked me to address what is the future of surgical oncology. I don't have a crystal ball, but I'll do my best to give you my perspectives. What do we do well currently? There's a lot on here, but in short, we've made a lot of progress in cancer outpatient management, operations, electronic medical records, and online resources, standardization, education, and recognition of global needs. Yet there's no shortage of opportunities. To highlight a few, as shown in red, we still have yet to utilize the full potential of robots. Electronic medical records have not harnessed AI, and education can be remarkably improved with AI. Some think of a dynamic between opposing forces of medicines and surgery, but the landscape is much more complex. Many forces, some shown here, will impact surgical oncology. Cancer interception.
하나 둘 셋. 하나 둘.
Ladies and gentlemen, we will now begin the afternoon session. If you are in the lobby, please have your seats in the main hall. For online participants, please click the check-in button to record your attendance. For on-site participants, if you have not scanned your QR code upon re-entry, please proceed to registration desk in the lobby to ensure your attendance is properly recorded. We will now begin Keynote Lecture 2. This session will be chaired by Dr. Keun-seok Lee from National Cancer Center, Korea. Please join us with him with a big round of applause.
Welcome to the keynote lecture. Did you have a good lunch? Yeah, I'm Dr. Keun-seok Lee, Director of Hospital National Cancer Center, Korea. It is my great pleasure to welcome you to this keynote session. We are honored to have with us today a true pioneer in surgical oncology, Dr. Kenneth K. Tanabe, Professor of Surgery at Harvard Medical School and co-chief of the Division of Gastrointestinal and Oncologic Surgery at Massachusetts General Hospital. He also holds the ESCO Slater Family Chair in Surgical Oncology. Dr. Tanabe has made remarkable contributions to cancer surgery, research, and education over the past 3 decades. He has published over 200 peer-reviewed articles, served on numerous editorial boards, including Annals of Surgical Oncology, Cancer, and Up to Date, and currently serves as President-elect of the Society of Surgical Oncology. His work continues to shape the future of the cancer care, from pioneering clinical trials to mentoring the next generation of surgical oncologists. Today, he will present a keynote lecture titled The Future of Surgical Oncology. The presentation will be delivered as a pre-recorded video, and Dr. Tanabe is currently standing by online to join us for a live Q&A session immediately after the video. Now, let us begin the video presentation of Dr. Tanabe's keynote address.
Good afternoon from Boston, Massachusetts. I greatly appreciate the invitation from Professor Young and Professor Young to give this lecture. I'm extremely sorry that my schedule won't allow for me to travel to Seoul to be able to deliver this talk in person. These are my disclosures, and they're not related to the content of today's talk. Before I begin, I'd like to express my deepest appreciation to Professor Young. I estimate that he has 10,000 students, or students of students worldwide, and I feel privileged to be one of them. I'll always be grateful. Professor Young asked me to address what is the future of surgical oncology. I don't have a crystal ball, but I'll do my best to give you my perspectives. What do we do well currently? There's a lot on here, but in short, we've made a lot of progress in cancer outpatient management, operations, electronic medical records, and online resources, standardization, education, and recognition of global needs. Yet there's no shortage of opportunities. To highlight a few, as shown in red, we still have yet to utilize the full potential of robots. Electronic medical records have not harnessed AI, and education can be remarkably improved with AI. Some think of a dynamic between opposing forces of medicines and surgery, but the landscape is much more complex. Many forces, some shown here, will impact surgical oncology: cancer interception, cancer prevention, the efficacy of non-surgical treatments, and finally, the evolution of surgical treatments. These will all shape the future of surgical oncology. One strategy for cancer prevention is prophylactic removal of the target organ. Discoveries in this domain have had a major impact on cancer surgery, with increasing numbers of operations performed to prophylactically remove target organs. We see more gastrectomies for CDH1 mutations, mastectomy and oophorectomies for BRCA mutations, and colectomies for various hereditary predispositions. Patient anxiety over a future diagnosis of cancer is on the rise, and additional genetic determinants of hereditary predisposition will be discovered. The number of these operations will increase in the coming decade. Primary cancer prevention campaigns have proven effective. I've listed several of them here, including tobacco, HBV, and HPV. They are all important, but truthfully, they will continue to have only a modest impact on cancer surgery volume. The future in this avenue requires greater understanding of molecular pathways to enable cancer prevention with medications, like tamoxifen for prevention of breast cancer. Let's compare risk identification and reduction in breast cancer compared to hepatocellular carcinoma. In breast cancer, we have a clinically defined population at risk, namely those with a positive family history. We've identified genes that account for hereditary predisposition. We have a good screening test in mammography, and we have surgical risk reduction with prophylactic mastectomy. We also have a very good prevention drug, tamoxifen. If we compare that to HCC, we have a clinically defined population, namely those with chronic liver disease. We don't have a genetic basis for hereditary predisposition. We have a good screening test with ultrasound and AFP. We have surgical risk reduction with liver transplant, but we don't have a good drug prevention approach. I would like to show you a glimpse of some of my research team's work in this area. To summarize a decade of our work, we've shown that erlotinib, an EGFR inhibitor, effectively prevents HCC and fibrosis in preclinical models. The upper left shows livers from rats exposed to a carcinogen DEN over 18 weeks. They develop cirrhosis and HCC. But if we intervene at week 12 and block EGFR signaling with erlotinib, we see two main effects. First, we see a reduction in the number of tumor nodules. Second, we see a reduction in the severity of cirrhosis as evidenced by improved or declining Ishak score. So erlotinib could be a wonder drug, reverse cirrhosis and prevent HCC at the same time. And erlotinib is already FDA approved for lung cancer, so it could be simply repurposed for treatment of cirrhosis and prevention of HCC. But the ideal drug prevention agent should be an inexpensive, effective, orally administered, have an accurate biomarker for efficacy, and have few side effects. But the problem with erlotinib is well known, whereby most patients receiving this for lung cancer develop a rash. This would be unacceptable in the prevention setting. But maybe 150 milligrams per day, the oncology dose, is not required to block signaling in the liver. So we conducted an erlotinib dose de-escalation study to determine the minimum erlotinib dose that would decrease liver EGFR signaling in cirrhosis. In this clinical trial, we obtained liver tissue before and after erlotinib, which was administered for only 1 week. We de-escalated from 150 milligrams per day all the way down to 25 milligrams per day, and as endpoints, we measured phospho-EGFR in the liver, and we measured liver gene expression. We successfully lowered the dose down to 25 milligrams per day. Minimal side effects were observed at these lower doses, and by immunohistochemical staining of livers, we showed that this dose blocked EGFR signaling in the liver. And gene expression changes were favorable for reversing cirrhosis. So this low dose can proceed in a larger prevention study. I previously used the term cancer interception, and so I want to return to that theme to point out that one form of cancer interception involves blocking progression of cancer. This includes use of SERMs like tamoxifen to reduce the frequency of cancer, or colonoscopy to remove polyps before they turn fully malignant. These maneuvers generally decrease the need for cancer surgery. Conversely, another form of cancer interception is cancer screening. By earlier detection of cancer, the result is more patients detected at a stage when they are operable and curable, and this increases cancer surgery volume. I'd like to change gears and talk about surgical de-escalation, which is a consequence of effective treatments. For gastric cancer, endoscopic mucosal resection in early gastric cancer has reduced the need for gastrectomy. And because of effective adjuvant therapies, we have de-escalated surgery in prostate cancer, rectal cancer, melanoma, breast cancer, and as indicated on the slide, perhaps next on the list will be solid tumors with mismatch repair deficiency. Here's a nice review written by 2 radiation oncologists at Yonsei University showing results of watch and wait for treatment of rectal cancer after total neoadjuvant therapy. The text is too small to read, but instead I simply wanted to give you an understanding that there are results from many clinical trials that now support watch and wait as a viable pathway. And I also wanted to show it to you because it was written by a couple of Korean radiation oncologists. Regarding melanoma surgery de-escalation, we used to perform total lymphadenectomy for patients with enlarged regional nodes. On the basis of many clinical trials, including PROTO, we have de-escalated this and now perform only what is called an index node biopsy. In melanoma patients with enlarged regional nodes, we have radiology place an RFID tag in the enlarged node, which is called the index node. The RFID tag enables us to locate and surgically remove the node after neoadjuvant therapy. Patients are then treated with neoadjuvant immunotherapy, following which we remove just the index node. The pathologist grades the response in the index node. The frequency of various responses observed in PROTO clinical trial are shown in the pie chart. Pathologic complete response and near complete response are shown in the green and account for a majority of patients. The full schema followed in PROTO is shown here, but if you focus on the pathway at the top, if patients had an MPR, a major pathologic response, they did not undergo the standard total lymph node dissection, and they received no additional surgery or treatment. And that represents major surgical de-escalation. These patients with major pathologic response did very well, with their overall survival shown in the green line on top. This is the timeline of surgical de-escalation in melanoma. In the 1970s, all patients had a total lymphadenectomy for staging. In the mid-1990s, sentinel node biopsy replaced total lymphadenectomy based on the MSLT1 trial. So the only patients that underwent total lymphadenectomy were those with positive sentinel nodes. But by 2017, those operations were discontinued in favor of ultrasound monitoring of lymph node basins on the basis of MSLT2 clinical trial. At that point, total lymphadenectomies were performed only for clinically enlarged palpable node metastasis. But now we are not even doing that. Based on clinical trials like PROTO and S1801, after neoadjuvant therapy, we perform only an index node biopsy. What is the future? Certainly it's possible that rather than surgical retrieval of nodes and PET imaging for staging and treatment of melanoma, patients in the future will have only gene expression profiling of their primary tumor, followed by systemic therapy. And this would eliminate surgery completely from the treatment of melanoma. What will be the impact on cancer surgery when a very effective systemic therapy is available? This interesting report was just published 6 weeks ago in the New England Journal of Medicine. Patients with solid tumors with defective mismatch repair were treated on a clinical trial with dostarlimab, immunotherapy for 6 months. Patients with a complete clinical response could then choose to forego standard surgery and instead be followed carefully. In the first cohort, rectal cancer patients, as shown in the blue on the left, all 49 patients had a complete clinical response and chose to forego operation. In the second cohort, Urothelial cancers, HPV cancers, colon cancers, and gastric cancer patients were included. Remember, these patients all had mismatch repair deficiency and were treated with immunotherapy. This plot shows duration of follow-up, and the stars that are perhaps too small to be seen represent recurrent disease. In the rectal cancer cohort, the 2-year relapse-free survival was 92% with a median follow-up of 20 months. In the non-rectal cancer cohort, the 2-year relapse-free survival was 85% with median follow-up of 15 months. For mismatch repair defective solid tumors, immunotherapy rather than surgery may be the future. What will be the impact of surgery de-escalation on cancer surgery training? This is the timeline showing the history of de-escalation of axillary surgery for breast cancer. It is similar to melanoma. Full axillary surgery is performed much less commonly, and in the future, even sentinel node biopsy may be eliminated. But currently, the SSO breast surgery fellowship training requirements state that fellows must have completed 10 cases of axillary lymph node dissection. While this remains achievable, it is getting more and more difficult in some fellowship programs. The future of surgical oncology will also incorporate image-guided intraoperative detection of tumor cells. The problem in breast cancer surgery is that lumpectomy is associated with positive margins in 20 to 40% of operations. A solution has been developed for intraoperative detection of residual cancer cells by bioluminescence. Dr. Barbara Smith at MGH conducted a randomized clinical trial that involved intraoperative randomization to either inspection of the lumpectomy cavity or inspection of the lumpectomy cavity with a dye that lights up in response to cathepsin D when a probe is placed into the cavity. This team demonstrated intraoperative detection of residual cancer cells at margins allowed for intraoperative re-resection of those margins. And this reduced the frequency of positive margins. And on the basis of this randomized clinical trial, the system received FDA approval last year. Some examples of images from lumpectomy cavities are shown, and the red spots, some very tiny and some very large, call attention to areas that needed to be re-resected during the index operation to achieve negative margins. What is the future of robotic surgery? We'll have more robotic platforms with better precision, tremor elimination, and better visualization. The future is exciting, and I'd like to show you some glimpses of the future with a very, very big thank you to Dr. Dan Hashimoto, a former Mass General resident who is now at University of Pennsylvania and director of the Penn Computer Assisted Surgery and Outcomes Laboratory. He provided to me all of the material on the following several slides. AI algorithms can perform complex tasks, as seen here in a version of Tesla's autopilot. The screens on the right demonstrate how cameras and sensors can automatically detect objects, lanes, and other things. If this technology can be used in cars, surely it can be used for robotic surgery. Work performed through the Global Surgical AI Collaborative was the first to demonstrate that computer vision can be used to train models to identify safe and unsafe areas of dissection. In the pilot example, they demonstrated that labels from experienced surgeons could train a GPS-like system, as seen on the right, where the safe area of dissection is in green and the unsafe is in red. This work is in near real time and is now in clinical trial at the University of Toronto. To avoid bile duct injuries, here we see a heat map of where the model suggests that the surgeon should dissect. But the surgeon is dissecting lower than the suggested area and creates a common bile duct injury. Other groups, such as Nicolas Padoy's group at IHU Strasbourg, have demonstrated that computer vision can be used to identify the critical view of safety and demonstrate clear anatomic criteria in real time to act as an additional check on visualization. Companies such as Ano in Japan are now demonstrating how computer vision can be used to highlight the areolar tissue plane in dissections such as laparoscopic robotic colectomy. Ongoing work at Penn is now focused on specifically tracking the instrument needle tissue interactions to better predict clinical outcomes in cases such as robotic pancreaticoduodenectomy. This leads towards a question of whether autonomous robotic surgery is possible. Work here from Professor Ken Goldberg at UC Berkeley demonstrates it is possible, but still quite slow. This particular video is from over 5 years ago. More recently, Professor Axel Krieger at Hopkins demonstrates that explicit instruction may not be needed long-term, and that autonomous robots may learn from imitating maneuvers demonstrated by surgeons. However, this is also quite slow as of yet, and we will likely see a long horizon before implementation. These are the folks in Dr. Dan Hashimoto's laboratory who contributed to this work. Switching gears, organ transplant is a very good solution for HCC and perhaps other tumors, but the demand versus supply has been an insurmountable problem. One solution is tissue engineering, which involves use of stem cells and bioprinting. This has been used for liver, and stem cells have been used to regenerate breast tissue, but these are still a long way off from clinical implementation. Another solution is use of genetically edited pig organs. This involves multiplex gene editing with 69 gene edits for glycogen antigen knockout, human gene insertions, and endogenous retrovirus inactivation. The pigs are raised in specialized facilities, and the first xenograft was performed at Mass General in March of 2024 and reported on the front page of the New York Times and subsequently in the New England Journal of Medicine. Better outcomes are seen after transplant for HCC compared to resection, but graft availability limits use of liver transplant. Genetically engineered pig livers could be used, and this would dramatically reduce the number of resections for HCC. And emerging data show value of liver transplant for some patients with colorectal carcinoma liver metastasis. The survival graph on the right is taken from the TransMet clinical trial. In brief, that trial studied patients with unresectable colorectal carcinoma liver metastasis, and they were randomized to liver transplant plus chemotherapy versus chemotherapy alone. They were transplanted within 2 months of their last chemotherapy treatment. The 5-year overall survival comparison is shown on the top right by intention to treat, and the bottom right shows the survival curves by protocol. Survival in the transplant group was excellent and on par with transplant for benign conditions, making transplant feasible for colorectal carcinoma cancer liver metastasis patients. We will see more transplant for colorectal carcinoma liver metastasis, and the availability of pig livers in the future could push this even further. A last topic in brief is artificial intelligence and deep learning. As physicians, you look at a patient's face and you ask yourself, how old does this patient look, regardless of chronologic age? Face Age is an AI tool trained on 60,000 healthy people and cancer patients. Face Age can determine age based on a picture of your face. The older you look on Face Age, the shorter your lifespan. Here is an example of a patient who is 92 years old, yet his face age is 66. If you take a look at him, you think it might have to do with the smile, but you cannot fake out the system with simply a smile. Yu-Hyun is a reporter for the Boston Globe, and simply by smiling, she did not fake out face age. Face age indicated that with a chronologic age of 35, her face age is 28, whether she smiled or didn't smile. In a published report last month in Lancet Digital Health, Face Age improved physicians' survival predictions in patients with incurable cancer who were receiving palliative treatments. and all it took was one current photo of the face. In conclusion, here are my predictions for the road ahead in surgical oncology. Surgical de-escalation will continue. One consequence will be changing of the training paradigms with more simulation and use of competency scoring like entrustable professional activities known as EPAs. Robots will become more capable and less expensive. We will use real-time cancer imaging intraoperatively and will use AI more frequently at our fingertips. Beyond that, say more than 8 years down the road, in clinical practice, we will be commonly using bioengineered replacement organs. We'll have eliminated operations completely with systemic therapy. Robots will be more autonomous, including robotic functions like automated knot tying and suturing. And AI will bring us more international harmonization as the language barriers will essentially disappear. There will be a global benefit to low and middle income countries, but it will lag behind. Thank you very much. It's really been a privilege and an honor to give this lecture.
Thank you, Dr. Tanabe, for the insightful and forward-looking presentation. Now, we are pleased to invite Dr. Tanabe to join us live for a Q&A session. If you have any questions or comments, please feel free to use the microphone. Yeah. President Yang.
Dr. Tanabe? Dr. Tanabe?
Doctor.
Are you inactive? Dr. Tanabe, thank you very much for your wonderful, really insightful lecture. And clearly you show us which area we have to pay attention for as a surgical oncology. And, just one simple question, as you mentioned, many of our data are key for future development. And, for example, surgical device you're using, and so surgery is a behavior, recording of a surgery is a very key for our understanding of some procedures for the safety and for evaluation, things on. And in our my previous institute, so national, we asked our all patients to approve for our recording and use for our research program. But I heard in the United States, many institutes, you are deleting surgical video 1.5 months or 2 months later. So how was your Harvard engaged policy about the data, the video data, because it's so big, day by day, it's getting bigger. How do you handle this precious data?
Yes, thank you very much for bringing up this topic. We are slowly improving the volume of data that we accumulate in recording operations. The two rate-limiting steps are that, number one, not all of our operating rooms are outfitted with recording devices. That number is, or that frequency is increasing. Of course, all the robots automatically track the data. We've improved the consent process whereby basically embedded in the surgical consent form are statements that a patient provides consent for us to record the operation and use it for educational purposes. And so the size of our data collection on this is increasing exponentially. I think that this is going on across the country as well. And so large data sets will become available. Some large data sets are already available, and others will become available very soon. You bring up a very important point, which is that the ability to perform AI deep learning requires a very robust data set, and robust means large. And so having thousands and thousands of operations to train AI is critically important.
Question about clinical trials in surgery. Our gold standard is a clinical trial comparing standard operation, standard treatment versus a new treatment. And as you know, colorectal cancer, gastric cancer, or breast, we have heard we have many important clinical trials that show that the comparison studies. But ironically, liver surgery, that's one of your specialty, and we saw many publications of different way of resection, but not much about some comparison studies. Do you any future expectation of some concept of a clinical trial in surgical oncology, or any special reason for liver surgery has a limited some reason for why we don't have some representing clinical trials?
Yeah, thank you very much for that thought. Very broadly, the trend in cancer surgery is to move towards neoadjuvant therapy, and neoadjuvant therapy has significant impacts on operations. As I'd mentioned, de-escalation or reducing the radicality of an operation following neoadjuvant therapy, the advances in that area have been huge. And I expect that to continue, and I expect that to gradually involve liver surgery, whether it's for colorectal carcinoma liver metastasis, hepatocellular carcinoma, or cholangiocarcinoma. Of course, neoadjuvant therapy requires having an effective systemic therapy. And as you know, the two largest trends in systemic therapy are immunotherapy and molecularly targeted therapy. Cholangiocarcinoma and hepatocellular carcinoma are cold tumors. They are not immunologically reactive, but we are also seeing many breakthroughs in making cold tumors hot tumors. I didn't show some of the data from our own laboratory where we're able to demonstrate that collagen is an important factor in pancreatic cancer, changing it from a cold tumor to a hot tumor. But I think that the future in clinical trials in HCC and in cholangiocarcinoma will be moving systemic therapies from postoperative to preoperative, and thereby changing the operation. And I think those therapies are going to be molecularly targeted, like FGFR receptor inhibitors are a good example of a molecularly targeted therapy in cholangiocarcinoma, and new immunotherapies. The clinical trial that I showed from the New England Journal of Medicine in patients with mismatch repair deficient tumors, solid tumors, is really remarkable, whereby a very large fraction of patients had a complete clinical response and forego operation, and their clinical outcomes were outstanding. So bit by bit, I think that systemic therapy will end up being combined neoadjuvantly with surgery and will change the nature of the operations that we perform. I think those are the clinical trials that we'll see in the very near future.
Yeah. Is there any comments or question? Okay. If not, this concludes our Q&A session. On behalf of all participants, I'd like to express our sincere gratitude to Dr. Tanabe for taking time to join us today and for sharing his expertise and vision for the future of surgical oncology. Thank you again, Dr. Tanabe.
Thank you very much. It's been a pleasure. And again, I'm sorry that I can't be there in person. I wish you the best for the remainder of your conference today.
Thank you, Dr. Gunseok Lee, for chairing the keynote lecture session. Please give him a big round of applause again. Now we will begin session 3 of today's symposium. The title of session 3 is Clinical Trials in Cancer Research, and it will be co-chaired by Professor Tae-young Jang from Hallym University College of Medicine and Professor Chan-gyu Kim from the National Cancer Center, Korea. Please welcome both chairs with a round of applause.
So now time to start section 3. Section 3 is a clinical trial in cancer research. First presentation is Dr. Na. Dr. Na is a professor of Yonsei University, and he is also chairman of Korean Cancer Association now, and he is a very excellent and diligent clinical researcher and international opinion leader of gastric cancer treatment. Dr. Na, please.
Thank you so much for the kind introduction. It's my honor to give this talk in this important symposium. I learned a lot in the morning session, and now it's a bit of a clinical, but as a clinician also, I will say a bit about the translation research part. So I don't know who of them are focused on the gastric cancer, but gastric cancer is a bit different from usual lung cancer, which has a lot of new agents based on the technology and the new targets. Unfortunately, gastric cancer does not have many targets, and also the drug is not working as we expected. So for last 20 years, there are more failures than the success in the drug development. And also in the gastric cancer, there are two different biology subgroups. One is HER2 positive and one is HER2 negative. And HER2 positive, trastuzumab is important as a targeted agent in the first line. And after almost 15 years, we have TDXD, which is a ADC targeting HER2. It's been approved. And also we have a new drug, immunotherapy, pembrolizumab, has been approved as a standard of care. In HER2 negative, most of them are cytotoxic, and adding cytotoxic with ramucirumab has been approved 2017, and then now we have immunologic agents, nivolumab and pembrolizumab is adding as a treatment in the first-line treatment. So how to improve these outcome of the metastatic gastric cancer? So various cytotoxic agents combinations are there, but still the outcome is limited. And the targeted agents, as I said, HER2 targeting and angiogenesis inhibitor has been standard of care, and now it's moving to novel agents and various novel combinations. So precision medicine means with the technology improvement, and then new targets are coming and the new drugs are available. So now we have novel targets, Claudin 18.2. It is very hot topic in the gastric cancer area, and the FGFR will be the next target. And immune therapy is also a part of it. So currently we have, as I said, compared to 10 years ago, gastric cancer has more options, but still there are many problems and the challenges. So when you think about the precision medicine, we should think about the hallmarks of the cancer. So there are more than 10 subcategories of the hallmarks, and we have HER2 targeting and ramucirumab has been successful, but most of our other agents has been failed. And we have new agents of Claudin and FGFR. So this is the older summary. And the new agents of immune checkpoint inhibitor and also the novel agents are coming. So just briefly touch that each target subgroup. So for the HER2 positive, trastuzumab has been approved, but unfortunately, pertuzumab, TDM1, those are not successful, different from breast cancer. But we have a TDXD, the new ADC, you know that with that HER2 targeting and the topoisomerase inhibitor. And we have novel bispecific inhibitor, bispecific antibody, which is a zanidatamab. It has a kind of a combination of Herceptin and the pertuzumab. So currently we are waiting for the phase 3 trials with zanidatamab and also the margetuximab or the B-cell vaccine, or even the cell therapies targeting HER2 is developing a lot. And how about the angiogenesis inhibitor? Most of the angiogenesis inhibitor has been failed, but one drug is ramucirumab is approved as a second line. So compared to monotherapy chemo, combination improved the survival. But not only for that, we have various TKIs, and currently the TKIs are combined with the IO therapy because we know that VEGF signaling leads to immunosuppressive status in the tumor microenvironment. So we have more and more information that angiogenesis induced tumor microenvironment through that, the various Tregs or the TAMs like that. So currently, some of the combination with the regorafenib, nivolumab, or lenvatinib and pembrolizumab, that type of various combinations has been under development. And recently, this year, we just presented this lenvatinib and pembrolizumab combination. So this is comparison of doublet chemotherapy versus doublet chemo and the pembrolizumab, immune checkpoint inhibitor, and the angiogenesis inhibitor lenvatinib. So in the induction phase, chemo and the IO targeted agents are combined for 4 months. And then after the induction phase, we change it to len-pem combination as a consolidation period. So this is try to make a big tumor control in the beginning and then maintenance with a lower toxic regimen. But still, lenvatinib is a TKI with various toxicities. So that is some issues we have some phase in this trial. So in the when we see that efficacy, the interim analysis, PFS in all patients and also the patients with PD-L1 high patients show the significant improvement. And anti-tumor response is also improved from 44% to 58%. So there is a almost 15% of response rate improvement. But in the final analysis at the OS show that there is no significant improvement. So you can see that all patients and also the CPS over 1 until 15 months, there is no big difference. In the end, some of the patient has a stable long tail of the outcome. So this study has been failed with that, even though it's a big phase 3 trial. So there are some of our learnings. We are questioning about the role of angiogenesis versus role of lenvatinib. I think still angiogenesis is very important. You will see some of the data from now on, but not only for that, the problem is lenvatinib as a TKI has a toxic. So sometimes the combination with a cytotoxic with a TKI has difficulties in full dose intensity. So because of that, when we have that analysis of dose intensity is really less than 50%. So that is one of the issues. And is there any role of chemotherapy-free regimen in the gastric cancer treatment? So gastric cancer is always needed a kind of a cytotoxic part. So still that's a very difficult area to move to just targeted agents. And also proper dose intensity is an issue. So in the gastric cancer, always think about not only for the biology, but also basically those are not very oncogenic dependent compared to other lung cancer or the breast cancer. So non-immune targets, as I said, FGFR and Claudin. So Claudin is in the adhesion molecule, and FGFR is one of the target molecule in the receptor tyrosine kinase. So Claudin 18.2 is very specific, expressed in the gastric cancer and the pancreas cancer. And as you see, this is adhesion molecule. So usually it's inside of the body, but if that structure is destroyed, it goes up in that tumor surface area. So they found out that gastric cancer has increased Claudin expression. So there is antibody, zolbetuximab, targeting Claudin 18.2. This is strong ADCC and CDC induced in the Claudin positive tumor cells, and also show that synergy with the cytotoxic chemotherapy. So there were two big clinical trials, Spotlight and Glow. These are two studies, Spotlight and Glow. These are both very similar. The only difference is chemotherapy regimen. So this is comparing chemotherapy versus chemotherapy with zolbetuximab. So this is comparing the efficacy of zolbetuximab adding on top of chemotherapy. And the data show that it's a combined population. As you see, PFS, OS, all those are significantly improved the survival. And as you see, the outcome of overall survival in this patient is over 16 months, which is better than previous data. And the objective response rate is very similar. And these populations are Claudin overexpression, which is a criteria about the 75% positivity. And not only for the PFS and OS, the 2-year PFS and OS are increased. The problem is this is a normal mucosa also expressed in the adhesion as an adhesion molecule. So when we see the toxicity profile, you see that the nausea, vomiting, and decreased appetite is really high in this zolbetuximab adding arm. So usually it happens just after injection. We can see vomiting within an hour. So without pre-medication, it's really difficult to control, but we know how to manage those nausea, vomiting. So with that aggressive nausea, vomiting supportive care, now it go down to like 20% something. So currently one message from here is not only for the gastric, all those tumor area with the novel agents, TKI antibody or the different targeted agents, immune agents, all they have very specific, unique toxicities. So we should understand about how to manage those toxicity properly is really important to maintain dose intensity of those regimens. So it's very important message for the clinicians. Then this is the one example of the very complicated drug development of targeting Claudin. So monoclonal antibody, ADC, bispecific antibody, CAR-T, only for the Claudin targeting in the gastric cancer, there are more than 25 new agents under development. So this is very lucky for gastric cancer. It never happens before, but it will be more and more new drugs are coming. And the FGFR is one of the another target. That is, we've known that FGFR has been amplified in the gastric cancer. It's usually FGFR2 amplification, and also the FGFR overexpression is there. So there's a company made that anti-FGFR antibody, bemarituzumab, that is also the antibody-induced ADCC and the NK cell activity. So in the phase 2 trial, in the FIGHT trial, they show that the benefit of adding bemarituzumab on top of doublet chemotherapy in PFS and OS. But when we see in detail, the survival benefit is more significantly improved in the highly overexpressed. So just not overexpressed is not enough. And now we know that it's more than 10% of tumors should have a strong positivity. So we moved to the phase 3 trial for targeting these highly overexpressed patient population. And currently we are waiting for the confirmatory phase 3 trial. So that means in the gastric cancer area, for the targeting agents, one is HER2, and next one is Claudin. It's been there. And this year, next year, we will have FGFR targeting agents, a new drug. So this is really good progress. And then move to the immune target agents. So for the PD-1, PD-L1 inhibitor, as I mentioned that nivolumab, pembrolizumab has been approved as a first line. But not only for that, we have a PD-L1 inhibitor or sintilimab, all those are tislelizumab. There are more and more drugs are coming. But not only for that, we have other immune targets. So we know inside of an immune checkpoint, not just a PD-1, PD-L1, but also LAG-3, TIM-3, these type of immune checkpoints are very important. But the reason why I make it dark is this has been failed in the gastric cancer. So LAG-3, TIM-3 is not that effective compared to PD-1 inhibitor. Then currently coming with a TIGIT or other markers. So the reason why I showed you that before is try to make a story more easily. So in the HER2 positive, not only for the HER2 negative, in the HER2 positive in the first-line setting, the standard of care is doublet chemo and the trastuzumab targeting HER2, but on top of that, adding pembrolizumab, immune checkpoint inhibitor, improves the survival. So this is a very important study, Keynote 811, to change the clinical practice. So with that, we have a new standard of care. But the problem is when you see that the subgroup of that, PD-L1 high, which is a tumor-inflamed tumor microenvironment, has a significant improvement with pembrolizumab adding. But the patients with a PD-L1 low, with a cutoff of 1, there is no survival benefit. And when we see the survival curve, it is worse than the standard of care. So even though the numbers are small, there is a problem of adding pembrolizumab. So different from HER2 negative, HER2 positive, PD-L1 negative patients should not be treated with an immune checkpoint inhibitor. And then the question is, what's the problem in that tumor microenvironment? So we did the single-cell RNA sequencing data with gastric cancer samples. So 41 primary gastric cancer samples with 15 HER2 positive and 26 HER2 negative, and all those are no dMMR cases with PD-L1 information, and we did that single-cell sequencing with freshly frozen tissues. So we confirmed that the cell types are similar between HER2 and HER2 negative, but we figure out in the HER2 positive, there are more endothelial cells and fibroblasts in HER2 positive and PD-L1 negative subpopulation. So this is what I mentioned in the beginning about angiogenesis important in the tumor microenvironment with immune suppression. So in the HER2 positive, PD-L1 negative and PD-L1 positive has a different TME. We can start to think about that. So we go into that more in detail. In the beginning, about that HER2 negative and comparison of HER2 negative and HER2 positive, we figure out HER2 negative has a more active immune environment. So it has a more higher number of CD8 cells and also the higher TCR clonality in HER2 negative, and also the TCR diversity is high. And not only for that, when you see this drug cell-cell interaction, in the HER2 negative, the cell interaction mostly with immune cells. But when we see the HER2 positive, looks like various cells are there. And the interesting point is Treg, mono-like T-stem cells, and the CAF cells, endothelial cells. So the cells are there, but those are immunosuppressive cells. So we can see HER2 negative and compared to HER2 positive, HER2 positive is more immune desert. And even inside of HER2 positive, the patients with a CPS less than 1 has more immunosuppressive. One example is in the patients with a PD-L1 low, the JAK-STAT pathway has been enriched compared to PD-L1 high. That means these are the inducing terminally exhausted CD4 cells, CD8 cells, and also we confirmed that not only for the HER2, but inside of HER2 positive and PD-L1 negative, there is a higher CD4 Tregs are there, and also there is a pro-angiogenic TAMs are higher in HER2 positive, CPS negative. These are related to the angiogenesis. So we check the angiogenesis score, and also this population has a higher angiogenesis. So it makes the whole story very similar that HER2 positive, PD-L1 negative is immune desert. And that is related to angiogenesis and the increase of fibroblast. And not only for that, some of the immune checkpoint inhibitors are different. In the HER2 positive, PD-L1 negative has a very high active with the TIGIT pathway. So not only for the TIGIT, CTLA-4, even LAG-3, TIM-3 are higher in this population. So that means these patients might not be enough with a PD-1 inhibitor. We should think about different immune checkpoint inhibitors such as anti-TIGIT antibody, and also Treg TAM inhibition or anti-angiogenic inhibition might be important for this subgroup of patients. Then we should think about the TIGIT. So I'm not sure, some of the lung cancer experts are here, but there are some data showing that TIGIT antibody has been also failed in other tumor types, including lung cancer. So there are some skeptical about TIGIT in combination in the gastric cancer, but until now we have a phase 2 data. So Edge Gastric trial is doublet chemotherapy with DOM enzyme. This is anti-PD-1 and anti-TIGIT antibody combination. And it has a very good response, and the PFS is over 10 months. So now we finished the phase 3 enrollment about thousands of patients in the comparing Nivo versus DOM-ZIM combination in this population. So this is, we are waiting for the data. And also another example is Gemini gastric trial. It's an early phase trial, but this is a bispecific antibody targeting TIGIT and PD-1. So currently it's testing in the combination with the cytotoxic and also showed a very good response. So it looks like there is some adding effect of anti-TIGIT antibody, but we are not sure. What if that phase 3 trial shows the negative? So we know some of the markers are there, but the activities are different in the tumor type. So we should wait for that the phase 3 trials. But these improved technology make us more understanding about those areas about the problem, and subgroups are different from different TME, different from others. And the other example is DKK1. This has been known as a stem cell property and AKT signaling activation marker, but it also has an inducing MDSC expansion and the immunosuppressive environment through CD45 cells suppression. So the drug has been developed, the TKN01 is anti-DKK1 antibody. And in the earlier study, showed that a good response with IO combination, and in the earlier phase 2 study, showing the significant improvement in the survival in the gastric cancer. So we started this Distinguished trial as a first line, as a randomized phase 2 study combining chemotherapy, pembrolizumab or tislelizumab, and adding DKN01. The problem is the data is in the middle of the interim analysis, didn't show good. So we finished the study enrollment as March of 2025. So even though the biologists are there, the outcome is not always what we expected. And also the other immune agent is CD47 SERP axis inhibition, which is suppressing macrophage activation. This is one of the novel agents, CD47 blocker, ALX oncology drug. And we started this phase 2 trial of ASPEN-06 trial in HER2-positive setting, comparing the chemotherapy versus chemo with this new agent. And the response rate seems to be improved compared to 26, and the adding the macrophage inhibitor is 41% and shows a bit of the improving survival. But this is a very smaller number of the patients, and also we confirmed that not all the patients with the HER2 positivity has been confirmed patients are better outcome. So this is one of the examples that serial biopsies to understand about the target and also the microenvironment information is important for the success. So in summary, this is a very good period for the gastric cancer treatment, and we have more information about the biology, and we will have more potential drugs. But the problem is still many failures, but compared to 10 years, it's better because we have more and more technologies to explain about those microenvironment and tumor cell characteristics. So hopefully we have more opportunities, but still have some challenges, and we can finish with the multiple cooperation from the basic to the clinical thing. And 2 advocacy slides. So several weeks after, July 3 and 4, we will have a Korean Cancer Association meeting with the Asian Oncology Society meeting. And this upcoming November, we will have AACR KC joint conference in Busan. Hope to see you there. Thank you so much.
Thank you, Professor Na. We will discuss after 3 presentations. Second presentation is presented by Dr. Kim. Dr. Kim is a professor at Seoul National University, and he is also famous medical oncologist. He was also president of Seoul National University Cancer Hospital, and also he was president of Cancer Genome Organization and Korean Society of Medical Oncology and the Korean Cancer Association. And he is also CEO of IMDx, the cell-free DNA screening company. Dr. Kim, the title is Future Perspectives in Cancer Research with Novel Therapeutics by Dr. Na, and current status and future perspective in liquid biopsy for cancer. Dr. Kim.
Thank you, Professor Yang, for kind introduction. So the liquid biopsy is initially developed as an alternative to tissue biopsy, but with the advancement of the detection technology, it is one of the promising precision medicine technology. So today I'm going to take a look at some of the developmental status of liquid biopsy from this perspective. So, as we know, the cancer cells exhibit the complex genomic characteristics such as the redundant signal, long tail mutation, clonal evolution and heterogeneity, and a variety of the technology has been developed to address this issue, and the liquid biopsy stands out as the representative, the precision medicine, the technology in cancer. So compared to tissue biopsy, it has three advantages: minimally invasive, and also it can provide the comprehensive profiling regardless of tumor location. And also it can detect the cancer earlier than tissue biopsy. So regarding the early detection, actually this is driven by the technological innovation of the ctDNA analysis. So in 10 ml of blood, there are around 10,000 DNA molecules, and a small portion of this molecule actually is derived from the cancer cells, so that is circulating tumor DNA. And this fraction is mainly depends on the tumor size. So the ctDNA fraction will be 1% for 5 cm tumor, and 0.01% for 1 cm tumor. And also with the development of this, the NGS technology, now we can track the below 0.001% DNA to detect the tumor. So now it is applied to these three areas: genomic profiling in advanced stage, and the detection of minimal residual disease, MRD, after surgery, and multi-cancer early detection, MSET, in healthy people. So I'm going to briefly take a look at each topic. So this slide is a little bit a busy slide, but showing that the commercialized in the NGS cancer panel for genomic profiling in advanced cancer. So blue is from the product from the global company, and the right side is the product from our team. So the number of target genes are somewhat different within the product, but you can see the druggable genes, number of druggable genes are quite similar. It's between 25 and the less than around 50. And also the sensitivity and the limit of detection is quite similar between the products. So the performance of the cancer panel is first assessed by tissue, the concordance. So previous study has shown that this concordance is 80 and 90%. And in case of this concordant case, for example, the tissue negative, the plasma, ctDNA positive, you should consider the three things: the heterogeneity, the emergence of resistance mutation, and the presence of the clonal hematopoiesis. CHIP is a somatic mutation of the hematopoietic cells. And recent report from the NCI match trial also showed the similar, the concordance rate is 81.1%, and also they showed that the concordance between the metastatic site and plasma is higher than concordance between the primary tumor site and plasma, so indicating that the importance of the time interval and the heterogeneity in analyzing this ctDNA data. We also conduct the simultaneous study as a tissue and plasma sampling in colorectal cancer. So we got the tumor tissue and the blood at the same time during surgery. And in this case, the concordance was way higher. It's 96%. So indicating there is no time interval between the tissue and the plasma, so suggesting the very high concordance rate. And this is just one example of the ctDNA analysis in lung cancer. That's the real-world data, and we found the mutation in 60%. The overall concordance is 85%. And in advanced stage, the driver mutations, the fraction of the driver mutation is usually the over 1%. So this is the data from the Foundation 1, the analysis, and they report that the 26% case showed less than 1% the ctDNA fraction. In our analysis, it's similar. So the 33%, so less than 1%, and 26% is less than 0.5%. So detecting the low BAP, the variant, is very important to avoid the missing very important druggable genes. So the detection of low BAP, the variant is important. And the other advantage of liquid biopsy is dynamic monitoring during the treatment. So during the treatment, there may be the subclinical molecular resistance before emergence of this clinical progression. So recently published this SERENA-6 trial in breast cancer. They screened the ESR1, the ctDNA mutation in every 2 to 3 months, and they found that the patient with the ctDNA ESR mutation without the clinical progression, when this patient was treated with the camizestrant, it showed the prolonged survival. So indicating that the ctDNA guide dynamic monitoring is very effective in selecting the molecular progression subset. And also this liquid biopsy can be tested in the other body fluid besides blood. So this is one example of the pleural effusion study, and we found that the driver mutation in pleural effusion is more commonly found than the tissue and the plasma sample. And this patient also showed a good response. So indicating that the ctDNA, the application can be extended beyond the blood. So now we have many experience to test this liquid biopsy in CSF and the urine. So looks we looked the very good positive data of this liquid biopsy. So next is the MRD. So MRD is a major cause of the relapse after surgery, but it cannot detected by conventional imaging, but with the technological, innovation of NGS sequencing, we can now track the emergence of the ctDNA, the MRD, by tracking this ctDNA. So there are two assays of MRD assays, so tissue-informed and tissue-agnostic. So tissue-informed requires tumor sequencing first, and then select the variant, and then make the personalized panel for the follow-up. Tissue agnostic does not use tissue. This method only uses the blood and also test the gene panel or methylation or fragment. So there are pros and cons of each method, but in terms of sensitivity, this red bar is a tissue-informed method. So the sensitivity of tissue-informed method is around 10 times higher than the agnostic method. So currently, the tissue-informed method is the standard method for the MRD analysis. So this illustration is from the Signatera the product. This is the globally leading the MRD assay, and they did the whole exome sequencing first and then select 16 genes. So we don't know why they only select 60 genes, but they do the PCR, so maybe the PCR may be the main reason for the selecting only 16 genes from the whole exome sequencing, and then follow up the emergence of MRD, and they show that the MRD positive, post-operative MRD positivity is very significantly related to the poor survival across the different type of tumors. The detection sensitivity of the Signatera is around 0.01%. So, because as I mentioned, the Signatera used only 16 genes for the PCR, so we hypothesized that if we increase the number of variants, like 100, 150, this the ctDNA detection rate also will be increased. So, based on this hypothesis, we developed the tissue-informed hybrid capture-based personalized panel for MRD detection in colon cancer. So we did the whole exome sequencing and select the variant and make this panel. And this panel can maximally include 500 genes compared to 60 genes in Signatera. And we tested this cancer detect assay in resected colon cancer and found that MRD positive at post-op 3 weeks is associated with poor survival and also persistent MRD positivity after adjuvant therapy is eventually the relapse. So looks very work very well, and based on this data, actually this test is recently designated as a breakthrough medical technology in last March by the government. And another indication of MRD is the monitoring of the immunotherapy, because ctDNA MRD test can track the very tiny amount of DNA, and many immunotherapy is currently on the test as adjuvant therapy. So in this trial, in this bladder cancer trial, post-operative ctDNA negative patient doesn't show any difference between the immunotherapy and the control arm. However, in ctDNA positive patient, so MRD positive patient, the immunotherapy arm showed a better survival. So indicating that this ctDNA MRD test can guide the adjuvant immunotherapy in bladder cancer, and this the theory is now applied to the other type of cancer. And also we can also track the neoantigen after cell therapy in this the phase 1 trial after cell therapy. In case of the relapse, this neoantigen was increased, while in case of no relapse, there is no increment. So now immunotherapy is now at the center stage of cancer therapy. I think the ctDNA-based MRD test will be a very useful strategy to monitor the response to immunotherapy. And as I mentioned, the detection sensitivity of Signatera is 0.01, and our method is 0.001%. And we can imagine if we increase this detection sensitivity, the ability to detect MRD also will be accordingly increased. So many new technology actually have now is developed to improve the sensitivity. So this now is MRD assay field is rapidly evolving into developing more sensitive method. Last part is MSET, multi-cancer early detection in healthy person. So compared to conventional screening, this liquid biopsy offers several advantages: non-invasive, multi-cancer detection with just one simple blood draw, and also provide the tumor of origin. And also this test is very flexible and very good accessibility, so many advantages. And this is just the process of the ctDNA, the MSET test. So usually do NGS sequencing and generate multiple features, including methylation, mutation, fragment, copy number, etc. And then this data is analyzed by using the machine learning and develop the algorithm for detection of a cancer signal and to predicting the tumor of origin. So most of the MSET tests are using this process. So this table is currently the globally developed the MSET test is most product use DNA methylation, because DNA methylation is very useful to identify tumor of origin. compared to other methods such as fragment and the copy number. And this Gallery product is globally leading the MSET test, and they can screen the more than 30 tumors, and they use DNA methylation, targeted DNA methylation, to target 150,000 the methylation site. And this is the first case control study using the MSET Gallery test, and they screened more than 6,000 people. And the test performance is specificity is very high, but the sensitivity is 44. And if you see the stage, the sensitivity is very low in early stages. So very disappointing the result. But please note that the specificity of this test is very high. So we also developed the methylation-based MSET test, the cancer find. So in contrast to the Gallery, we perform the whole genome methylation sequencing first and generate the 3 feature: methylation, copy number, and then fragment, and then the ensemble analysis by using the AI, machine learning, and then detect the and then develop the algorithm for the cancer signal and TO. And 1,000 around 500 patients are enrolled in our case control study. And our specificity of our test is a little bit lower than Gallery, so 97.3%, and then the sensitivity is 89%. So looks the better than the Gallery test in this 8 cancer type. This is a sensitivity according to stage, and overall it showed good the sensitivity even in early stage. But please note that the specificity of this test is a little bit lower than the Gallery test, and the TO prediction was also possible in 85%. And there are 2 important prospective MSET trials, which is a Pathfinder study and Detect-A study. I'm going to explain this Pathfinder study. And so they use the Gallery test over the 6,000 patients, and then the test positive rate was 1.38%, and then the PPV is 41. So final cancer confirmed rate is 0.57%, and this is fairly very consistent with the average annual cancer incidence in general population, which is 0.3 and 0.6%. So reflecting real world situation. We also tested the performance of our cancer find in the real world setting. So far, the 460 tests have been conducted for healthy person. Test positivity was 2.2. The 2 cancer was found. It's 0.43%. So quite a little bit similar to the Gallery test. This is one example of our cancer find test, and we analyzed the methylation, copy number, and fragment. So methylation score was very high, and there was the copy number change, and there were short the fragment distribution. So overall score was very high, and this patient was found to have the colon cancer. So although the MSET test is promising, still there are many unanswered questions, many challenging issues. So what is the target performance of this test? Who should get this test? How to follow the positive test without any clinical evidence of cancer? That is an issue. And also how this MSET test can be positioned in conventional cancer screening. So these are the very important question. And to answer this question, there are 2 ongoing randomized trial of the NHS Gallery test, NCI Vanguard study for total 160,000 people to compare the MSET test versus the cancer screening. So I think the data will be available and 2-3 years later. So will give us a good answer for the many unanswered questions. And we are also involved in this 2024 K-Health Mire initiative project, which is ARPA-H project, and the main goal of this project is to develop low-cost, high-performance MSAT test to screen 30 cancer types. So we are going to screen more than the 3,000 patients for 30 cancer type to develop this technology. So this is my last slide. As I mentioned, the ctDNA liquid biopsy holds the significant promise in the and precision oncology in the profiling, monitoring, MRD detection, and the screening. But still, the liquid biopsy is clinical adoption rate is less than the 20%, I think. So to facilitate the clinical integration, there is a pressing need for more active clinical trial, the clinical validation, and also regulatory framework, the development. So that's the very important thing. So what we need to do. So I'd like to thank my team and at SNUH and IMBDX. Thank you.
Thank you, Professor Kim. As I mentioned, the discussion after one more presentation.
This speaker is Bang Eun. She's a gastric cancer surgeon. He graduated Seoul National University and then joined the Center for Gastric Cancer, National Cancer Center as a staff surgeon in 2012. She published many articles, include functional preserving gastrectomy. Dr. please.
Thank you, Chairman. It's really an honor to be here. And my ladies and gentlemen, today's my topic is the function preserving gastrectomy. Actually, in gastric cancer surgery, function means the function of pyloric rings. This pyloric ring is the sphincter muscles, like the anus. So this muscle have the contraction and relaxation, and to control the food passage from the stomach to duodenum. If there is no pyloric ring, the food can pass through the duodenum so fast or too much. So in conventional gastrectomy, this pyloric ring was sacrificed. So there are key clinical trials for function-preserving surgery to save this pyloric ring. At first, the pylorus-preserving gastrectomy can be performed instead of distal gastrectomy for the early gastric cancer in middle location. And the second is proximal gastrectomy can be performed instead of total gastrectomy for upper part, the tumor. So the key trial is the class 04 and class 05. The class is the Korean laparoscopic study group for this trial, and this group compared the pylorus preserving gastrectomy with the distal gastrectomy, and in this trial, the proximal gastrectomy was compared to total gastrectomy. The class 04 was the PI is Hyuk-jun Lee from Seoul National University. The hypothesis was the pylorus preserving gastrectomy can reduce the dumping syndrome because the pyloric ring is preserved. But unfortunately, the dumping score was no significant difference between the two groups, and also no difference in quality of life. And only the PPG have higher reflux symptoms. So it was unfortunately it was failed in aspect of the primary endpoint. How about the class 04 and 05? The hypothesis was the proximal gastrectomy, specifically double-track method, can improve the hemoglobin level and the vitamin B12 level, but unfortunately, the hemoglobin level have no difference between the two groups, and only the proximal gastrectomy group have the less the vitamin B12 supplementation. So today, I'd like introduce more dramatical the function preserving surgery like this local resection, wedge resection. Actually, this local resection is for the benign tumor, not for malignant tumor, because For this local resection, we should consider the lymph node metastasis around the gastric cancer. As you see, there are lymphatic channel is so complicated and it is difficult to expect where the tumor cell is go. So the sentinel navigation surgery, sentinel lymph node evaluation is the first step and it is mandatory and important. So this figure shows the sentinel node. The sentinel node is the closest lymph node around the tumor. So when we before we dissection of tumor, we evaluate this sentinel lymph node, and if there was no tumor cell, if there is tumor cell in sentinel basin, we can perform the subtoral or toral gastrectomy, which is standard gastrectomy. If there is no tumor in sentinel basin, we can preserve the stomach. And our goal is this preserving surgery. The proportion of this sentinel positive is only 10 to 15%, and the majority of patients can receive the wedge resection which was more than 80%. So this is my content. I will briefly review the previous study and I will introduce this sentinel navigation surgery trial, Senorita 1, 2, 3, 4. Actually, the researches, there are many researches for sentinel navigation surgery. This is the list in textbook. And this sentinel navigation surgery was evaluated since 2001 for clinical T1 and 2 tumor. Initially, the single trace was used by some surgeons, but as the surgeons' experience accumulated, the dual trace was standard, including the isotope, technetium, and the ICG as the dye. Endoscopic submucosal injection was usually performed. So overall, the detection rate was more than 90%, and the sensitivity have the wide range from the 50 to 100%. So the sensitivity was somewhat unsatisfied. So the confirmative trial was needed. This is the trial of the Senorita trial. The PI was Gunonyu, my senior doctor who worked in National Cancer Center. This is the prospective randomized control trial, and 7 tertiary or general hospital was involved in the Republic of Korea, and patient diagnosed as gastric adenocarcinoma and less than 3 cm was enrolled this trial. And in this trial, the sentinel navigation surgery was compared to standard gastrectomy, and primary outcome was 3-year disease-free survival. And secondary outcome was overall survival and quality of life and nutritional outcome. Actually, quality of life and nutritional outcome is functional outcome. And this study is non-inferiority design, and the hypothesis was the 3-year disease-free survival was 97% in standard gastrectomy, and we accept the non-inferior margin of 5%. This picture shows the procedure of sentinel navigation surgery. The first step is endoscopic trace injection around the tumor in operating room, and the dual tracer was used, including technetium and the ICG. After 15 minutes, the sentinel basin was detected. Actually, this green area is you can see because we use ICG green dye, and also we detect this using this probe for the isotope. And after we decide the sentinel basin can be dissected like here and extracted. The harvest lymph node basin can be identified by the surgeon in the back table in the operating room, and all single the lymph nodes were classified like here. And we wait the frozen examination, and if there was no tumor, no metastasis in the sentinel basin, the primary tumor can be dissected by this the stapling or the energy device. So then we close. Primary outcome was oncological result, which was the 3-year disease-free survival, and the upper blue line is standard gastrectomy, which was 95%, but the sentinel navigation surgery was 91%. Unfortunately, we cannot prove this non-inferiority because the absolute difference was 3.7%, but the 95% confidence interval was over the 5%. So it was failed, but please focus on the disease-specific survivor and overall survivor. The two lines, blue and red, is almost identical. Let's have the what makes the difference in the disease-free survivor. Actually, the lymph node recurrence was occurred in one patient among the 258. So false negative rate was almost, we considered 0.4%. The major reason is metachronous cancer because the sentinel navigation surgery preserved the almost stomach volume, so metachronous cancer can occur like ESD, but there was no event in the standard gastrectomy. How about functional outcome? As you expected, the quality of life, in respect of quality of life, the sentinel navigation surgery showed better physical function and less pain, reflux, eating restriction, anxiety, and taste change. As you see, the sentinel navigation surgery have a blue line and it's lower. Lower means the less symptom. And how about the nutritional outcome? As you see, the body BMI and hemoglobin and protein level was higher in sentinel navigation surgery. We said preoperative BMI was 100%. It was decreased, but it gradually increased. And at postoperative 3 year, it was almost reached to baseline 100%. But standard gastrectomy, it was stay in the 90%. So the sentinel navigation surgery is mostly benefit in the nutritional outcome. And hemoglobin and protein level have the similar pattern. And based on this outcome, the Korean practice guideline include the sentinel navigation surgery for early gastric cancer. Of course, it was the conditional recommendation. But the big limitation of this sentinel navigation surgery to generalize is the dual tracer, which include the isotope. If we want to use this isotope in trial, they are tough because all involved people get the special health checkup and safety education, and the pathologists, the nuclear medicine, and the nurses are very reluctant to this trial. So we started Serenita 4. The PI is Hongman Yoon, also my colleague, and we wonder the dual tracer can be replaced with ICG only by the fluorescence image. So this is the fluorescence image in the operating room. As you can see, this is a sentinel basin, and you can see the tumor marking and around tumor here. And we dissect the sentinel basin like here, and the wedge resection can be performed. In this trial, more than 4 hospitals was involved now, and this trial was started 2 years ago, and the target number is about 200, and currently enrollment proportion was 40%. I'd like to introduce the Serenita 2. This is for the additional surgery after non-curative ESD. According to treatment guideline, after ESD, some tumor is out of indication for ESD according to the tumor size or differentiation or the ulceration. For this patient, the additional standard gastrectomy is needed because this tumor is out of indication of ESD. But after additional surgery, we frequently face this pathological report. There was no residual tumor and no lymph node. Actually, frankly speaking, these patients have overtreated. And additional gastrectomy is not needed, but we didn't know this patient have lymph node metastasis or not. According to previous study, the probability of lymph node metastasis was around 10%. If this sentinel navigation surgery can be applied to these patients, and we can save the stomach. So if ESD margin was pre, we don't need more resection, and if ESD margin positive, the wedge resection will be enough. But we did this trial because we wonder about the lymphatic flow. After ESD, the fibrosis will be occurred and it can block the lymphatic flow. So feasibility study was needed. So this picture shows the procedure of this feasibility study. The first step is same with one tracer injection around the ESD ulcer, and the two methods were accepted in this trial, the dual tracer or ICG only and fluorescence image, and then we dissected the sentinel basin, and finally standard gastrectomy was performed. The endpoint was detection rate, and the hypothesis of detection rate was 95%. It is based on the previous study, and secondary endpoint was sensitivity. This is the result of Senilta 2. The enrollment was finished last year, and we enrolled 243 patients. There was some technical problem or multiple reason. We excluded 5 patients, and we have also 7 patients of detection failure. So the detection rate was 97%. It was somewhat high, and the sensitivity was just 69.6%. What do you think of this sensitivity? It looks somewhat lower than our expectations. So the conclusion of Senilta 2 is sentinel Bayesian mapping after non-curative ESD achieved sufficient detection rate of 97%. However, high false negative rate of 30% seems unacceptable regarding the oncological safety. Finally, I will briefly introduce Senilta 3. This is non-exposure simple suturing EFTR. The upper part is intralumen of the stomach, so tumor is here. And if the surgeon do the suturing or out of the tumor can be protruded in the intralumen, and then the ESD can be endoscopy full thickness can be performed, and the edge was closed with clip and snare. Actually, the PI is Chang-Kyu Kim, the chairperson today, and this study is prospective single group, phase 2, and the 2 hospital was involved, and the indication was same with Senilta 1, and the hypothesis was complete resection rate of 90%, and the 88 patients were needed for this trial. And this is brief picture for NAS EFTR. After sentinel navigation evaluation, if there was no tumor in sentinel node, the endoscopic marking and serological marking and simple suturing, and then tumor was protruded in the intralumen, and then the edge was the clip closure with snaring and clipping. This is the result. The hypothesis was complete resection rate of 90%, and there was 13% of lymph node metastasis, and these patients go to the standard gastrectomy, and among the NAS EFTR 71 patients, the 60 patients was successfully performed. The primary endpoint was 92% of complete resection, and the secondary endpoint, the successful NAS EFTR was 85%, and there was 11% of intraoperative perforation, and there was no postoperative complication. So let me summarize. The laparoscopic sentinel navigation surgery as the function-preserving surgery is recommended as treatment option for early gastric cancer less than 3 cm in Korea. Currently, we we evaluate the ICG only with the fluorescence image in clinical 4, Sentinel 4. The sensitivity of sentinel Bayesian evaluation was approximately 70%. After ESD, it may be acceptable for safety. And less EFTR with sentinel Bayesian dissection can be safe and feasible. But there was still, we have homework to be solved, method standardization, and cooperation with other departments, and sufficient reimbursement reward. Most important thing is surgeon passion and effort. Thank you for your concentration. Thank you.
Thank you for excellent lecture. Now we'll start the discourse session. I'll introduce 3 discussants. First is Professor Hyundong Kim. He is a medical oncologist and he obtained PhD in KAIST and currently assistant professor in Assam Medical Center. Another content is Professor Shinhae Park. She is a surgeon. She is currently work as a clinical assistant professor in Catholic University of Korea. Last discussant is Won-young Choi. He is a medical oncologist and obtained PhD in KAIST and joined in National Cancer Center since 2019. Is there any questions in discussion?
Thank you for the introduction. I have a question for Professor Ra. First of all, thank you for the nice lecture. And as compared to the previous failures with many clinical trials, what do you think is the main reason for the recent success of targeted or immunotherapeutic trials in gastric cancer?
So for the Claudin and the FGF is waiting, but for the Claudin is very unique and specific to the gastric cancer. And also, as I mentioned, that it's a combination of cytotoxic and the targeted agent. It's not just monotherapy. So, and also that's the same for the immunotherapy. I think still there are some proportion of cells are needed to be treated with cytotoxic, and adding targeted and the immunotherapy might be the success reason.
So with more and more emerging biomarkers, what do you think would be the best strategy to incorporate many biomarkers at the same time for the future design of clinical trials?
Right. So the problem here is co-expressing cases. And among them, we should differentiate what is more oncogenic dependency. So for example, if there is MSI high versus other markers are there, we know that in that case, immunotherapy may be the highest priority. But for the HER2, we know that the biology, so it's more easy to do. The problem here is the Claudin and the PD-L1 positivity. We don't know which one is good and what is a proper cutoff. So currently we have more and more molecular epidemiologic data to figure out who are the more important biomarker to select the treatment. And then the other issue is adding those drugs are better. So but the problem in that case, cytotoxic and to put targeted agents, immunotherapy, that's too much. So we are expecting more of multiple integrated drugs, such as bispecific is one of the agents we can expect a better outcome.
My final question is, gastric cancer is featured by substantial intratumor heterogeneity. So do you see any potential role of ctDNA to complement the intratumor heterogeneity for the design of future trials?
Sure, there is a role. The problem is how big enough or how practical. So gastric cancer with peritoneal metastasis still has a less sensitive detection in the ctDNA, and also some of the tumors does not have the proper target. So hopefully, as you heard from Professor Kim, we have a more sensitive method and want to see those activities in the gastric cancer. In that case, we can use it.
I would like to express my gratitude to all the speakers
for the latest knowledge on gastric cancer. I have two questions for Professor Sunyoung Ra. Thank you for introducing various biomarkers and new drugs. What percentage of our gastric cancer patient has FGFR expression, and which percentage of the patient can benefit from these target agents?
So for the FGFR, as I said, highly overexpressing is only 10%. So in the beginning, it was tested with amplification, which is lower, it's only 5%, but with overexpression with the IHC, that comes about 10%. So in the beginning, we are expecting about 30%, but not that high. It's about only 10%.
Yeah. Second one is immune checkpoint inhibitors and target agents have not been applied in adjuvant chemotherapy settings after gastrectomy. Will there be a change in the treatment policy regarding the combination of immune checkpoint inhibitor or target agents with cytotoxic drugs in adjuvant chemotherapy?
Right. So there are big debate about the upfront surgery and adjuvant versus perioperative, but you know that in this East Asia, it's better with the upfront surgery with chemotherapy, and on top of that, adding IO is not that great. But this ASCO, just 2 weeks ago, we have a perioperative therapy with FLOT and immune PD-L1 inhibitor, the Durvalumab, show very good outcome with the PCR increasing, and the event-free survival has been increased. So now it's one of the options for the perioperative setting. The question is, we can use it in the Korean cancer patients. I think it's not the all patients, probably some advanced cases and has a potential benefit with the IO might be a subgroup of the patients.
Thank you. And I have one question for Professor Kim. Thank you for your insightful presentation on ctDNA and liquid biopsy. I understand that gastric cancer releases less ctDNA into the blood than lung cancer and colon cancer. And the detection rate of ctDNA is also low in peritoneal metastasis in gastric cancer. And is MRD assay is also applicable for gastric cancer to determine the commission?
I think so. So, as you said, the shedding rate of gastric cancer is very low compared to the lung cancer and ctDNA cancer. But as I mentioned, the testing, the sensitivity is getting the higher. So we expect gastric cancer can be a target of ctDNA analysis in the future. I think the MRD test is extremely sensitive to detect the tiny tumor in our body. So actually, we are currently doing the episodes in the gastric cancer trial with two professors to see the MRD after and this, but interim analysis shows some positive data. So MRD positive has higher I think we will report the final result in 1 year, hopefully.
Thank you.
Thank you for the lectures. I have one question for Dr. Ra and one question for Dr. Kim. I'll ask my first question to Professor Kim. When we order ctDNA analysis, we sometimes encounter mutations that are classified as CHIP. And for well-known genes like DNMT3A, we know that it's a CHIP, but for some unfamiliar genes or maybe some genes that we think might be a driver, sometimes they're classified as CHIP. So how do you determine whether it's a CHIP or not when you analyze the mutations?
Yeah, actually we have developed our in-house filtering.
Professor Kim, please use your microphone, please. Don't look at him.
I want to see him. So we have developed our in-house the program to identify the chip from the ctDNA analysis. That algorithm, so there is
the chip gene list already, and we add some the bioinformatic process by using some the fragment analysis, because the chip genes doesn't show the fragment change. But if the ctDNA that's really derived from the cancer cell, it shows some of the fragment change. So we add some also the chip gene list and the plus and the fragment, and plus I think we use around the 50 the genomic features to differentiate between the real mutation and chip mutation, and this is a kind of the company. It's a business secret. We are not going to publish all in detail, but I think in the JCA and AACR cancer meeting will be coming early July, and we will publish the abstract about our chip analysis. I can share the abstract.
Thank you for the detailed answers. My next question to the Professor Ra. You've shown that some targets, the drugs have succeeded in clinical trials before. Targets like DKK1, the target, although biologically it has a meaningful result, the outcome was not so meaningful. So do you think it could be a problem? Is it a problem with the target itself, or do you think it could be a problem with the mode of the blockade? So would there be a potential to change the mode of blockade and pursue for other types of drugs for the same target, or do you think the target should be just ignored and move on to other druggable targets?
Both. So you know that's same for the HER2. So breast cancer has working a lot with the pertuzumab and the TDM1, but it doesn't work in the gastric cancer. But with a better ADC, such as TDXD, it works for the gastric cancer also. So target is there, but sometimes the target inhibition is not enough, but improve the drug is overcoming those problems. And also, some of the markers, like DKK is not active or the predominant as a PD-1, PD-L1 pathway, but still there are some roles. So if you can control them, there is an added effect. The problem is how big it is. So I think still all those targets are druggable and it's important, but we should think about how we can inhibit efficiently and costly. So I think we don't have to stop all those things. So that's the same for the TIGIT. TIGIT has been negative for all other tumors, but there is a potential because domenzyme and domozim, that is a different mechanism, even it's very similar. And we know that even with a PD-1 inhibitor, that the GSK drug is more active. So with increasing technology, we have a potential better drug. So I think we don't have to give up.
So we have the 7-minute limit. Why don't you ask to Dr. So especially Dr. Shin, you have to ask to Dr. the respect of the surgical approach.
Yeah. I have one simple question to the Professor Am. In Senorita 2 trial, there have some case in lymph node metastasis beyond the sentinel basin. In that case, there are more than perigastric lymph node or the suprapancreatic lymph node.
We have the 7 patients with out of the sentinel node. They are somewhat remote lymph node, not around the tumor, just close the. For example, the middle tumor, the tumor metastasis, lymph node metastasis was around the 6 or the 9, and it is case by case. So usually the false negative case have removed the lymph node metastasis.
Thank you. We have to do the standard gastrectomy.
So actually, I have one question. So, Doctor, you showed some endoscopic surgical resection, and there is a safety margin. So there is a residual tumor in the resection margin. You have the gastrectomy, and otherwise is watch and the observation. So what is the proper safety margin of the endoscopic resection?
We check the all 4 direction margin in intraoperative frozen section. So after in the frozen section it is positive, we further resection is performed and until the negative. So the resection margin is okay in sentient navigation surgery, but yeah.
So even though it's 0.1 centimeter is okay as the safety margin. I mentioned about what's the criteria of the safety margin in terms of the number of centimeters.
Yeah, there was no criteria, just 3 is okay, like the ESD.
How about the professor? What's your opinion?
Yes, safety margin is usually 0.2 centimeter. So one slide is okay, we think. But in case of 4 different customers, so much more it may require more distance, but usually it's okay.
So actually, I have one question for all the presenter and the discussant. So in colon cancer, MSI high tumor, some people say they know surgery because it's a very good response by the immune checkpoint inhibitor. How about in the gastric cancer and other tumors? The surgical role in MSI tumor.
So there are some of discussion about that. But when you see the response, depth of response in the colorectal cancer versus other tumors, colorectal cancer is really good. And but the gastric cancer with the monotherapy is only 70% or something. So that's I'm keeping saying that gastric cancer is a bit of different from other tumors. If we have a really good MSI the immunotherapy to suppress all those tumor clones, there is a chance to not to do the surgery. But until now, I think still some of the surgical control is important.
Yeah, to add to Professor Ra's comment, the PCR rate, pathology complete response rate, was around 60% with dual checkpoint blockade in MSI high gastric cancers. So I think it's still 40% behind. So there should be additional biomarkers like ctDNA to avoid surgery in that case.
Because of time limitation, we will close this session. Thank you.
Thank you for the great talk. That concludes session 3. We thank all the speakers, session chairs, and discussants.
이렇게 돼 있어서 앉아요.
Now let us start session 4. The title of this session is Advancing Cancer Survivorship Through Healthcare Research and Innovations. We are honored to have Professor Mi-sun Jeon from Aju University and Dr. Jong-hoon Kim from National Cancer Center Korea as the session chairs. Please give them a round of applause.
Good afternoon. The last session, session 4, is on the survivorship care. Its title is Advancing Cancer Survivorship Through Healthcare Research and Innovation. I'd like to introduce the first speaker, and the chairperson, Mi-sun Jeon, will introduce the next two speakers. And we have two designated discussants after the three presentations. So I'd like to introduce Dr. Yutaka Matsuoka. Yutaka Matsuoka is now he is the director of National Cancer Center, Japan, Institute for Cancer Control, NCC-ICC. It's only last April when he got the position. It's a very important position in Japan for cancer control. He graduated from the GK University and got a PhD from Hiroshima University. He was trained as an internist. Then he trained as a psychiatrist. So he was a research resident of Psycho-Oncology Division, National Cancer Center Research Institute East. And from 2021, he joined the Japanese government as a medical specialist. Then he returned to National Cancer Center and became the director of NCC-ICC. Today, he will talk on the challenges of cancer survivorship, R&D, and policy in Japan. So please, Dr. Matsuoka, and make a big applause. Thank you.
Thank you for kind introduction, Professor Kim. It's my very honor to be invited here to talk about our talk today. My talk today is challenges of cancer survivorship, policy, and research and development in Japan. I introduce first my National Cancer Center Japan. The NCC Japan consists of 6 organizations and the Institute for Cancer Control on the down right side, which I serve as a director, is responsible for social medicine and public health. And the guy is President Hiroyuki Mano. He is also in this April. He was a president, appointed president. Okay, this is a history. The Institute for Cancer Control has its origin in the Research Center for Cancer Prevention and Screening, and renamed the Center for Public Health Sciences in 2016, which was established in 2004 with a mission of researching and developing effective cancer prevention and screening methods. And Center for Cancer Control and Information Services, which was established in 2006 in response to the Action Plan for Promotion for Cancer Control 2005. Then the Institute for Cancer Control was established in September 2021. as a specialized institution to lead public health and social medicine research and cancer control project, national project. This is a graph showing the trend in cause-specific mortality rate in Japan from the mid-20th century to 2021. In 1981, cancer replaced cerebrovascular diseases as a leading cause of death, and their number have continued to increasing steadily to the present day. The exact number of cancer survivors in Japan is not evaluated, unfortunately, but in 2020, 945,000 new cases of cancer were diagnosed, and 5-year relative survival rate for individuals diagnosed with cancer in Japan between 2009 and 2011 was 64% for both men and women. This is a fourth basic plan, Japanese government for basic plan to promote cancer control program in Japan. This outlines a comprehensive approach to cancer measures. The overall objective is this one. Overcome cancer with all, cancer control leaving no one behind. Based on 1, 2, 3 key pillars: cancer prevention and fulfilling cancer treatment, and living with cancer. Plan defines the current status and challenges and necessary measures within each pillars. In the effort to living with cancer, the right side one, 4 key challenges have been identified. First one is consultation service, and second, information provisions, and third, cancer control or patient support such as employment, appearance care, and suicide prevention. And fourth, accommodating major life events. In addition to 3 key pillars, the plan also emphasizes the development of infrastructure to support them. Just Circle 4 is. In Japan, cancer treatment collaboration hub hospitals systems ensure that people can access standardized cancer treatment regardless of their place of residence. As of April 2025, a total of 463 hospitals have been designated by the Ministry of Health, Labor and Welfare. In addition to standardized cancer treatment, we have established multidisciplinary conference about 10 to 12 times per year, and coordination of care in the community and the cancer registry system. Designated cancer criteria for cancer treatment collaboration hub hospital require that they treat at least 20% of patients in the respective region or meet all of the following conditions, such as new cancer cases more than 500, and surgery more than 400, and chemotherapy more than 1,000, and radiotherapy more than 200, and palliative care consultation more than 50 per year. Institute for Cancer Control, our institute play a key role in advancing Japan's national cancer control policy by conducting both independent initiative, as indicated on the left side, and project commissioned by the government, Ministry of Health, Labor and Welfare, as indicated on the right side. I will now provide explanations regarding the highlighting the red text one. In the left side, Cancer Information Service and Cancer Information Service Support Center. And right side, development of regional palliative care and other network system and survey on the end-of-life care of cancer patients. First one, to ensure that accurate cancer information reaches the public, we operate a website called Cancer Information Services. This is a top page. This platform records approximately 3 million page views per month. And in our regularly conducted survey annually, 90% of visitors have reported being satisfied with the information provided on the Cancer Information Service website. Regarding cancer consultation support, our Cancer Control Research Institute provides nationwide service, and annually there are approximately 400 chat consultations and 3,600 telephone consultations. Each cancer treatment collaboration hub hospital across Japan is equipped with a cancer consultation support center where in-person cancer consultations are conducted, face-to-face consultation service. To ensure that the cancer patients receive medical care in the familiar communities, we provide training for regional palliative care coordinators. So far, now we have 1,400 individuals have completed the training, and they are actively supporting patients nationwide in Japan. This one is just survey's result. While advancing their cancer control measures, we are simultaneously conducting evaluations of their effectiveness. However, our cancer control journey is still ongoing, and there is much more to achieve. Here is a summary of the survey result aimed at revealing cancer patients' subjective evaluation of their cancer treatment experience. The survey targeted 32,000 randomly selected individuals from cancer treatment collaboration hub hospital, and response were received from 15,000 participants. And results shows 65% responded that they could consult medical staff immediately in a hospital when experiencing physical distress. And as a result, said 48% responded that they could consult medical staff immediately when experiencing psychological distress. So it's not enough now. This is as a result. There were results of nationwide survey conducted to understand the realities of end-of-life care and living conditions among the bereaved families of patients who passed away in 2021. We have assessed many measures, but this is a very topics point. During the period, the study period, the COVID-19 pandemic led to visitor restrictions and movement limitations and increase in home-based care, causing significant changes in the healthcare delivery system in Japan, and as well as all over the world. And the proportion of cancer patients who experiencing minimal physical suffering was 37%, marking a 5-point decline from the previous survey in 2019 to 20, 42%. This finding also indicates that this proportion was relatively lower compared to patients who passed away from natural aging. This group shows 53%. Patients with cancer experiencing psychological distress throughout diagnosis and treatment and mental comorbidities are very common. We examined data from the National Cancer Registry, which includes the entire population in Japan, and evaluated 100 million patients with cancer. The standard mortality rate at 0 to 1 month, this graph, 0 to 1 month was 4.40, and at 13 to 24 months was 1.31 for suicide. These risks increased with shorter duration, a shorter follow-up period after cancer diagnosis. Suicide prevention countermeasures for such patients are warranted. So, optimal cancer survivorship care may need its comprehensive guideline for all clinicians. Then, we previously identified cancer survivorship guidelines in Japan. However, unfortunately, no specific cancer survivorship guidelines was available in Japanese. Then we have started to develop comprehensive Japanese guidelines that address these issues would help to improve the clinical outcome for cancer survivors. To realize a society for living with cancer, extensive research and development as well as implementation are necessary, such as support system in society, and guideline for care providers, and survivorship care for survivors. This slide shows a comprehensive 10-year strategy for cancer control provided by Ministry of Health, Labor and Welfare in Japan. It highlights importance of strengthening consultation service and supportive survivorship care. In section 3, research contributing living with cancer, 3-1 indicate ensuring accessible consultation support and information provision for all. And 3-2 indicate realization of well-supported cancer survivorship. The total amount of research grant provided by Ministry of Health, Labor and Welfare for studies related to living with cancer has increased and has tripled over the 5 years period 2020 to 2024. This slide shows the best 10 topics in the health and labor science research area, governmental fund. We focus the Gothic and red character text is important things. For example, rank 1, psychosocial support system, pediatric and IR system, And second one, information provision and consultation support network for rare cancers. And third one is network for rare cancer diagnosis and consultation. So such as a support system or network has been adopted government of Japan in 5 years, recent 5 years. This slide shows the funding amount allocated to research on living with cancer from the National Cancer Centers, Japan's intramural research budget. It remained at 68 million yen over time. It's stable. And this slide shows the key research of NCC Research Intramural Research Development Fund. And it also, its slide shows this slide indicated the importance of supportive care and improve the quality of life and cancer survivorship guidelines and solution for distress in adult and young age to middle age and cancer survivorship care implementations. So, this is just for example, Japan's cancer survivorship guidelines led by me. We organized a committee for cancer survivorship guidelines based on the US NCCN survivorship guidelines. Before that, we defined the technical term. For example, cancer survivorship is defined the entire experience of a patient after receiving a diagnosis of cancer, regardless of cancer result status. And cancer survivors is defined a patient become a cancer survivor upon receiving a diagnosis of cancer and remains a cancer survivor throughout lifetime. Cancer survivors also include family members, friends, caregivers of cancer patients because they are strongly affected by the survivorship of their patients themselves. And we discussed many times and so many frequently. And we finally decided 11 key clinical issues for cancer survivorship guideline in Japan. This is a result. And today we introduce physical activity guidelines. This is just published this year. And psychological distress guideline was published by the Japan Psycho-Oncology Society. And sleep disorder guideline is now under development. Maybe will be published this year or next year. This is the Japan's cancer survivorship guideline for exercise and physical activity. This slide shows question 1. We said the research question 1 and question 2. How many days per week do you engage in moderate or vigorous exercise? And question 2, how many minutes per day do you engage in this exercise? And clinicians calculate if the response to question 1 multiplied by the response to question 2 is less than 150 minutes, the individual shall be classified as not regular exerciser. So this equals this one, right side. And if the patient aged 18, between 18 to 64 years, and if aged over 65 years, this is a recommendation shows for younger patient, exercise should be suggested to cancer survivors aged 18 to 64 years who are not habit exercisers. And if the patient over 65 years, recommendation is same.
And after that, we developed the exercise program for cancer survivors. Especially breast cancer survivors at first.
Matsuka, 5 minutes.
Okay. And confirmed the efficacy of cardiorespiratory fitness and muscle strength. So this is the last slide. In my humble opinions, by maintaining stable research funding, we can move closer to the society where people live with healthy and dignity. Thank you.
Thank you. Yeah, I will introduce the second speaker, Dr. Joo-hee Cho. After she graduated from Hopkins School of Public Health, she moved to Korea, Samsung Comprehensive Cancer Center. She was a great researcher in psycho-oncology and supportive care in Korea. She will talk about esophageal cancer nutritional development. Dr. Cho, please.
Thank you. I know this is really hard time. Like 4 p.m. is a perfect time for sleeping. So I wanted to make sure all people get the attention. So it's my pleasure to be here. And I think I was talking to Dr. Matsuoka about this is the project. I've been doing this psycho-oncology project about 20 years. And this is studies, I would say, the most fruitful and having fun, and also practical study for the cancer survival I've been done. So a little bit of background, you might know esophagus cancer is 15 common cancer with a high mortality. So 2 facts you should remember for my presentation. So the average age of diagnosis is 65 plus 90% are male. So I know there is a male's audience here. I wonder how much proportion they cook at home. This is the question begin. So esophageal cancer in Korea is a little bit different than Western pattern. So they are more likely squamous cell carcinoma, and they're more likely to locate it to not near the stomach but upper part. So the major treatment for the esophageal cancer is actually surgery. So as you see here, technically you're actually taking out whole esophagus, then you move to the stomach here. So then they don't have a stomach, then they have many problems for eating. So they said, I feel tightness in my chest and whenever I eat. So over the time, the esophageal cancer, because this is like surgery, so you cannot really going back to the status before the surgery. So it's a permanent issue, and they keep experience fatigue and appetite loss, diarrhea, constipation, and dyspnea. So specifically, they were talking about eating difficulties and reflux and dry mouth is like really forever problem. According to the multi-center study, this is actually the largest data of esophagus cancer. 67 symptoms are related to surgery, and eating symptoms and fatigue and reduced activities persist more than 5 years. So this is like a vicious cycle. So you cannot eat, you get tired, you cannot move, then you cannot eat. So then this is like, then they lose weight. So esophagus cancer patients, on average, they lose more than 30% of their body weight after surgery. So persistent weight loss, not only is the problem of weight loss, you're not happy with the weight loss, actually it affects the survivor. And then we really recommend patients, you should eat, you should take lots of protein, and you should try to eat. But then they said, I have no problem with drinking, but I have problem with eating. So this is the problem. So I said, you have to remember 2 things, 65 years old male. So they said, so according to the study, low energy intake, Korean elderly is associated with malnutrition. So seniors living alone, especially, they lack out of food diversity. So this is typical, we actually take the photo when we visit them. So this is like a typical how they eat. Little bit of soup, and maybe egg, and this is pretty much. So they don't have really food diversity and they're malnutrition. So diet after esophagectomy, at least for the first few months after surgery, it required really high level skill and self-management. So they have a difficulty to eating regular meal. So recommendation is small and frequent meal. So we recommend 6 meals per day with a small amount. So you really like a chopped diet. And also because you cannot really eat the large and hard food, well, we recommend them small, dense, small portions. So like high protein, high nutrition. And also they have to actually do the physical activity because it requires the diet. So bowel movement, it's not really feasible for them. So we always tell them after you eat, don't lie down. So you have to walk and do the exercise. But exercise, we are not telling them exercise hardly. So exercise which help your digestion, you have to really consume your energy inside. So life management is also very, very important because it actually helps them to symptom management such as reflux and like dry mouth as well. So this is their problem. So then we decide how can we help this 65-year-old male who live alone? They cannot cook for 6 times a day, right? So this is how we think about it because we cannot. So I directing the Cancer Education Center for past 15 years, and I found out delivering naris is not solution. So it helped, but it couldn't really. It's really multi-layer. Although we try to really intervene personalized behavior, but there is always limit. So we try to figure out what are the environmental factor level we can change or we can do. So we took the precede proceed model, which is very typical behavior intervention model for the health behavior, which considers social, epidemiological, and behavior environmental factors to intervene somebody's behavior or policy. So even at the end, we are going to talking about the policy. So this then what we did is we are lucky to be part of a COCAS study and survivorship, and we were able to execute this study for past 3 years. So we are in the middle of the last year. So we conduct the qualitative interview and home visit and developing nutrition program and meal care plan for the last year, past 2 years. And we developed the food prototypes and actually we are develop dietary management services. Now we are evaluating the refined program and conduct the implemented study. So I'm going to tell you why this is comprehensive. To find out their unmet need and health behavior factors, we did first literature review and we interviewed a patient. So I think the most fun part for this study, we visit the home. So we not only visit the home, we actually go to their village, their living environment. So we go to grocery store with them. We see them how they buy, what they buy, and how they buy, how they cook, and how they save the meals and how they eat. So stay with almost a day with the patients and their family. It helps us to understand what is their behavior. So in terms of literature review, it's really what they are telling us is that we need diet improvement plan for the survivor. So they didn't take enough protein, they need more soft and moist food, and they try to they should avoid high fiber vegetable and tough meat. But, whenever cancer patients got cancer, they said, oh, we need organic. And they believe they has wrong, certainly, not the 100%, but for esophagus cancer patients, I would say it's the wrong perception about diet. So they should drink plenty of fluid, but they're not drinking enough. So, and they really need a supplement micronutrient. So we conduct an in-depth interview with 27 esophageal cancer survivors, which I'm going to share with a little bit of it. So they said we were trying to figure out what are the challenges in terms of meal preparation and eating and after eating. Because as I said, they have difficulty to eat. They also have difficulty to digest. then they got fatigue and they have physical problem. So in terms of meal preparation, at least they have a hard time to take minimum energy intake. So they usually take half of them, and they couldn't really find appropriate nutrition information. That this is what they say. Even we do hardly educate them. We educate them very much before they discharge. And they don't have, because it's a male patients, they don't have cooking experience. And within the eating, they always say they lost the appetite. And they want to eat slowly, but they cannot really do it. So then most of esophagus cancer patients are males or have smokers or drinkers, heavy drinker, then they have a difficult time to behavior control as well. So in terms of physical challenges, because they lost the appetite and they also experience symptoms that actually helping them not interesting eating. And reflux and dumping syndrome and bloating, constipation. So because they are saying, if I eat, I experience reflux and bloating, then I don't want to eat. Then I don't eat, I have a constipation and pain and fatigue. So then psychological challenges are even they try to eat, they don't have motivation to eat. They don't really feel pleasure to eat. So this study, we are saying, we want you to bring your joy for the eating. So that's what we try to do it. Then it actually connected to the anxiety and depression. They got lots of stress about eating. Then they're losing weight. It connected to fear of cancer recurrence. So in terms of environmental challenges, because these are the male, and even their caregivers, they have limited experience of preparing meals for the esophagus cancer patient, and they limited the choices of menu at the restaurant. So they hardly go out for eating because there is no menu for them. So social challenges, they reduce the social activities in eating out because they eat very slow. They eat very small amounts. So they said they avoid meeting friends or having dinner with them. Of course, this is summary table, so they definitely the information need and technical need and psychosocial need. Then we decide to develop the this home meal care food delivery services. So as I said, we also did home visit and dietary environmental assessment. So we visit the different area of Korea, like even Jeju Island. It was actually fun to go there. So we go to grocery market and we see how they cook. And we also see how they save the food and how they meal. So this is a little bit of a summary of it. I just to bring the summary of it. So they try to cook every time, the wives, because they believe this is healthy. So they don't want to recook. So technically they cook at least 3 times a day. And they spend a lot of time at the kitchen. So they prepare steamed meat. They try to eat home. And they said they rarely use delivery services. So a lot of time. So dining experience, they said typical meal duration is 30 minutes. And limited food choices for dining out. That's the reason why they don't go out. And they prepare to egg, tofu, and fish. They try not to eat the meat, but we really encouraging to eat the meat. So we developed the e-care intervention program, which is actually, we developed the meal. So we invented it. We work with actually a company called Well Story because we are not producing the meal. So we designed the product, nutrient based on the essential nutrient needed for the older cancer survivor. So if survivor requires like 2,000 calories per day, so let me show you how it looked like. So because these are the patients who just discharged from the esophagus cancer surgery, they cannot eat hard foods. The first 2 weeks, they can only eat very soft and liquid-based one. So if you're thinking about the porridge is really like, I would say it's low calorie. So we make sure like a small amount has high protein and calorie. So we develop the porridge for the patient, but also we develop the topping, like sprinkling series for the excluding sprinkles. But what they really want, they want it to taste what they used to taste. So they don't want something like patient food. So we also develop the food for the similar taste. We insist it's the same taste. The patients still say it's not the exact same, but anyway. So we make the bulgogi or galbijjim and several types of Korean food. So we also did a test with the patients and then try to make sure they also liked it. Then this is example of how we're doing it. So this is application. We monitor the patient, how they eat it, and then how much achievement they are doing it. So now we are at the stage of implementation study. So we have 3 different patient groups who are just discharged from the hospital, and 4 months and 6 months plus survivors, because they require different nutrition and they have a different condition. This is how we do before they discharge. So we ask them how to cook, how to prepare, because they have to experience the food we provide, then they can do at home. So these are actually what we provide for their home. So because we need to measure exactly how much they eat. So feedback from the patient, they really liked it. But these are the positive, but there are also negative feedback as well, because we don't have much choices. I mean, even we try to varieties, but it's very limited. So lessons learned from this project, we believe patients are really key. And what I learned past 2 years, it's really worth for us to visit there and see how they live and what are their environments. And we were able to do this study because we have a really multidisciplinary team, not only academia or industry. So this is actually first study we work with a real company industry. And I think my homework from this study is now its next step. So translating this clinical effectiveness into scalable and industrialization solution, because I believe maybe we can develop the voucher for these survivors or patients who really need these type of services. According to the statistics, 1/3 of Korean populations are living alone and single housing. So we have to consider this not only options for the esophagus cancer patient, but probably all the options for any seniors who need nutrition requirement. So we make sure bring back the joy of eating to our cancer patient. Thank you very much.
Thank you. It's a very nice talk. We will have more discussion at the end. I will introduce the 3rd speaker, Dr. Ji-hyun Kim. She is a specialist in gynecology oncology. She presently is a clinical assistant professor at National Cancer Center. And she published a few articles on treatment of ovarian cancer. She is interested in quality of life. Yes, Dr. Kim, please give us talk.
Okay, thank you for the introduction. Today, I will present on the development of a quality of life improvement program among ovarian cancer survivors. In this presentation, I will not limit our discussion to ovarian cancer alone, but first address the common quality of life deterioration issues experienced by gynecologic cancer survivors in general. I will then share our current healthcare development research progress based on the studies we have conducted. The number of the gynecologic cancer survivors in Korea continues to increase. As of 2023, there are approximately 64,000 cervical cancer survivors and 34,000 endometrial cancer survivors and 26,000 ovarian cancer survivors, and nearly double the numbers from the early 2000s. Endometrial cancer in particular shows rapidly increasing incidence rates and maintains a very high 5-year survival rate of 89%, suggesting that long-term survivors will continue to increase. While ovarian cancer incidence is also on the rise, the relatively poor prognosis of advanced disease presents some limitations in overall survival rates improvement. At our center, we have implemented quality-assured decision-making strategies that reflect individual patients' characteristics in primary ovarian cancer treatment. As a result, our treatment outcomes have improved to levels exceeding international clinical trial results. According to the data presented at ASCO this year, analysis of 1,256 advanced ovarian cancer patients from 2016 to 2023 showed a median overall survival of 92.8 months and a 5-year survival rate of 65%. With these improved treatment outcomes and the increasing number of long-term survivals, quality of life management for survivors has become an increasingly important challenge. Let us now examine what specific issues impair the quality of life for these survivors. Gynecologic cancer intergroup has identified essential symptoms that must be monitored for long-term survivors management. These include fatigue, peripheral neuropathy, cognitive impairment, chronic pain, sexual dysfunction, menopausal symptoms, depression, and the risk of secondary cancer development. In survival surveys conducted by GCIG, approximately half of the patients reported still experiencing these symptoms, and many perceive themselves not as cured patients but as cancer patients. Among these various issues, the lymphedema is particularly noteworthy. The lymphedema is not uncommon in gynecologic cancer survivals and is frequently encountered in clinical practice. And when we evaluate the patients using the surveys, the GCLQ, we found much higher prevalence rates in Korea, 67% in endometrial cancer patients and 55% in cervical cancer patients and 37.5% in ovarian cancer patients. While these differences may stem from various factors including race, BMI, the treatment accessibility, the important point is that lymphedema occurs at a very frequency that cannot be overlooked in gynecologic cancers. We developed a Korean version of questionnaires and confirmed very high validity. And this survey's instrument can serve as a useful tool for early assessment and follow-up management of lymphedema in Korean clinical environment, as demonstrated in the previously mentioned study. And in a prospective observation study using these instruments, patients with lymphedema showed lower quality of life scores and a greater economic burden. According to the QLQC 13, lymphedema patients scored an average of 62 points for quality of life compared to 71 points in the control group. The degree of financial difficulty reported as also more than twice as high in lymphedema patients, with an average score of 16 points compared to control group. Lymphedema thus extends beyond simple physical discomfort to wide-ranging effects, including emotional and economic burden. And the second issue is sexual dysfunction. We developed and validated the reliability of the Korean version of Female Sexual Function Index. The results showed sensitivity of 89%, specificity of 96%, with a high AUC, demonstrating high diagnostic performance. And in another study, we compared the sexual function between ovarian cancer survivors and healthy women. While the women overall sexual function score difference was not large, the vaginal dryness was more severe in the survivor group, and social function scores tended to be lower in survivors. And one more thing, the important change we have noted in the survivor quality of life management is that since the introduction of PARP inhibitors in 2021, an increasing number of ovarian cancer patients receive maintenance therapy for up to 3 years, while no evidence of disease status. So quality of life management during this period plays a crucial role in treatment continuation. According to our study, 24% of 131 respondents were unable to complete the full course of PARP inhibitor therapy. Higher compliance was observed in patients with better quality of life and lower symptom burden, as well as those who were unemployed or retired. The important finding is that creating an environment where the patients can actually tolerate the treatment beyond just the drug efficacy impacts long-term treatment success. And we also conducted a prospective study analyzing the temporal patterns of nausea and vomiting during the PARP inhibitor treatment. Interestingly, the symptoms peaked at approximately 3 hours and 11 hours after dosing, then gradually decreased over 12 weeks. The results demonstrated that providing intensive supportive care targeted to the predictable timeframes can simultaneously improve both compliance and quality of life. We are currently conducting a customized healthcare development project for ovarian cancer survivors as a part of the development research for cancer survivors led by the Cancer Survivor Healthcare Research Group. Ovarian cancer often requires very extensive surgery compared to other gynecologic cancer procedures along the adjuvant chemotherapy and long-term maintenance therapy. So the survivors frequently experience many physical and mental disorders such as lymphedema, sexual dysfunction due to the cancer itself and the treatment side effects. So the core objective of this research is to structure the symptoms that vary according to the extent of surgery and treatment stage in ovarian cancer patients and manage them through the digital platform. So we have planned and developed the 손난로, the healthcare application that can be used by both patients and healthcare providers. And the app is currently registered on the iOS App Store. So patients can self-report symptoms and their meals and medication compliance and move through the app. So while healthcare providers can review the data in real life through the web platform and incorporate into the clinical care. So from now, the 4 hospitals, including our centers, are currently participating in this project and validating this clinical effectiveness of the application through a multi-center pilot trial. To summarize, the gynecologic cancer survivors continue to experience various quality of life deterioration issues even after the treatment, and these problems require early assessment and structured management systems. So based on our ongoing healthcare application development research, we will continue to develop the practically applicable programs for clinical use and work to reduce the emergency room visit and unexpected hospital visits in the bad conditions through the lifestyle management through these applications. Thank you for the attention.
Thank you, Dr. Kim. Now we have 2 designated discussants for those topics. I'd like to introduce 2 discussants. The first one is Jung-Hee Kwon. Dr. Kwon graduated from Jungang University, and she was trained in Jungang University and Seoul National University, and she has been in Halim Medical Center and MD Anderson Cancer Center. Now she is working at the Jungnam National University College of Medicine, Sejong Hospital. She is a very renowned scholar, especially in oncology and palliative and supportive care. So Dr. Kwon, would you like to make some comments about those 3 topics, please?
Thank you very much for a nice introduction, and I would like to have some summaries and have some questions about three speakers. Well, these presentations looks a little bit different, but they're all the same because it looks like after cancer treatment and how patients cope day to day and how system and policies can better support them. So whether it's helping a patient enjoying a meal again, dealing with the symptoms that don't show up on a scan, or designing national programs, survivorship is about the whole person, not just the surviving cancer, but truly living after it. We need care that personalized, community-driven, and backed by smart system and policy. That's how we make survival seem not just a phase, but a supported, meaningful part of life. Dr. Matsuoka's team at the National Cancer Center looks like having a tremendous nationwide support to people living with cancer, and their approach combines policy, technology, and community-based care in their program called Living with Cancer. So I have two questions about Dr. Matsuoka's presentation. In your slide, the survey showed an increase in the suicide rate in the first year after the diagnosis of cancer. Is there any study to find the related factors to the suicide problems? And is there any specific programs to prevent those problems or help them to prevent the suicide by themselves? And second question is, from what I saw on the presentation slide, the budget for the research related to supportive care, palliative care, or community-based care have increased threefold. But I wonder what percentage of the cancer-related funding do those studies on symptoms account for?
So, okay, Dr. Matsuoka, can you
Thank you for
answer?
questions. First one is regarding the suicide prevention program in Japan. Now we just observe the suicide rate after cancer diagnosis and treatment. However, we don't have effective suicide prevention program. Now we are thinking about what kind of and how intervene is more effective to cancer patients. So now this is a challenge for us to prevent cancer patient to prevent suicide. So my answer is there isn't any effective intervention. And second one, sorry, I cannot catch up your questions.
Yes.
The budget.
So you said the funding budget about the living with cancer increased threefold before, and then I just wonder, there's a budget for whole cancer and
what cancer?
The budget about everything about the cancer, and then what percentage of the budget is support for the supportive care, palliative care, or living with the cancer, or end of life care, not the innovative treatment or molecular works or laboratory works.
Okay. You mean the cancer budget is the cancer budget portion of the whole cancer fund. Okay. Sorry, I don't examine the portion of cancer survivorship research, but it's not so big, just a little one. Just my impressions are about 10% to 15% of all cancer research found. And in Japan, we are now organizing the very big funding agency we called AMED, but we don't have cancer survivorship budget program for AMED because cancer survivorship research is the goal of cancer survivorship research is very is not it's fragile and soft. So the government is very difficult to decide the amount of budget. So we now the Ministry of Health Welfare having budget for cancer survivorship and NCC intramural, but it's not so big rather than and basic research on the treatment research, less than 10%.
And Dr. Kwon, can you continue? Yeah.
And then the second presentation about Dr. Juijo's research about the eating problems of esophageal cancer survivors. And this patient often struggled with eating for years after treatment, things like reflux, early fools, or just a general loss of appetite. And her team didn't just give a general advice. They went to people's home and talked to them and designed real-life solutions. Soft high-protein meals like porridge or sprinkles tailored for older patients. This work is about bringing back confidence and joy in eating, and that's something we often overlooked. And I have two questions about the research. And head and neck cancer is also associated with a high number of diet-related issues among clinical conditions. Is there any particular reasons why you choose to address these issues in the esophageal cancer patient instead of the head and neck cancer patient?
Dr. Chung?
So I totally agree with you. Head and neck cancer patients have a similar problem, but esophageal cancer, we actually did some other study before we conducted this study. We were interested in quality of life of esophageal cancer patients past previous years. So we found the diet was really issue of these cancer survivors. Then that's how we end up being. So in terms of response to your question, we are actually having a plan for expanding this study. Head and neck cancer and gastric cancer, cancer patients have a similar problem and similar issues.
And my second question is, the socioeconomic status is one of the biggest barriers to dietary intervention, as you mentioned already. But how do you address these issues when conducting a study? Not just helping them, doing by themselves or outside the study, how can they handle with these problems by themselves?
So I couldn't really introduce detail because of a time limitation, but we clearly see there is a gaps and disparity in terms of shopping and preparing food, actually supporting by somebody in related to their economic status. But I think we are very much focusing on average person and all, especially the vulnerable population for this program. But we clearly see the differences between people who are rich and people who are poor. Yeah.
Thank you very much.
Okay.
And about the third presenter, Dr. Jin Kim, her work is about the ovarian cancer survivors, and some works are about the endometrial cancer. And what's striking here is how many issues go unnoticed. Her team studied patients with low and swelling and found that they had worse quality of life, more stress, and more financial difficulties. Even when their sexual function scores look normal compared to healthy women, survivors were more likely to report vaginal dryness and less social engagement. This may seem like small details, but they matter. They affect how people live, how they feel, and how connected they stay to others. And it looks like she's building a new study with the application to collect the data about the survivorship of the ovarian cancer patient. And I have a very simple question about your study. Your study found that the sexual problems are important among ovarian cancer patients, and when you found that result, what programs have you offered your patient to address these issues, and to what extent have they improved this situation?
Thank you for the questions. Regarding the sexual dysfunction, the difficult one is we cannot consult the details of the sexual dysfunction in the real clinic. So with the application form, the patients can self-report about the details about the sexual dysfunction, so we can understand and we can identify the details of the symptoms and very structured. We can structure their details about the sexual dysfunction, so we can more deeply manage their problems when we from the patients in clinic.
So are you having a plan to have some specific clinic for those patients?
Not for the details, just.
Thank you very much.
Okay. We are the second designated discussant, Dr. Sujung Che. Dr. Che graduated from Gachon University Graduate School of Medicine, and she is now in the clinical associate professor at the Department of Family Medicine at Gill Medical Center, and she is from last year. She is the director of the Cancer Survivorship Center in the city of Incheon. Dr. Che, you can make some comments or questions.
Thank you for the introduction. Good afternoon, everyone, and we're finally towards the end of the conference. I am honored to serve as a panelist for this discussion. As I was introduced, I'm a family medicine physician at Gill Medical Center, and I serve as the director of the Adult Cancer Survivorship Center for the city of Incheon. My professional focus is on implementing and advocating innovative survivorship program. The majority of my patients in our center are breast cancer survivors. So I was especially pleased to learn more about survivorship in esophageal cancer and ovarian cancer today. So given the limited time of 10 minutes, I'd like to pose a few questions. Actually, I was going to ask 2 questions per speakers, but I will only ask 1 question per speaker. To enrich our discussion. Thank you, Dr. Masaoka, for sharing your extensive experience and knowledge on the challenges of cancer survivorship in Japan. It was especially interesting to learn about Japan's comprehensive government policies from 1984 to 2025, showcasing the government's evolving strategies to address the needs of cancer survivors. Actually, the stable increased government funding for survivorship research and development is actually commendable and something we hope our government adopts in the near future. So I actually have 1 question for you. What lessons can other countries like us learn from Japan's experience of cancer survivorship? Are there any tips for integrating the public health, the research, and patient services?
I'm sorry, I'm not sure what you asked me.
Any lessons that we can learn from your Japan's extensive experience with survivorship?
Yes, we need to push the necessity of the governmental budget or private budget to increase cancer survivorship research and practice for all. So the people in Japan and the funding agency also want to have a budget for basic science and clinical trial for big money, but we speak more loudly to need budget for cancer survivorship research. So because it's very important for all cancer survivors to have the life with dignity and health. So we need to push voice to government and founding agency.
Thank you. Thank you, Dr. Cho, for your insightful presentation and supporting esophageal cancer survivors in Korea. As always, your research is very interesting. I was very impressed with your anthropological approach towards your patients. So post-treatment, well, from what I understand, I hear approximately 3,000 new cases of esophageal patients, cancer patients happen every year. And post-treatment challenges such as difficulty swallowing, weight loss, and emotional distress can significantly impact their lives. And it seems like your pilot program demonstrated very positive outcomes with patients reporting like improved well-being, increased food intake, and reduced symptoms. So, which I find it very interesting. And so my question for you is, so what are the sort of like primary barriers to implementing your innovative program, and how can they be overcome? And is there any and what hospitals and health organizations can do to overcome these barriers?
So, I mean, there are many barriers, I would say. This is possible because we are doing this as a study. So if you're thinking about, you're actually competing not only, you're actually competing many delivery services and commercial services. Even though we said this is good for you, but there are many other food and competitors, I would say. They are saying they're the healthier and better. So that's the one of the thing. And another thing is patients have a perception about this type of prepared food. Because we already prepare the food and then ask them use microwave and reheat. And they said reheating might not be good. The patient says fresh food is always better. So patients has some, I would say it's a wrong perception and misunderstanding about nutrient. So that's the one of a barrier for making sure they eat it. So this is the reason why we actually show them how we produce it and how we deliver it. So we wanted to make sure the pouch is actually not really harmful for them, because whenever we show them deliver the food with a pouch, and they say the plastic is always bad. So these are the things we should overcome. But at the same time, if we wanted to scale up this to the public, it's all about the money, because we are able to do this study because of the funding from COCAS project. But if you don't find any company who is willing to produce this type of a product, I think we were not able to do it. But positively, we are living in the advanced aging society, so we think this type of food can be applicable to anybody who are older, not only the esophageal cancer patients.
Thank you. Lastly, thank you again for Dr. Kim for sharing your expertise in survivorship in gynecological cancer. I personally lost my mother with ovarian cancer like 23 years ago. So, and I still remember how limited information was available back then. So I'm truly touched by the progress we're making today. So your emphasis on the need for healthcare solution, as well as a crucial role for initiating early and structured survivorship program was very inspiring. And so my question is, it's actually a clinical question. So how should clinicians address issues of sexuality, intimacy, and so on in culturally sensitive ways? Because especially when survivors, they hesitate to speak up to their clinicians. So do you have any tips?
First of all, sorry for the loss, and I am a woman doctor, so I am better understanding of the sexual dysfunction for the ovarian cancer patients and also gynecological cancer patients. So I am more beneficial compared to the male doctors, and after the active treatment, I am asking about some important issues, including the lymphedema and pain, and also the nausea and vomiting, and also the sexual dysfunction for the married humans. So maybe but But even though my a little effort, the consultation time in Korea is very, very limited, so I usually cannot ask many questions about the symptoms for the patients. So it is really hard.
Okay, no further questions. Thank you, Dr. Chen. So all the three presentations and discussions were over now, but we have about 4 or 5 minutes left, I think. And if you have any questions or comments from audience, please raise your hand. And so we today in the last session we had 3 speakers, and the first speaker talked about the very broad range of national cancer plans and cancer survivorship care, and many Koreans was impressed by the triplet increase of budget funding in the field of survivorship and supportive care. It's very envious of that, I think. I think Dr. Matsuoka is endeavoring very hard to get more and more research funds from government and other resources. And next 2 topics on the specific intervention program for a limited group of cancer patients, and where we are also very impressed of their clever way of approaching, designing the program, and how to overcome the barriers. So it was very interesting session, I think. And finally, the chairperson, Dr. Chung, will close this session.
Well, I guess cancer survivorship care is very important since the mortality has been decreasing, and then especially ovarian cancer now with the more target therapy, immunotherapy, and then the survivor is live long enough. And I see some patients who live with on chemotherapy, third line or fourth line, and then they under treatment long time with the quality of life is very poor. So I guess our interest have to be expanded beyond who live after treatment, but along with the treatment. Thank you very much.
Thank you. So we are finished the last session.
Thank you, all the speakers, session chairs, and discussants. Please give them a big round of applause. Thank you. This concludes all sessions of the 17th National Cancer Center Korea International Symposium. We'd like to extend our heartfelt gratitude to all the speakers and participants who contributed to today's symposium. To officially close the symposium, I would like to invite Dr. Yoo-seok Chung, Chair of the Symposium Organizing Committee, to deliver the closing remarks. Please welcome him with a round of applause.
It's ending. Yeah. Thank you, everyone, especially for staying here until this late hour. And it's been such a great honor to organize and host this symposium. Over the course of the event, we've shared innovative ideas and meaningful discussions. I hope you enjoyed a lot, as much as I did. This year, 560 participants registered and joined us here or online. We were honored to welcome 5 international speakers and a wide range of researchers. Many NCC faculty members also shared their valuable insights, helping shape a truly enriching program. I sincerely thank the organizers and volunteers whose behind-the-scene efforts made everything run smoothly. And also special thanks to COCAS for their generous support and partnership. I encourage you to carry forward the ideas and connections made here. Let's continue to collaborate, innovate, and rise to the challenges ahead. Thank you again for being part of this memorable experience. We look forward to welcome you again next year with another exciting topic. Once again, thank you very much.
Thank you, Dr. Yusop Jung, for your closing remarks. This concludes today's symposium. We kindly ask all the speakers, session chairs, and department heads to proceed to the bus boarding area in front of Xingguan for transportation to the dinner venue. Our staff will be available to guide you to the boarding location. Thank you once again for your participation.